Hello, hello. We're gonna kick off the event. It's a full day event. I firstly appreciate everybody's time, especially our guests traveling from everywhere, all across the country. And we also have speakers dying from different countries today. So let's try to keep the time. Without further uh, delay, I'm gonna kick off the introduction section. Firstly, it's my great honor to welcome you all to the third iXCells Rare Disease Symposium. Uh, February is the Rare Disease Awareness Month. This year, we are extremely honored to have a stellar lineup of speakers from pioneering parents, prominent researchers, and thought leaders in the industry to share their experiences and insights about patient-focused therapy for rare diseases. And we are excited to have you joining us to learn how we can join forces to tackle the debilitating diseases and advance the personalized medicine through the collaborative ecosystem. Rare disease is becoming a global public health crisis. By definition, a rare disease is a disease or disorder affecting fewer than 200,000 people in the United States. Based on the recent reports by Global Genes, there are about 11,000 rare diseases today, and this number is increasing rapidly every day. About 25 to 30 million people in the United States, or 350 million people globally, are affected by rare diseases. So the rare diseases are not rare as many of us feel. Indeed, the rare disease impact more people than cancer and AIDS combined. Moreover, 50% of the rare disease are affecting the young children, and most of these conditions results in the premature deaths of infants and young children. 80% of rare diseases are genetic. However, 90 to 95% of rare diseases do not have an FDA-approved treatment. So there are many challenges for the long tail situation listed in this slide. Developing drug for rare diseases like climbing mountains, an immense challenge that demands resilience, strategy, and unwavering teamwork. Just as a climber navigates shared rock faces, setbacks, and unpredictable conditions, researchers and innovators must overcome scientific, regulatory, and financial obstacles. Success requires bold vision, relentless perseverance, and a network of experts working together to reach the summit, a life-changing therapy. So we're going to hear a lot of stories today about collaboration and resilience. At iXCells, we have been collaborating with numerous rare disease foundations by offering the preclinical development services. So with our expertise in human cell models, especially iPSC-derived models, we help the foundation to establish the patient-specific cell models, 2D and 3D. We also help with the antisense oligos or drug repurposing screening. And with all these cell models we're offering, we're also helping with in vitro toxicology testing, in vitro efficacy testings, especially phenotypic screening. So over the years, we have accumulating a list of assay-validated cell lines and antisense or compound controls for some of these assays. And here is the list of some ongoing and completed ASO screening projects. These projects use different ASO design strategies, including allele-specific knockdown, gene upregulation, microRNA or long non-coding RNA targeting. We also help the foundations to select their suitable cell models for the screening. For example, immortalized cell lines, which is good for primary screening, and primary neuron or patient fibroblasts, iPSC-derived cells are more suitable for downstream target validation or dose-dependent curve testing. And these are, here is a list of our repurposing drug development projects. Most of these projects are under collaboration with Rare Base. We also have a base editing project with N of 1 labs or Uncommon Therapeutics, trying to develop a gene therapy. Last but not least, we have helped many foundation leaders to make their patient-derived cell lines, including iPSC lines and fibroblasts. We are also working with several foundations for cell repository services. So by making these cell lines readily accessible to the research community, we are hoping to attract more researchers to study these ultra-rare genetic disorders and eventually accelerate the drug development. So over the past 4 years, we have been establishing a strong partnership with a growing list of biotech companies, nonprofits, and patient foundations. These Q phases of our patients are the most powerful fuel for our team working day and night to accelerate the life-changing therapy for those who need it most. And we are happy to see some of these foundation leaders joining us today. We truly appreciate your trust in iXCells team. That means the world to us. Thank you, Leo. And this is our today's event agenda. There are 4 sessions. First session is parents' perspective. Second one is the modeling rare disease using patient-specific cells. Number 3 is the industry collaboration. And number 4 is our special section this year, which is focused on the ALS research. And there are 15 minutes break in between of the session, and there are 40 minutes lunch break. And after the whole event, we have the social networking, and you are more than welcome to stay and interact with our invited guests and visitors today. So our first section, so we invited 5 pioneering parents to share their experience and perspectives as the driving force for patient-focused therapy. All of them are my favorite heroes. And they all have very distinct stories behind them, but they have the common feature or characteristics, which is never give up. So our first parent speaker is Yiwei She. Yiwei is a dedicated mother and advocate. Upon her son Leo's diagnosis with ultra-rare neurodevelopmental disorder caused by a TMPO2 gene mutation, she took extraordinary measures to find a treatment. Despite being told that nothing could be done, she founded the TMPO2 Foundations, and through collaboration with biotech companies, they developed a personalized medicine, a personalized antisense oligotherapy for Leo within just a year. Encouraged by Leo's progress, Yiwei expanded her mission to assist other families facing similar challenges. With her background as a machine learning scientist and mathematician, through initiatives like Project Baby Lion, Yiwei aims to revolutionize the diagnosis and treatment for ultra-rare disease in children, striving to make personalized medicine accessible and affordable for all. In a recent initiatives, Yiwei and I are exploring the opportunity to collaborate with hospitals and patient foundations from China through the large patient population in the country and China's investigator initiative trials or IIT program. We are hoping to accelerate clinical practices with more flexibility. Without further ado, Yiwei.
Thank you, Nianwei. Thank you for that introduction. And it's a pleasure and an honor to be here speaking to you all. So I will be telling you about my son Leo's journey and focusing on the part that everybody I hope is most interested in is how do we get a drug made for a baby? And as Nianwei said, it's a very collaborative effort. So these are just a small number, these are the leads of our teams, but there's a lot of individuals that helped in the effort. Our principal investigator, Dr. Nicole Colfell at Rare Disease Children's. Oh, not this pointer. Okay, thank you. Dr. Colfell, Dr. Dimmick, who was the chief medical officer at Crayon, or former, and then Chris Hart, also the former CEO of Crayon Bio. And there were a lot of collaborators at UCSD as well. So here's Leo when he was born. And I will spare you the diagnostic odyssey because I think everyone here is very familiar with it. But there were signs at birth. And we got Leo sequenced when he was 3 months old, getting the results back when he was 4 months old. And the paper, so he had to be sequenced in a clinical trial. So sequencing and diagnostics is still not widely available or accessible, and this is a problem at a systemic level. But when the clinical trial for the diagnostics came out, there he is, the lone TMPO2 patient, in the study to kind of find genetic diagnoses for infantile onset epilepsies. And the incidence rate, the prevalence is extremely high. So almost every baby, I think that the incidence rate is up now to 80 to 90%. So many babies with epilepsy have a genetic etiology, and so that should be a first line tool when you see a seizure in a baby. So that set me off. So people told me there's nothing you can do, and that didn't seem to be true to me. And even if it were, I was not going to let it stop me. So there's also this thing that brain development happens, for young children, for babies and young children, and it's a very, very fast and dynamic process, and so much is going on from even in utero, but also after birth. All of these things are happening concurrently. So we were on a race against time, and our child, luckily for us, he was not terminal. He did not have a neurodegenerative disease. Nevertheless, his quality of life was on the line, and it was the quality of life for the rest of his, possibly 8-9 decades of life. So we wanted to make a drug for him as fast as possible. So there was a crash course in drug development, and this is what the process looks like. So there's the basic science, there is preclinical work, then there's clinical work. And this is the design of the FDA and the system that we have in nowadays. And then after phase 4, there's all of this commercialization stuff, which actually is extremely important for accessibility and equity. Right. So for me, I thought, okay, what is this all for? I don't care. I just want a drug in his brain ASAP. But then, looking backwards, in order for, every child who needs one of these drugs to benefit, we do need to find a way forward for this step, and also, for every single step in this process. So how do you make a drug? Well, we found the cutting edge partners. So Crayon Bio is an AI first, drug discovery and development company. And with my background in machine learning and AI, I was able to read their patent and do the due diligence and have enough confidence in this company that had never made a drug before. But their patent looked, solid. And so we signed a contract with them and we started working together. And we also, started the whole system of model organoids. So this is the other half of the drug development process, is trying to find, a model of the disease in something other than patient, so that you are not taking the risk with the patient. And so we didn't make Leo yeast, we made Leo worms, we made Leo flies. Actually, Leo flies were already made, but we had to make, more faithful Leo flies. We made Leo mice, and we turned down a proposal for Leo monkeys. They were not only extraordinarily expensive, but then you have to, once you have the monkeys, you have to take medical care of the monkeys, which is also actually very, difficult. And the most promising, and then what ultimately succeeded, were Leo brain organoids. And so we were very, very thankful to the Motri Lab and its collaborators. But these organoids, they come from iPSC, so they come from stem cells, and so the first step is to make the stem cells, which iXCells is a great a resource for. And on the other side, concurrently we were making the leads, and we were qualifying the leads, and we were going through. This is a standard workflow for ASOs. There's a different workflow for different modalities, but the shape generally looks the same. You start with some leads. This number might be millions for small molecules, or smaller for gene therapies, but then you select down, you try to design rationally when you can and engineer when you can, and apply AI when you can. And increasingly, AI is more and more applicable and effective at each stage individually. And also for ASOs, the company that we worked with, Crayon Bio, they could, out of 100 leads, they could select over 80 of them were safe in kidney and liver tests. So that is a very strong bias towards safety, even at the first step of the screening process. Our drug necessitated allele selectivity. That is a technical point I won't go too deep into, but we found a drug that could hit the target with a very beautiful, dose response curve. So this is what it looked like. This is the Gantt chart of everything running in parallel. And this was, we wanted Leo to have the best drug, as fast as possible. And there's a lot of failure. So there's a lot of hedged risk. So I guess, it's not, I don't want to say failure. I want to say, we mitigated a lot of risk. And so if things happened differently, we would have still gotten to, our end result within about the same time frame. And that is what a significant amount of resources were expended on. So ultimately, the disease model, actually, so everything other than the top two is disease models. And all of these disease models, they all failed for different reasons, except for the the iPSC-derived organoids. So we barely succeeded in some way. So this could have also failed, and then we would have had no disease model. And then the question would have, or the decision would have been, do we go into the human? Do we go into the clinic with no wet lab backup data and only on the back of some bioinformatic data? And that is, that is a harder question to answer than what we had to ultimately answer. And so here is, thanks to the Motri lab, this is the on the right-hand side, you can see that these are the Leo brain organoids. The vertical axis is the diameter of the brain organoids, and we see that his microcephaly phenotype is recapitulated in these brain organoids. And this is actually, these brain organoids are a very faithful representation of late fetal brain development. And so in all cases of microcephaly, which occurs with an incidence of about 1 in 10,000, this is an excellent, disease model, a place to try to find a disease model. And then our rescue data also came back to be very, promising. So the red line is the control. So, this is what happens to the brain organoids if you don't apply anything. And then we have 3 different ASOs at different concentrations. And we have 2 time points. And we see that ASO B in particular has very good performance because we are not only clearing the 50th percentile, there's a big jump in the 50th percentile, but there's also a big jump in the 25th percentile. And I think actually we got our dosing a little bit wrong. These 5 to 10 micromolar doses are a bit too high. Sorry, I'm jumping into the technical. I'm a scientist by training, but this rescue data was extremely promising and encouraging for us. And it came in January of 2023. So this was after we had the leads. Yeah. And ASOC, it was a control ASO. So this is just a scramble and a non, it's not supposed to do anything. And we see indeed that, it doesn't do anything. And in fact, it's a bit harmful at the highest dose. Okay. So with that, onto the clinic. Well, so between January, this is in January of 2023, and July of 2023, there is the 6 months of applying to the FDA. So, that is one of the problems in this space, is that we have safety data in non-GLP trials. We have efficacy data in patient-derived cell models, but it took 6 months for us to get into the clinic. And something about that risk-benefit, outcome doesn't sit right with me, because Leo was having just, sometimes up to a dozen seizures a day. And often he would stop breathing during these seizures. So there's a conversation to be had with FDA about how they regulate these therapies. And so here is our clinical protocol. We doubled every month during the escalation phase, again, for safety. And then after that, we did every 3 months at the maintenance dose. This is again a science slide, but it's trying to find the pharmacology. So trying to figure out, are we hitting the target in the CSF, in the brain? And this is a assay that measures RNA in the CSF. So there's actually a tiny amount of RNA in the CSF, and it turns out there's maybe five molecules of the TMPO2 transcript. This was 200 milliliters of CSF. So there's a very minute amount, and we tried to measure it, and it's incredibly noisy. So, at one point there was 100% of it was the mutant RNA, and at a different point, only 25. So only like 1 or 20%, 1 in 5 of the transcripts. So this is, we tried our best to measure it, but it's very hard. And, this is one, indication of the challenge of trying to do these trials scientifically. Next slide. There. Okay. So seizure data also came out to be very promising. We saw the decrease in seizures, especially these dark, severe grand mal seizures right away. The second thing we noticed was that the seizures would recur about a month in to the 3-month maintenance cycle. And I think the issue there again is kind of it's both the FDA and our lack of pharmacology study before we went into the patient. One thing that helps FDA applications is if you hew to precedent, then they will approve you. One thing that does not help you is if you're proposing something new, and then you might get bogged down into a conversation. So we hewed to precedent, and we proposed a protocol that was that had been submitted and approved many times. And so, this between these two doses, this is when we tried to have a conversation with FDA about increasing the frequency, which they did approve. So thank you, and kudos to them. So we are now dosing every 6 weeks, and we have been seeing increased, seizure control as well as developmental progress. I should skip this. And yeah, so the last piece is, the thing that we as Leo's parents, thank you, as his parents wanted most out of the clinical trial was not seizure control. It's neurodevelopment. And so this is one of maybe the other half, or maybe the main challenge is how do you measure, childhood development scientifically at an N of 1, scale. And so some of the things we did, so these are the successes, right? So we got a new milestone, which is he went from not having head control to being able to sustain a tripod sit, which is, a significant improvement. So he can't get himself into the sitting position, but if we put him there, he can balance himself. There's less spasticity. This is both an objective and a subjective measure. So with AI and newer technologies, we can, have objective metrics for spasticity. He has new social emotional developments where he will laugh and cry with his sister. So the crying was very easy to notice. Before, I had one child crying at a time, and then all of a sudden, whenever I had one child crying, within 5 seconds, I had a second child crying, and it was very fun actually to have this experience because I knew what it meant. Strabismus, a lot of these children with neurodevelopmental diseases, there is some manifestation in the eyes. And this is another opportunity for an objective metric. And again, the developments in AI, especially these multimodal models, can make, a clinical measurement of this kind of a symptom, objective, cheap, and easy. You just have to tell the model what you want, and it will measure it for you. This is really hard, but we noticed, a big jump in responsiveness and presence. He went from a baby who sometimes, we would ask, like, is he in there? Is someone in there? And I know a lot of parents have this experience to a baby. We think, yeah, there's definitely someone there, and there's something he wants, and and all these things. And in terms of there's something he wants, he will attempt to reach for toys. He will interact with toys. He will interact with sounds. He will notice cause and effect when he makes a sound. And we are really excited about the new sounds that he's making himself. And since the increase in dosing frequency this past summer, we've noticed there's less regressions between doses and some new sounds that are consistent. So I think, in terms of development data, in terms of clinical data for childhood development, I don't have a complete answer, but the tools are becoming available, right? So there are tools for getting biomarkers for EEGs, for blood sampling and other, biomarkers that you can take at higher frequencies. So there are, there are real, blood droplet tests, which you can send to like, say, the Snyder lab, and you can measure hundreds of different biomarkers in the blood. And there's now, dry scalp EEGs where you can measure, at a high frequency, at a high resolution, so that you can get objective data from a single patient, and it can power a whole trial from just a single patient. The data from a single patient, let me say it this way, the data from a single patient can power an entire trial if you take the measurements frequently enough. Again, acknowledging the team. And the takeaways, if nothing else, I hope you take away from this that the neurological symptoms are almost always genetic. So doctors are somehow behind the curve on this. The parents know this, and we should somehow inform as many doctors as we can. AI is becoming ubiquitous, and it's really good for rare diseases because it's incredibly deflationary as a tool, and the economics of rare disease has always been the biggest barrier to therapies. And so AI at almost every single stage of the pipeline of drug development, from the literature review to the discovery to the development to the clinical trial to just clinical care and practice, is it's entering everyday practice. And I hope that rare disease parents, advocates, researchers can leverage the productivity and scientific gains that are now available, and find the most creative ways to apply it to our field and our children. Yeah, our partnerships were critical. Our partners, all of the people who were part of Team Leo, we couldn't have done this without any one of them. And the standard of care needs to be better for rare disease patients. Standard of care does not cut it. And again, this is where AI can outshine human doctors and human knowledge by so many orders of magnitude that I think it's enough. The change in quality is enough to overcome the inertia and the other barriers towards adoption. Being this end-to-end process, so we know we have all of Leo's patient information thanks to HIPAA. We have all of the costs of his care, and this was the cost of standard of care before the trial, right? So this is what was billed to insurance, and this is not even including any of, say, the lost work time for the parents. This is not including the reduction in quality of life. Leo, had he been a healthy baby or treated earlier, he could have been a productive member of society. That is an economic cost. There's all this therapy that the state provides him. There's caretaking for probably the rest of his life. And so these are, when you add this up, it comes out to millions, maybe $10 million. I don't know, but it's a number that is not so much less, maybe the same as the cost of the drug development program. And so, when we as a society and our representatives and our policymakers and all of these people think about this, maybe the economics of creating drugs one at a time, one patient by patient, they're not so far off from break even. And if that's true, then why aren't we doing it that way instead of, you know, dumping a lot of this money into anti-epileptics and all of these, you know, these specialist referrals that ultimately really did not have any impact on his quality of life. And last, so I am a mathematician and a machine learning engineer. So this first principles concept, you know, trying to do things from first principles, this is kind of a core part of who I am. And that is how I went into this project when he got diagnosed. So all of these are kind of, if you were a scientist and you wanted to solve a problem, you know, you might not start with, well, this is how things have always been. You might start with, what are the constraints? And, you know, are there any constraints? Are there any, you know, laws of nature that prevent Leo from being dosed in 2 weeks? No, there aren't, right? What are the constraints? There are regulatory constraints. There are operational constraints. There are, you know, there are economic constraints. But these constraints can be overcome in ways that laws of nature cannot be overcome. These are human constraints. And we as fellow humans can work together to remove these constraints so that in the future, one day, maybe in the near future, these children can be treated within weeks of being diagnosed by a drug that is safe and efficacious, and the whole life can be recovered and rescued. And that is a vision that I deeply believe in and would like to work and am working towards. And, you know, technology and the advancement of science will make this future possible. I think that's it. Yes, that's it. Thank you so much for your time.
Thank you, Yue. I saw there's some questions pop up from remote, but we do have a Q&A section after each session. So there will be 20 minutes on Q&A after that. So we were gonna hold on that. So really appreciate Yue's experience, sharing her experience. So I also appreciate your squeeze your time. I know she's gonna go rush to Lady Hospitals for the next dose for Leo. So, and the whole event is pretty much scheduled on this day because of you. And thank you. That means a lot for us. So our next speaker, Michelle Jeanette. She is also one of my favorites, really favorites. The foundation leaders. She is the founder and president of Cure KCNH1 Foundation. Michelle is a passionate advocate with a crystal clear mission. Her goal is to create a meaningful impact by raising awareness about KCNH1 genetic disorders and positively influencing the lives of children and families. And she is unwavering in her commitment to drive research, constantly seeking improved treatment options and holding on to the belief that a cure is within reach. With relentless efforts, Michelle has established a collaborative network of partners, including Lauren, iXCells, and Parora, and many other organizations for developing therapies for KCNH1 kids through different roadmaps. Without further ado, let's welcome Michelle.
All right. Hi, everyone. I hope I can get through this without coughing. I had pneumonia over Christmas and my lungs are still not anywhere close to recovered. So I want to share a bit about our family story. These are my 4 beautiful boys. I had always wanted a big family. You know, I dreamed about all the normal things like camping and hiking and sports games and movie nights and beach days and family dinners. And we were lucky to have 3 healthy boys. And then Tristan came along in November of 2019. And I took one look at him and I thought something looked a little off about his eyes, but none of the doctors or nurses mentioned anything. So I didn't think anything more about it until he started missing developmental milestones at about 4 months old. We got him into PT and OT, and we got referrals for genetics and neurology. And the first rounds of genetic testing came back negative, and he had clean EEG. So we figured, you know, it must be some mild thing, minor thing, and with lots of work, he can catch up. And then he was about 15 months old when we got his genetic diagnosis, and it turned our lives upside down. He was not that far behind at the time, and so we had no idea how severe it would be and how it would essentially change every minute of our family's life forever. And then about a month after he got his diagnosis, he started having his first seizures, and that changed everything again. We are lucky that Tristan is relatively stable compared to a lot of rare disease kids, but we have still had numerous ER visits and ambulance rides and a couple of hospital stays and canceled trips and really hellish medication side effects that he's had to go through. And in addition to all that, it's all the everyday other effects of rare disease on our family's life that are really, really hard for us and all the other rare disease families. I picked this picture of our kids at the beach because I think that's just about the one time in Tristan's life that we have all been able to enjoy an activity together as a family. The stars aligned and he was actually enjoying the water and the temperature was okay. And he actually had fun, which was amazing. But normally he basically can't participate in any normal activities or he doesn't tolerate them. And so it means that we always have to divide and conquer. Somebody has to stay home with Tristan or we have to line up a sitter for him. And it breaks our hearts that he can't participate more in life. It's also really devastating that he spends 20 hours a week in therapies and he makes essentially no progress. I don't think he's learned a new skill in almost 2 years. He spends holidays zoned out while our other kids are, you know, tearing open their presents. And one of the most stressful and heartbreaking aspects is that he just constantly cries and screams, and no matter how hard we try, we can't figure out, we don't know what's bothering him. He's completely nonverbal, so we don't know what's bothering him or how to help. And it's extremely heartbreaking to see him and stressful on all of us and our other kids. So as soon as Tristan was diagnosed, I decided I was going to do everything possible to create a better future for him and for our family. So I started reaching out to researchers all over the world. We started the process to create a foundation. We started fundraising. I started doing a million things that I never thought in a million years I would be doing, like learning molecular biology and electrophysiology and neuroscience and grant writing and event planning and website design and meeting with local leaders and advocating at state and national levels, public speaking. And we've really come a long way. I want to back up a little bit and give some context about our disease. KCNH1 related disorders are an ultra rare developmental and epileptic encephalopathy caused by mutations in the KCNH1 gene, which encodes a potassium channel primarily expressed in the central nervous system. We have about 100 cases diagnosed globally, and it's very similar to a lot of the other DEEs characterized by intractable epilepsy, profound intellectual disability, severe movement and speech disorders, autism, GI issues, feeding difficulties, sleep disturbance, and then we have a couple kind of unique features to KCNH1, which are gum overgrowth, which requires repeated surgeries to address, and smaller missing finger and toenails. Most of the mutations studied have been shown to be de novo variants that cause a gain of function, so the channels open too easily and stay open too long. And KCNH1 also has a connection with cancer. 70% the gene is overexpressed in 70% of tumorous cancers. And so it's possible that any of our treatments we develop that either downregulate the gene or inhibit the channel could potentially help cancer patients. Our foundation is really laser focused on getting translational treatments to the clinic. But of course, to do that, we also need to do all these other things in parallel. We need to generate models. We need to collect patient data and establish the natural history of this ultra rare disease. And we need to organize and engage the patient and professional community, and of course, raise money. Sorry, it's got a lag. I can't tell if it's gone or not. Oh, okay. So when we started our foundation 3 years ago, no one was actively working on translational treatments for KCNH1. We've made huge strides, and we now have multiple modalities in development. We have a repurposed drug that Tristan and a few other patients have tried with mixed results, some having great benefit, some none, some adverse events, not serious, but and side effects. Tristan has experienced some benefit, but nothing life-changing. We funded a repurposed drug screen and identified a couple promising hits that now need to be validated. We designed, manufactured, and screened a batch of general knockdown antisense oligonucleotides, but what we're most hopeful for Tristan is that he was accepted into the NLORM program for the development of an allele-specific ASO, and I hope we might be hearing a bit about that later today from Nadina. We have academic and industry partners working on novel small molecules that are potent and selective channel inhibitors. And one thing that I'm really excited about for the long term that theoretically is the most curative option is base editing. And our foundation just received a $100,000 grant to fund a pilot base editing project. And the preliminary data is really exciting. We have a couple highly efficient editors. Okay, our disease models, basically from the beginning, we started working on disease models. And we now have simple cell lines, we have iPSCs, we have neural networks, we have cortical organoids, and we have a couple mouse models. But everything takes a while, and with delays and various issues, none of our disease models are fully completed and characterized other than the simple cell lines. And so we haven't really gotten to that place where we can put all of these exciting treatment candidates into our disease models and test them. The theme of today is collaboration, and we have been so lucky with our foundation to have numerous academic and industry partners who have been super collaborative and wonderful. But I know a lot of my friends in the rare disease space have not always been so lucky. And so our priorities as patients, of course, are urgency and involvements. We need better treatment options for our kids now. They don't need to be perfect. We know they won't be. We know there will be risks and that there could be adverse events, but we are willing to take those risks because our kids are suffering. And we ourselves, personally with Tristan and countless friends in the KCNH1 community and other communities, have encountered so many barriers in just trying to try repurposed drugs. So for example, Tristan, the drug that he's on, is on label for pediatric populations for bedwetting. You can hardly imagine a more benign indication. And yet we jumped through hoops for a year. We did all kinds of in vitro tests, medical tests, developmental tests, consulting everyone and their brother, all of whom were supportive. And then the neurologist that we had specifically been working with, because she's in the translational space, got cold feet, didn't want to prescribe it, and it was devastating. So, not all families are going to want to take risks, and that's fine. But for those of us that do, we need advocates and allies who are going to be supportive of that. And then, of course, we all know that it's very important to involve the patient communities in clinical trial design. It leads to the selection of better endpoints and outcomes and more successful clinical trials. Some of the things that have been really wonderful with all of our partners that we've worked with is communication. You know, when the research you're working on literally determines your kid's future, just being kept in the loop, even if there's no news, makes all the difference. And then focus on the big picture, share everything possible. We've had partners who have been so generous with their time and their info and their assets. So, you know, don't let competition get in the way of progress. And then take the time to invest in the patient community and get involved. Show up at our research roundtables and our events and even our fundraisers. This builds trust with the community and it motivates everyone. You know, I've heard so many times how meaningful it is to researchers to get out and see the real world impact of what they're working on. And likewise for the patients, it makes all the difference to meet the people who are working on their kids' disease and build those connections. So yes, it takes time, we're all busy, but I promise you it will be worthwhile. I think we all know many of the benefits of collaboration, you know, access to the patient community. We've hooked up our researchers with many patient samples, data collection, clinical trial recruitment. I do not enjoy being a professional nag and hounding people down for survey completion or lining up a blood draw, but I will do it and I can help take that burden off of you and probably be more successful at it because I have those relationships. Of course, working together, meeting regularly, breaks down data silos, reduces duplication of efforts, and avoids waste of our minimal resources. We've been really successful in working with our scientific advisors on funding applications. So we've submitted letters of support and patient cell lines that have helped some of our academic collaborators win multi-million-dollar awards. And in turn, our scientific advisors have helped us, you know, edit and review our grant applications and very much helped us win the really large CZI grant this past fall, as well as the $100,000 one for the base editing project that then we, you know, grant back to a lot of these researchers that we're working with. So it's a really mutually beneficial relationship. And then of course, again, meeting regularly really helps everyone optimize their research protocols and, you know, get questions answered quickly or come up with new insights that you just wouldn't, you know, likely send an email. But if you're all connecting on a regular basis, it can really help accelerate things.
Hi, my name is Hazel, and I have KCNA1.
Can you say hi? Say hello. Hi.
Okay.
Hi. We call him Daddy's tail because he follows him around everywhere. And he's trying to catch up with me because he'd rather be holding my hand.
Sterre may not be able to walk independently, but she knows how to use her new walker to get what she wants. She even managed to make off with everyone else goats at school one day.
She smiles in the face of adversity.
Tristan is the sweetest, most loving little person. He's always toddling over to lay his head in our laps or grab us as we're walking by.
Casey and H1 affects everything about Joseph. It makes him who he is. And it affects obviously everything about our lives as well. Our lives as a family are maybe not what we thought they would be when we had our little boy all those years ago.
Mais c'est ça, malgré son âge de 16 ans, c'est que elle a quand même elle a un âge mental de 8-10 mois, ce qui est très difficile. Elle fait rien toute seule, elle dépend de nous dans toutes les sphères de sa vie. On l'appelle notre bébé géant d'amour. Fait que c'est sûr, s'il y avait un médicament qui pourrait alléger, faciliter plus son quotidien, puis notre quotidien, mais c'est ça, ça serait, j'aimerais vraiment qu'elle ait plus conscience de ce qui l'entoure. Qu'elle puisse nous dire je t'aime, je t'aime, je t'aime, je t'aime. Que j'ai l'impression qu'elle nous reconnaît, sa famille. J'ai pas l'impression qu'elle nous reconnaît. Puis ça, ça me fait de la peine des fois. Tu sais, mettons qu'on va la chercher à l'école, on arrive à l'école, c'est comme si on était des purs étrangers, là. Elle semble même pas nous reconnaître. Ça, je trouve ça difficile. Je trouve ça vraiment difficile.
Onun gelişim basamaklarını tamamlayamadığından itibaren hem özel eğitim hem fizyoterapi aldırdık, ancak ilerlediğimiz yol çok uzun bir yol olmadı. Bu yüzden de dünyada yapılan bilimsel çalışmalardan da umudumuz yüksek. Belki geliştirilecek bir ilaç ya da genetik bir tedavi Melih'in yürümesine, konuşmasına ya da farklı bir şekilde iletişim kurmasına.
My brain sends a message to my mouth that my mouth doesn't get.
I would say that the most difficult part of having a child with this disability is the unknown. Bentley could have just, a wonderful start to his day like he did today, a little bit tired because he decided that he was going to stay up all night, to, having a full-blown grand mal seizure this morning, which is very scary, very traumatic.
The hardest part, which is Rafi's seizures. They're extremely scary. My husband and I feel often very helpless because there are no triggers, many times no warning signs, and even at 8 years old, they're really uncontrolled.
Mi mayor esperanza, me diga lo que quiere, lo que siente, lo que le gusta.
So if we could eliminate seizures altogether, that would be a dream. Our hopes for the future and with future treatments are for Bentley to have cognitive growth. To us, it's more important to have that cognitive ability than physical abilities. So if treatments could help with that, that would be a dream come true.
love for Arisa to be able to understand us and talk to us, but in the end, it doesn't change the fact that she is our sunshine.
Thank you all for learning a bit about the KCNH1 community. And of course, this is representative of all the communities we serve. I just appreciate everyone being here and all of the work that we're doing together to improve the futures of the rare disease community.
Thank you, Michelle. Every time we're here, Michelle's story is very, very touching. We've been working with her for many years, and her story has been inspiring, not only my company and also my families. And today I have two of my children here today, Laura and Andy. So sometimes they have the school assignment to ask their parents, what do you like about your job, right? So I tell them, right, it is the mission, and there's so many things in front of us, right? So, and we're lucky that we're in an era that there's so many scientific breakthrough in front of us: gene therapy, cell therapy, iPSC, and CRISPR. So many things that we can potentially bring those, which is, personalized medicine, which is unbelievable a couple of years ago, but now it's becoming, we already bring this treatment to life. So without further delay, I'd like to introduce the next speaker. His name is Piotr Kosla. He's dialing from Poland. So he's a proud father of 3.5 years old twin girls, Lena and Zuzia. Piotr comes from Poland, and where he currently lives with his family. His professional background is finance and international management. He graduated from Warsaw School of Economics and National University of Singapore. Before joining Biogen, he spent several years in finance and business development in several big firms. 3 years ago, Piotr's daughter Lena was diagnosed with the ultra-rare genetic disorder, PACS2 syndrome, PAX2 syndrome. He and his wife founded the PAX2 Research Foundation, driving treatment discovery for about 150 people affected by this disorder. In 2022, he reached out to us for iPSC generation for his daughter without knowing too much background of this disease. And ever since then, I've been following his journey in developing the treatment for children with PAX2 condition, and was super impressed by what he has achieved with only 3 years. And today we are going to share he's going to share his journey with us. Peter, can you dial in?
Yep. Hello, everyone. First of all, can you hear me and can you see me? That's the most important.
We can hear you. We're working on a slideshow.
Okay, perfect. So let me first. Okay, I'm trying to share my screen. Just wait a sec.
Yes.
So as long as you share the screen, I cannot share my screen. So you have to stop sharing the screen.
Yes, hi, Piotr. I just stopped sharing.
Almost there.
Perfect. Okay.
Let's see how it goes.
Perfect. We can see that loud and clear.
Okay, that's great. Great, that's great. Thank you. So first of all, welcome everyone. Good morning to everyone. I guess it's around exactly 9 a.m. On my side, it's 6 p.m. So I'm dialing in, as it was mentioned, all the way from Poland, which is 1,000 miles away. But it doesn't stop us from collaborating together with iXCells and with many other parties, which I'm going to mention during my presentation. But before I get there, I wanted to say a big thank you to Michelle for having a great and very emotional presentation. And she always inspired me as well in my job around the PAX2 when I see what she's achieving and what steps she's making with KCNH1. So thank you, Michelle, for that. And I hope at some point we'll get to meet each other in person. Because for those who don't know, we've met like 2 years ago. And every now and then we update each other, how we're doing. And I'm always inspired by amount of motivation that Michelle is having to push the treatment development further for all the kids with the KCNH1. So thank you for that. Moving to our case. So today I want to talk for, let's say, 15-20 minutes about the journey we did in the last 3 years when we're starting completely, and I mean it by saying completely from scratch, when there was like no interest around the PAX2 syndrome at all back in 2020-22, and where we are today after 2.5 years, to be very specific, from the moment we decided to do something about it. So as it was mentioned, me together with my wife, so my name is Piotr Kosla, and with my lovely wife on the left-hand side, Małgorzata, Gosia Kosla, we do have some background, which in fact helped us to, let's say, start a bit faster than some of the other parents that decide to take care of developing the drug for the rare diseases, because I have some medical background, as well as my wife. However, both of us, we finished business schools. We didn't study medicine. We didn't study biotechnology and any of that. So we had to learn very, very fast. But something I'm gonna link during the presentation. I'm going to link the fact that the fact that we finished the business school helped us a little bit at the very beginning to, let's say, to manage a project, because at the end of the day, I guess every drug development is a project that it also requires specific competences to push it further. And on the right hand side, you can see our reason. So Lena, that today she's 3.5 years old, but she has twin sister, which is very important here, also from the research perspective. They are fraternal, as you can see also from the picture. But in fact, it allowed us to move quite fast with the cell painting research, which I'm going to talk about later on, and how Zuzia cells were beneficial in advancing the treatment development for PAX2. But starting from the very beginning, so basically our story. So before December 2021, we're a normal family with two kids, with twins, which were back then 3 months old, and we didn't see it coming that at some point Lena will have the first epilepsy, which very very moved into the status epilepticus because of the fact that they were twins. Some of like Lena, she had some issues with the tension in the body, together with Zuzia, but it was always explained by the fact that they are twins. So as simple as that. So, and apart from that, everything was fine. So if you look at Lena when she was 1 month old, 2 months old, 3 months old, you wouldn't tell that she might be having some devastating disease in front of her. But in December 2021, during the holiday time, she experienced her first epilepsy, which followed by 30 plus in 48 hours when she was in the status epilepticus in the hospital. And then quite quickly, we've managed to diagnose her thanks to whole exome sequencing in February 2022 during Valentine's Day. So it was exactly 3 years ago, as we have 18th of February today. And we quickly learned that she's going to face a very rough and unpleasant future because of the clinical phenotype that was described in the first publication from Heather Olson. And when you read the publication, you see that she will be facing intellectual disability, autism spectrum, psychomotor delay, cerebral dysgenesis, and so on. So very similar to other DEEs as well, and also to what Michelle was speaking about minutes ago. And back then, Lena was second in Poland, and around 40 case diagnosed globally. So as you can imagine, the interest around the disease was very limited. Back then, when we decided to start the foundation in May 2022, there was 0 scientific publications on the disease, apart from the fact that there were some case reports, and only one scientist, one professor interested in the PAX2 syndrome, and only because he was studying healthy gene. So at some point, he also decided to spend some time understanding the syndrome itself, and that was it. So that was our starting point. So really from scratch. And then I will tell you, how we, what kind of progress we made in May 2023, May 2024, and where we are today in our journey. And at the very beginning, that's all we knew about PAX2. So we knew what is the healthy gene role, what is the protein structure, and as you can see, the PAX2 is located in a highly unstructured region. And that there were cases of mutations in E21IK and E11K, which were leading to the disease. And the last thing was that the PAX2 gene expression was highly expressed in brain and skeletal muscles. And regarding brain, it was mainly oligodendrocytes. And that was it. So this is what we started from, and also from case reports, which were first described by Heather from Boston Children's Hospital, explaining how the clinical phenotype looks like. But today, before I move to the scientific part, today we are connected to more than 150 patients worldwide. And as you can see, not only we are connected to the patients, but we are also connected to the physicians, because building on what Michelle was saying about the natural history study, we are also preparing for that, because we are trying to tackle everything at the same time. So not only focus on the research itself, but also looking from the clinical perspective, what will be needed in case treatment is ready. However, what we see is that the same mutation variant, because 95% of the cases of PAX2 are kids with E21IK mutation, really like 95 or even 98% of the cases. However, phenotype differs a lot. So we have different onset of epilepsy. We have different spectrum of intellectual disability from mild to severe, or even we start to see patients with borderline or even almost normal intellectual development. You can have autism spectrum disorder. Most of the kids, they have it. And the same with verbality and non-verbality. More than 3/4 of the kids are non-verbal. So, but the striking thing is that despite the same variant, you have a really very phenotype of the disease, and we also try to understand why is that happening. And in May 2022, when we decided to start the whole journey with the PAX2, this is where we were in terms of the reagents. So it was quite lucky that we found out very quickly that very close to Poland, in Czech Republic, today called Czechia, there was actually non-humanized mouse model which was frozen. And we found about it by coincidence, in a way. And that was our kind of situation that, hey, so we kind of have potentially non-humanized mouse model with the PAX2, which is frozen. Let's do something about it. And then this is where we are today. So down the line, when we decided to start the whole journey, we got into collaboration with a couple of different places all around the world, including iXCells, because as Nyanwe mentioned, in fact, the fact that we are here in Poland and it didn't stop us from sending the blood all the way via FedEx from Warsaw to San Diego. And in fact, the first iPSC line was generated with iXCells, which was then in person delivered by Nyanwe to Northern Foundation, which I'm going to talk about in a second. But today we are basically we have I mean maybe not all, but most important reagents to work on, meaning fibroblasts, APC lines, neuronal cells, both humanized and non-humanized mouse model. And with some of them, like for example, you can see here with the non-humanized mouse model, we actually did some initial phenotyping as well. So we are kind of try to move as fast as possible. But what is very crucial is that from the very beginning, we have this approach of doing everything at the same time. So we focus on basic research, translational research, clinics, all at the same time. And from our perspective, it's all about the cooperation. It's all about reducing the bottlenecks from administrative perspective, and also maximize the investment. So we are looking at every dollar from every side. And what we also do, we try to monitor all the work on the PAX2 syndrome across the globe. So in case there's something new happening around the PAX2, very often researchers, scientists, they approach us first, and we straight away we update them what is happening in all the places in the whole world, so that we don't duplicate the job, and we allow this flow of the information between everyone involved in our journey. So as you can see here on the slide, we are focusing on the in vitro, in vivo aspect. From the translational research, we are super lucky to be collaborating with the NLRM, because Lena is part of NLRM family since late 2022, and that's the most promising approach in PAX2 syndrome. Silencing the mutated gene, or the whole gene as such. But we are also trying to approach the problem through drug repurposing, and I'm going to talk about it in a second. But we are also looking at more long-term perspective. I'm talking 5+ years from now, regarding RNA editing, genome editing in mice. And last but not least, as I mentioned, we are focusing on collaborating intercontinentally on the natural history study, because with such limited patient pool, sometimes it's sometimes it's impossible to do the proper natural history study within one country because of the number of the patients. Because obviously doing the natural history study with 2, 4, 5, 10 patients is not enough. So that's why from the very beginning we work intercontinentally, as you can see here, for example, between Boston Children's and our medical university here in Warsaw. And we are part of many different initiatives like EpiCare, which is epilepsy care in Europe, or Simon Searchlight. Today we do have some progress around PAX2 on many fronts. So first of all, we have a couple of compounds as a potential drug repurposing candidates based on our cell painting study. research with Charles River. As I mentioned already, but I'm gonna mention it once again. Lena is part of an orphan family, so we are part of this active program for Parkinson's syndrome, hand in hand with many different diseases, including KCNH1, as Michelle mentioned. We do have the mouse model ready for the preclinical examination, but we are also focusing on things like publications. We do know it's important also for the researchers to publish about the job, and we try to do anything we can to support it. And we are also focusing at some point to move from crowdfunding and using our private funds, because something that I didn't mention is that 95% of things we're doing is our private money, either our private money or crowdfunded through friends and friends of friends and so on. However, we are doing also some progress around the grants. So we got 2 grants awarded for basic research in 2024 from NIH. So we're actually helping or closely collaborating with researchers that actually got them from Pittsburgh and from Chicago. However, we are not stopping here. We are in process of submitting or awaiting decisions for different grants all around the world, including US and Europe. So kind of to summarize, before I move to the video, kind of summarize the situation right now. So from May 2022, with one interest among scientists and one project of the Parkinson's syndrome, today we have more than 50 scientists which are somehow involved in the disease, and more than 10 active projects happening all across the world. Which I wanted to kind of highlight here is that it doesn't matter that we started from 2 patients in Poland and 40 patients in the whole world. I tend to say that today more scientists are involved in the project than number of patients diagnosed 3 years ago, which for me is amazing to be able to say that. And also to kind of to send the message out there is that at the end of the day, science doesn't choose, and science doesn't differentiate if you have 2,000 or 2 million patients. It's all about the science at the end of the day, and you can make some scientific breakthrough even when you decide to spend the time on such a ultra rare disease like Parkinson's syndrome. And at some point, we decided to make things more official and kind of collaborate even closer with the community. So as I mentioned, we do have a couple of families which we are connected to, but also a couple of scientists which we are connected to. And at some point, we decided to kind of bring them as much as we could bring them together to Poland. So actually, we had a couple of people flying all the way from the US, from UK, from the Netherlands back in September last year for our first conference, which was mainly focused around the Parkinson's syndrome, but we're also talking overall about the rare diseases and what is happening, in both US and Europe and all across the globe around the rare diseases as such. And before I move to the last slide around the advocacy that we are doing in Poland and in Europe, let me invite you for a small video, and hopefully it will be working and the audio will be there.
So welcome on the conference.
I'm just amazed by how many strides have been made in such a short time and how many people have been activated to the cause.
The scientific level was very, very high. I got to interact and meet a lot of very interesting people. I think I come back home with a very renovated vigor, let's say, to work on PAX2 on a lot of rare disease challenges.
That also allows me to see the patients, their problems, their disorder or their disorders in different ways that would allow for different approaches and different treatments.
I'm surprised. I'm surprised because I find that it's extremely incredible how you manage to bring, even though it is an ultra-rare disease, small community, that you manage to bring so diverse people with such a different experience, with such an excellent scientific talks, with people, with really from the diverse horizons.
So I hope the audio and the video is working. And the last thing I wanted to tell you is that, when you focus on the rare disease, when you focus on the ultra rare disease, very often naturally you start to advocate for the whole community. So obviously 80% of our time we are spending on PAX2, but every month it's moving towards, the percentage is growing towards overall rare diseases. And we are advocating around it in Poland, we are advocating around it in Europe and also in the US. For example, here on the left-hand side, you can read that we were invited to open the symposium of UK platform of nucleic acid therapies as the main family speaking around the progress we have done. On the right-hand side, on the top, you can see us talking with actually the Polish Minister of Health, Izabela Leszczyna, about the need for the increased public support for the innovative projects on the rare disease therapies. And last but not least, what is for me very proud is, despite the fact, again, we are talking about the disease where we have today 7 patients in Poland and roughly 150 to 200 diagnosed globally. So you could say, hey, we are talking about very small community in Poland, but despite this fact, we were noticed by the Ministry of Health and Polish government. And my wife, she was invited to join the Council for the Rare Disease at the Polish Ministry of Health, which is very prestigious council, because there are only 3 patient representation there and around 50 to 20 most important professors which are involved, which are actually day-to-day involved in the rare diseases, together with the representatives of the Polish government, which allows us also to advocate for the overall community and access to innovative therapies and innovative projects, because it's not only about therapies, it's also about allowing a regulatory pathway to be able to treat these patients faster. Because in some of the situations, we actually sometimes have the treatment, but if you don't have the regulatory pathway that allows you to move fast, you tend to lose time with the treatment being there on the shelf. So this is also what we are doing. We are advocating for overall the community, which also is important right now in Poland, because we are leading the European presidency, which also is allowing us to to target and focus on more and more people, not only in Poland but in the whole European Union and Europe as such. And that was it. So thank you for having me here. And again, I'm super happy that I was able to talk once again on the iXCells Day around the rare diseases, because I was able to talk about it 2 years ago. And once again, I want to say that despite the fact that we are miles and miles away from each other, it didn't stop us from collaborating together. It didn't stop us, the parents, from jumping on the plane and flying all the way from Warsaw, for example, to Boston for the NRM meeting every year, or meeting other scientists in a different part of the world. So thank you for that, and I leave it to the last presenter from patient's perspective.
Thank you, Peter. Thank you, Peter. So that's another very, very inspiring journey by the patient. So we've been seeing so many parents that they are switching themselves from a non-biology background into within a very short time frame become a PhD level scientist. So we have been hearing so many stories, and our next speaker is also from a similar journey, but a little bit distinct journey. Noah Auerhan. He is one of the earliest N-of-1 patient parents collaborating with our organization for developing personalized therapy. He is a serious entrepreneur in tech. After his daughter, Ellie, was diagnosed with Rett syndrome, he devoted himself to find treatment for Rett syndrome. He began by identifying repurposed drugs that had improved Ellie's phenotype and quality of life. And over the last 2+ years, he has been developing a series of novel compounds that are working in Ellie's cell lines and are now beginning animal studies. He founded Uncommon Therapeutics, a VC-backed startup, to pursue his effort to build the curative therapies for Rett syndrome. So,
thank you. Thanks, everyone. First, thanks to a lot of people in this room doing actual work for us. So that's really, really exciting. So started a company called Uncommon Therapeutics to basically kind of house all the work that we're doing. As Yamwe mentioned, basically my entire adult life has been building companies in tech and initially e-commerce and then health tech. Started my first company actually down here in Pacific Beach. So was here from 09 to 15 roughly. And then sold that company to Rakuten and then took some time off, started another company in health tech, and tell you about my journey from there. My co-founder, Ryan Lim, is spent about 15 years kind of studying CNS diseases at UCI, and most recently at Module is working on an ALS drug that's going through IND enabling right now. So a bunch of really awesome people behind us and great partners that we've collaborated with over the last almost 4 years now. So my background, I was, while I was building my last company, my daughter Ellie was diagnosed with Rett syndrome in 2021. And so I stepped back from my role running that company to focus all my time on building a care plan for her. When you get diagnosed with something like Rett syndrome, which is more common than a lot of the other diseases that folks are talking about, still the general recommendation is PT, OT, and pray for gene therapy is essentially what a doctor will leave you with. And that just didn't really work for me. So Rett's caused by de novo mutation to MECP2. Basically imagine your child is developing normally, and then around 18 months they lose the ability to walk, speak, and use their hands. And then there's essentially a cornucopia of terrible things that happen to them: seizures, scoliosis, dysphagia, on and on. It is a neurodevelopmental disease. So in 2007, there was some exciting work that showed that simply replacing the gene could rescue the phenotype in mice. And so that kicked off a lot of funding. And that was part of my kind of experience going into Rett syndrome, which is, one of the first day or so, Googling or going to PubMed and you search MECP2 or Rett on it, and you'll notice 3,400 papers, 200 new ones every month. At the time, no available treatments. So this is like the dream that everyone would want, their rare disease to get this kind of attention. And yet PTOT and pray for gene therapy was essentially all you could ask for. So I think it's a lesson as we figure out where we prioritize our efforts in terms of what are we bringing to patients. Rett does have its first approved drug. We'll talk about that a little bit later now in 2023. But basically, I dove in deep. I probably read about 300 papers in the first 9 months, met with every expert until I became one. And it seemed obvious to me that we could not develop a gene therapy in a timeline that would be able to slow down this regression, this loss of skills that happens over a period of 3 to 12 months, really. But we could try to bring drugs off the shelf, and so that's what we did. We developed a thesis around oxidative stress, neuroinflammation, a neurochemical imbalance that exists within Rett syndrome, and a lack of growth factors. And so we attacked that with a handful of repurposed drugs in series, and a couple of those medications that have been explored in Rett syndrome in those 3,400 papers, really purely based on lit review, and a couple of which Ellie is the only girl in the world with Rett syndrome currently taking. And so while Ellie is still limited by the disease, here's just a video I took of her the other day walking around her yard. Ellie is 5 years old. She can walk around. She does have an apraxic gait. She has limited hand function, but can touch for yes and no. Limited speech, but things like yes, no, more, done, I do. And you'd be amazed at how helpful those things can be when she can get them out. She doesn't have seizures. She doesn't have dysphagia. She's on her height chart, weight chart, head growth chart. And she's using her eye gaze and is also learning how to read. So her life doesn't quite suck the way that we thought it would, and our life doesn't suck quite the way we thought it did. And that was kind of like the first kind of 18 months was getting to 18 to 24 months was getting to a place of like stability around this. And we're fortunate because it's a neurodevelopmental disease. I think if you're dealing with a neurodegenerative disease, it's a different story in terms of the timelines that you have. But this is giving us an opportunity to then go to the next step of, can we attack the underlying cause of the disease? In this case, the gene of interest, MECP2. So could we leverage, our expertise and the latest technologies to target the underlying cause of the disease? And the more and more time we spent kind of thinking about this, from both a scientific perspective on the disease, but also a business perspective on the disease, on how to attack the problem, is essentially, thankfully the investment community has spent billions building lots of new technologies and ways to build drugs. It's really awesome. And so we have, all these new modalities to attack the problem, yet many of them are not reaching our patients. And then also we have, a lot of really great partners who are building capabilities like here at iXCell to do specific activities for us that we don't have to build our own lab to do. And so, can we leverage these technologies, tools, and really great people that are available to us to guide us? And so, what we believe is, an alternative perspective is maybe the platform actually was never the technology, it's actually the disease. And in a disease like Rett, every single drug you would bring to market would be worth billions. And the reality is we know that these rare diseases typically require multiple drugs. That's what we're seeing. And so we think it actually makes sense to build a company there. And that is actually where the value creation point is, and likely better for investors because there's just so much dilution that happens here during the platform creation part. And so we've been proving that out. So for about $1 million, we've developed 3 different approaches to addressing the gene of interest directly and across 3 different modalities that are all working in patient cell lines. So we have an ASO that will increase MECP2. Basically MECP2, the mutation of it, it's a transcriptional factor, kind of epigenetic chromosomal regulator, really important in the brain. And basically the mutation causes it to lose affinity for the DNA, and then it gets downregulated by 70%. In 2017, a researcher out of UPenn showed that simply increasing levels back to normal was sufficient to rescue the phenotype, because it kind of does this, and that's good enough. And that's a much simpler problem in general than trying to replace the gene everywhere. And when you have different communities, it probably took, I think, the Rett syndrome community like 5 years to adopt that into the orthodoxy, where everyone finally agreed on the research. But meanwhile, that seemed like an approach we could take, so we designed an ASO that would bind to the 3' UTR and increase expression of MECP2. And also we did a large screen looking for basically existing repurposed compounds that could also increase MECP2. And then most recently, got some CRISPR base editor data back, working with the folks here, and in collaboration with some folks formerly from Beam to help us design a base editor. And now we have one that's hitting the mutation. Her mutation is the most common Rett causing mutation, which would account for about 10% of Rett syndrome. And it's correcting about 50% in the cell lines. And so all of these we're working to basically take into the animal models of Rett syndrome. Meanwhile, just because we just started at Y Combinator, we're going through their process and have access to like incredible computational talent in there. And so starting to look at ways, if there's ways to use AI to basically predict compounds that could stabilize the protein as well. So one more project to keep us busy. And so why we're approaching this a little bit differently. So one of the things is, I really don't love the word rare. I think it connotes something that will never happen to you. That's why we call ourselves uncommon, because Rett is not rare. It impacts 1 in 10,000 girls. This is the first Rett approved drug. This is their first full year. They've done 350 million in sales. This is the chart that got them approved. This is the chart on a y-axis that is a full y-axis in terms of the impact on the phenotype. So pretty negligible. Meanwhile, 85% of the patients had diarrhea during the trial. So it's questioned whether it's actually was an unblinded trial. Anyways, that's what's available to us. I think what it does show is they at least know how to really run a trial. And there's a strong desire for treatments in this community and the willingness to put up with shit. So I didn't mean to say that, but no, it's and this is not unique to Rett. This is all of us. And so, you have a lot of companies, you have Taysha and Neurogene that are working on gene replacement. It's very exciting for our community. We're seeing those go into phase 1. We're seeing early data off of that are very, very small ends. You're seeing some improved hand function. That's encouraging, but you're not going to transform that full chart. We might see a meaningful step, I think, with the gene replacement, but the reality is AAV gene replacement can't get to enough cells with a single intrathecal or ICV injection. It's just not possible. And so we already know that this disease is going to require multiple more modalities until we get to the finish line. So this is the work we're doing, and brought in really excellent people who have done this kind of work before. And basically, now that we have our 3 candidates, we're taking all of them into animal models. So the ASO is in partnership with Loyola. They're bringing that into mice right now. It's already being injected. And the small molecule study, we're doing our first target engagement study right now with Cyogen. And then we're starting AAV manufacturing on CRISPR with the goal of doing talks over the following 18 months and then entering phase 1 soon thereafter, and trying to collect some early patient data and then raising the additional capital that we need. Just quickly, some of the key experimental results. This is our most recent base editor data. Really excited about the over 50% editing and the no bystanders, especially in the context of this mutation. There's only 2 non-A's on either side. So the fact that we could hit that was good luck, but also working through a lot of combinations with the folks here at iXCell. We also designed, these are some of the ASO data that showed increasing levels, 1.5x or greater. And then our small molecule work and even seeing some correction of the phenotype and other downstream genes that we did. That's what we're up to. Thank you very much.
Thank you, Noah. So we are closing our first section. We will have 20 minutes Q&A. So can I invite the 3 speakers here facing the audience, and also Peter, if you're online. So we're gonna take questions. So there are several questions from online, but I would like to see anybody here in our room that have questions.
Okay, hello. So I have one question for Yue and another for Noah. I'm going to start for the one for Yue. So I was very excited when I saw your phenotype in the microcephaly organoids. I was doing my postdoc at Mootri Lab like a few years ago, and I actually used the same, I actually look at the same phenotype in other disease. So when I saw that violent plots, gave some memories. Did you see also any other kind of phenotype apart of the organoid size? Did you also look at electrophysiology, or did you look at any other stuff?
Yeah, we did look at electrophysiology. Unfortunately, there was a lab-wide incident which killed all the organoids partway through the project. So, that luckily we got the microcephaly phenotype and the rescue study done before that happened. But by that point, it didn't make sense to restart the whole thing. But we were looking for electrophysiology in the thank you, Leo, in the cells as well as the organoid. So it was on our radar.
So with that data, then it was the only kind of data you had in order to push it through the FDA and then to go to the clinic?
That's correct. That was the only efficacy data we submitted to FDA. And in fact, if you look at the N of 1 ASO guidelines, so they only apply to N of 1 ASOs, they do not require efficacy data to approve a phase 1 N of 1 ASO. So that is a very kind of special exception that FDA is making, and I think it's a good decision on their part.
Yeah, indeed. Thank you. And then also for you, Noah. Hi.
Hey, good to see you.
Yeah. I see that at the end that you are looking at 3 different strategies, in order to tackle the disease, using ASOs, using small molecules, and then using CRISPR in order to deliver to the cells. This is a little bit more speculative. What do you think is going to be the most efficient approach?
Yeah, so I think the highest probability is the existing repurposed small molecule that has the highest probability of getting into patients soonest. But the way that we're thinking about it is not that they're separate. shots on goal, but that they're a series, and like we're not done. And like I think each drug comes with its own challenge, right? Like so an ASO, it's like, can we get to enough of the brain regions that matter? An AAV, similar but different brain regions, potentially depending on how you did the injections. BBB, small molecule, well, like how okay, it's a repurposed drug, so like is it just really going to be as efficacious? You know, do you build a custom drug? It's like, well, then you're going to do all the other, you know, med chem afterwards. So I think it's, you know, if you just look at what's happening with like SMA, where it's like multiple drugs, everybody's kind of working the problem with multiple drugs. So I have what I think will be the first, but I wouldn't say, I think it also that I believe will modify the phenotype sufficiently to get approved. But I think there'll be a series of them in the end.
Okay. Thank you very much. Other questions in the audience?
There are a few questions from online. The first question, I believe, is for Yi-Wei. It's a very practical question from Anna Rita Morella. So she said, so can you summarize the timeline and money spent during your Odyssey? I would love to have an overall idea of what to expect.
Yeah, so timeline was we got diagnosed at, you know, month 4 when Leo was 4 months old in February of 2022. It took us about 4 months or so to pull all the partnerships and all the researchers together. And from there, it was about 12 to 13 months before we got into the clinic. And we had multiple repurposing efforts going during that time as well, knowing full well that that is the fastest way to some kind of a therapeutic. Unfortunately, most of the repurposing is literature based, right? And so this protein is so little studied that there was basically one paper on TMP02. That was the paper that established it as a disease-causing gene. And so, unlike Noah, I had one paper to read, which was, you know, it's good and bad, right? So after reading that one paper, I knew, you know, I became the expert or one of the experts, right, after reading this one paper. But you know, the other downside was there was very little to go on in terms of finding a repurposed drug. In terms of budget, I'll just say that it was in the millions and not the tens of millions. So it's in the high millions. And again, you know, if you look at the Gantt chart, or there's so many dead ends and there's so many things that did not work, and that, you know, a lot of if you take the critical path, you know, it's in the low millions. But if you wanted to fully kind of make sure that, you know, I wanted to make sure Leo was covered in all these eventualities, it was in the high single-digit millions.
Yeah, I have a question for Noah. Yeah, so I know we did some ASO screening and identify a couple of ASOs which can increase the target protein by like 1.5, 1.7 fold. And that's your goal, right? You want to increase at that level, a small change would be enough. But since I have worked in a pharmaceutical company, in general, such a small assay window is very, very challenging for translational science. Yeah. How can we maintain such a small effect from in vitro cell culture system in the human? We see the similar effect. And also, I was wondering whether if we overshoot, let's say we overexpressed by 2-fold or 3-fold higher than expected, is there any adverse effect? So any thoughts on that?
Yeah, so a few things. So one is the ASOs that we designed. What was really interesting was while we were doing that, we found another researcher out of Loyola in Chicago who's doing the same thing. And so he had similar ASOs that were kind of in that 1.5 to 2x range. His appeared to be less toxic than ours. So we're basically in licensing that work right now. He's right now bringing that into animals. I think it, you know, mechanistically, so we're trying to inhibit a single microRNA. There are multiple regulatory microRNA that are targeting our gene of interest. So I just don't think you can overshoot that much. But this is like some of the other questions that our space in those 3,400 papers, for some reason, hasn't answered, is the duplication phenotype. So there's a MECP2 duplication disease, but it's unclear whether if you introduce it postnatally that there's even a phenotype, which is like a strange, simple question that we probably should have answered a decade ago. So we don't know. The kind of general thinking is that there's not, it doesn't matter postnatally. But there's at least high confidence there's a tolerance zone of like around that 2x number, just staying below that. So I think, again, like mechanistically, it's hard to imagine how you could, just by inhibiting a single microRNA, that you're going to overshoot like 4x. It just doesn't seem possible.
Hi, thank you for the inspirational start to the day. Quick question in regards to, we've all spoke about the importance of setting precedent with the regulators and gaining access to the work and the advancements that we're making. It's a general question to all of you. It's, in your experience being so close, how have you seen that changing over time? You know, is it getting better as we build better tools, better data? Are we really getting to where we need to get to, which is getting these things into the patients? And I don't know how you want to answer that. It's really a broad topic.
A lot further along in the process. We are not close to IND enabling studies quite yet, but I hear from lots of fellow rare disease leaders, and it seems like there's some slow progress. I mean, everybody's doing listening sessions, engaging as early on in the process as possible. And there's still a lot of work to be done, letting regulators know what are meaningful changes to the patient community and the risks that we are willing to take because of what our kids and our families go through. So yeah, I think baby steps, but we have a long way to go.
So I think there is some progress. It's slow. It's almost like one step forward, one step back. Or I mean, 2 steps forward and 2 steps. I don't know. It's like 1.8 steps back. So it's like, yeah. So and I think maybe the most helpful thing I can say is this: physicians have a lot of leverage in these conversations. So for Leo, he had a neurodevelopmental disease, and it puts a physician in a very difficult place because, when the patient has a neurodegenerative or a terminal condition, there's a much higher willingness to take risk. And I think that is probably where we will get the most progress is, for the physicians, the principal investigators who are treating the terminal cases. I think we can get the most flexibility on precedent there, and then try to push that forward to other patients. In Leo's case, I was kind of, I don't know, like, I would say I didn't regret that he was neurodevelopmental instead of neurodegenerative, but I was wondering, like, if this had been neurodegenerative, would he have a better outcome because we could get the drug to him sooner, and that's just a really weird and convoluted, logical place to be in, that the system, the regulatory system and everything has set up for the parents.
Just from my experience, we did expanded access, which is compassionate use for one of the drugs. And it was surprisingly easy. There's like a phone line you can call FDA on and get approval over the phone to start using a repurposed drug. And so I don't know that clinicians realize the power that they have, which is if they think it's in the best interest of the patient and the patient properly consents, it's a phone call to introduce a repurposed drug.
And I think that actually applies to new drugs too. If the physician is willing to put their career and their reputation on the line and say, I'm willing to go out ahead of the FDA here, they can get through these barriers.
So that's what I meant. It's phase 1. If it's been dosed in a single human, it is now eligible for a phone call to FDA.
There are more questions. So I believe the question is for Noah. So this is from Anna again. We have a community that says they really want treatment, but they don't engage as much as we would like to get the data fast. Any tips on how to ignite the community from saying they want treatment to help and do the work to actually reach them? Fundraise, data collection program, etc.
I don't think I've actually done a good job of that. I think Michelle's done a good job of that. I've been among the fortunate to have fallen into a community where a lot of people have already done that legwork, and kind of community building, natural history studies. So like I got to skip that part, and I think.
Sorry, can you repeat the question?
So basically, how to encourage the community to gain data faster.
Just outreach, a lot of Facebook messages, a lot of, just community engagement. Again, engaging the scientific advisors and the researchers to communicate directly with the patient communities. That goes a long way. That really increases their engagement and motivation. But it's just, building those relationships, building trust, and taking the time to engage with the community.
I have a question for Piotr. Are you still online?
Yep, I'm still online.
Awesome. Yeah, I have one kind of scientific questions and one non-scientific question. So the first one is, I saw you have done the cell painting assay. My question, I do see you have several candidates, like 23 candidates. So how do you validate those candidates with more physiological relevant assay or any phenotype related assay? Because in general, for these unbiased assays like cell painting, you can always see some difference, but the difficulties or challenges is how to relate those principal component analysis to a particular phenotype.
Okay, so let me start by saying that in our case, what was very beneficial was that we have twin sisters. Despite they are fraternal, but they carry, the DNA from the same parents. And at some point, we decided to approach the issue from the, let's say, precision medicine point of view. So actually, we decided to only do the cell painting on Lena and Zuzia cells, and we were quite struck by the fact how fast we were able to see the phenotype using the machine learning between these two cells. Basically, at the end of the day, comparing morphology of the cells on a very different levels, and it doesn't happen very often with the rare diseases. Sometimes you look at the cells, even like the fibroblasts, and you see almost no difference or no significant difference. In our case, because probably because of the fact that we are talking about the twin sisters, yet fraternal, we were able to quite quickly, distinguish the difference on the morphological level. And to your point, at the end of the day, after screening, 6,000 plus FDA-approved drugs, we are currently looking at the ones that are moving towards. We are looking at the drugs that are moving Lena cells as close as possible to Zuzia cells. And obviously, we'll do at the end of the day some filtering regarding, toxicity, regarding the side effects connected to that. And we want to filter down the list to 5, 6, which we can use on the mouse. However, if we were approach the whole issue from biased approach, meaning, hey, let's look at the publication, what we know about the PAX2 syndrome, we would never ever list these 23 compounds. They were like super surprising to basically everyone involved. We started to classify them in some mechanisms of actions. So it's not like we're not talking about 23 totally different drugs with 23 totally different mechanisms of actions. Actually, when you start to kind of gather them, you start to see some pattern. But if you would approach it from purely publication approach, so biased approach, you wouldn't pick any of them. So that's kind of the where we are with this. So at the end of the day, answering your question in a very simple way, we are going to focus on the ones that are moving Lena cells as close as possible to Zuzia cells, based on the 3 replicates we did during the final screen of the drugs from the FDA list.
Thanks. So I guess my second question is a little bit associated with the first one, and also a very general question for all of the foundation leaders. So, because you guys did a tremendous job to join all these patients globally, but eventually all these studies, it's going to be case by case and focus on one patient, right? So how do you really come up with a strategy to prioritize your pipeline? And how can you scale up or make sure the resources spending in one particular patient can eventually benefit to most of the other families? I think there's I asked the question to several foundation leaders, but I know it's very, very difficult and kind of ethical related stuff. So I'd like to hear you guys as a thought leaders.
So my thinking about it was that if we could, get enough efficacy for Leo, then we could prove that these other children deserve a chance too. And then, along the way, if we publish and open source, so to speak, what we did, then that would be the, giving back to the community, and people could take what we learned and use it, themselves. I think, in terms of how it actually played out, we got the drug in Leo much later than I would have wanted. And, the efficacy is, I'm deeply grateful for any efficacy and for any, avoiding adverse events. But, it might not be so compelling to a payer or insurance. But, there is the open sourcing, and we've committed to publishing our IND documents, which is often a very big barrier to getting into the clinic. And so that is the thinking that, it's a learning experience, and if we give the learning to the community, then that will help every single foundation get to the next step.
So I start with that your fiduciary starts to your child. And so that's kind of how I initially approached the problem. And I made a bunch of effort to try to share what I've done from a repurposing standpoint with the community pretty unsuccessfully. I think that the community, there are folks trying to run trials with some of these different meds that she's on. Some of them, they've already ran great trials in other countries. And, every kid with Rett should be on some of these drugs. And it's just not the standard of care here in the States. And that's a really hard problem to change. It's like a societal thing. I think what I've seen a lot of rare disease parents is they'll spend a bunch of time trying to run a repurposed trial to prove that a drug that their kid's already on and kind of some folks in the community are already on works so that it can become the standard of care prescribed by doctors. Just a lot of effort to do that work. And, one of the things I talked to David Fagenbaum, who's, probably a lot of you guys know, but Kurt himself of Castleman, it's like, well, David got to the finish line, so he can help everyone get there. And we're still running the race. And so it's a complicated dilemma, I think, to the point of prioritization for us. And so definitely could be better at sharing, but I think as we, prioritize, now I'm once you start to take on capital, you have to your fiduciaries change. If you take money from other families, then your fiduciaries change. If you take money from investors, then your fiduciaries change. And so you just have to be, I think, comfortable with what they are.
Thanks. There's another quick questions for Noah. He's asking about a compound you mentioned, a repurposed drug. Do you mind sharing?
The ones we're on?
Yeah.
There's 5 different drugs that she's on. So she's on vatiquinone, which we had to go to FDA for. That's the one that's not approved. It's a PTC drug that they're trying in Friedreich ataxia. Dojolvi, which is an ultragenics drug for medium chain triglyceride, so reduces oxidative stress. There's a doc at Emory who ran a 12-patient trial and still hasn't published on it. and at least anecdotally, it seems that it, in his study showed reduced seizures in that patient population. The third is mirtazapine. That's the one that's like blows my mind. That isn't the standard of care. There was an 80 patient study in Italy, which is pretty robust. Showed improved sleeping, reduced apneas, increased appetite, which are like amazing for a drug that you can literally, get at Rite Aid for $1. And then she's on low dose ketamine intermittently. Again, they tried to run a study in that with Rett syndrome, but it kind of collapsed during COVID. And then the most recent one was Aricept. That was a study done by actually some of former Mootri people at Viant Bio, and they saw it as one of their top hits, kind of normalizing the organoids. They were trying to run that in a study out of Vanderbilt, and that study unfortunately just collapsed from they lost their financing from the DoD. So that's it's good ideas that I don't know, I didn't have to invent. They're there, and but you can see the challenges of getting them to patients over and over and over again. And I think, frankly, it requires insane willpower to eventually get there.
We have 1 minute to take one last question. So we have online questions for Yiwei. It's also one of my curiosity. So how was AI was used in drug design and development, and particularly how I'm gonna, because in a Baby Lion project, you are trying to use the data and the experience from Leo's case to help to diagnosis and design the drug in a faster way, right?
So I think in Leo's in making Leo's drug, Crayon Bio, using Crayon Bio's model to generate the leads was the main application of AI. There was also, I also used a few of the earlier protein folding tools to try to model the mutant protein. And I think going forward, in the last 3 years, there has just been a tremendous amount of progress in AI, in biology, well, in everything, but also in biology. And so going forward, I think that the answer to that question is very different. AlphaFold 3, AlphaFold 3 came out during the course of Leo's project. It's not perfect, but it can predict interactions between proteins and non-proteins. That the performance in the model is not so good, but again, the next generation will be better, and so on. And AlphaFold 3 has competitors. They have open source competitors. There's all kinds of, Schrodinger is an older company, but there's just a plentiful number of companies who are approaching biology, chemistry, biochemistry, even cell biology. There's an effort at CZI at building an in silico cell model, and that will just change, it will change everything. And so I'm really excited for, where AI is now. There's also the Baker Lab, they can predict protein binding from the sequence alone, the sequence of amino acids alone. And they can get up to 50%, they can get 50% success, at just picomolar, dense, anyway. And it's incredible, right? So that's where we are already. And so, even a year or two from now, I look forward to the progress.
Thank you. So let's closing up this first section, and we have 15 minutes break, and we can continue discuss.
A warm welcome to everybody who's joined us here in person today. Thank you for attending, and to extend a welcome to the speakers in our next session, which is titled Modeling Rare Disease with Patient-Derived Cells, a Platform for Discovery and Precision Therapies. We are pleased to have a panel of 4 speakers from leading foundations and biotech firms who will share their approaches and insights into this field. Our speakers will discuss how patient-derived cells can enhance the understanding of rare diseases and lead to more precision and effective therapies. So as we come out of the first session where we see the impact of these therapies, understanding how we can keep advancing research accelerating the path to the therapy to get these important therapies to where they matter most. Just before we start, as a reminder, we'll take questions at the end. We have a Q&A session with the panel. For those online, if you'd just like to enter your questions in the chat, we will get to them during that session. So without further ado, I'll move to our first presenter, who is Dr. Irene Morgenstein. Irene is the Director of Preclinical Initiatives at the Children's Tumor Foundation, leading drug discovery in neurofibromatosis, or NF, as I'll refer to it from now on. Irene earned a doctorate in pharmaceutical sciences from the University of Sciences in Philadelphia and completed her postdoctoral training at the Rockefeller University in New York. Before CTF, she worked at generics, designing preclinical drugs for the central nervous system. She has managed data strategy at Regeneron Pharmaceuticals and as part of research and development IT team. Irene's expertise includes preclinical sciences, data innovation, and collaborations to advance treatments for rare disease. Her presentation is entitled Accelerating Drug Discovery in NF. Thank you, Irene.
Thank you very much, and thank you for having me here. What a wonderful event. First time attending and speaking, and I'm excited to learn from all the presenters today. So I'll spend the next 20 minutes or so speaking about our work at the Children's Tumor Foundation and how we put all of our effort into accelerating drug discovery for it's actually neurofibromatosis and schwannomatosis. There are a few reclassifications recently, so hopefully I can clear those up in the next slide or two. For those that have not heard about NF, or collectively, this is neurofibromatosis and schwannomatosis. It's a family of progressive debilitating genetic disorders. Two main types. There's neurofibromatosis type 1, and then there's a set of schwannomatosis. Within schwannomatosis, there's NF2-related schwannomatosis and non-NF2-related schwannomatosis. Collectively, tumors grow throughout nerves on the body, throughout the body, but that's just a very general way of speaking about it. For NF1 specifically, some of the manifestations could be disfigurement, blindness. Unfortunately, these benign tumors can turn into a malignant type of tumor called MPNST, malignant peripheral sheath tumor. You can have cognitive deficits and bone deformalities. And although it's a rare disease, it's actually not that rare. You can see by the numbers, 1 in 2,500 individuals. This is an unmet need. And I'm excited to say, as of last week, we actually have 2 approved treatments now. We had one in 2020 and another one last week. However, for schwannomatosis, there are no approved treatments currently. The incidence is lower, and one of the key manifestation in NF2-related schwannomatosis is hearing loss. So the tumor will grow around the 8th cranial nerve, leading to hearing loss and loss of balance, difficulty walking, and again, these are all progressive. Non-NF2 schwannomatosis, these patients have excruciating progressive pain. And again, there's no available treatments. Before I jump into a few other slides, I wanted to show a video because it's always better to hear it from the patients and learn a little bit about their journey.
My name is Leanna, and I want you to see me and see NF.
My name is Blake, and I want you to see me and see NF.
My name is Jayshawn. I want you to see me and see NF.
People who have NF are so varied in different types.
I basically tell them it's genetic disorder, can cause tumors to grow anywhere on or in the body, and it's like something that you're born with.
I look healthy on the outside, but on the inside I have over 100 tumors in my body.
But what people don't see with my diagnosis is that I'm losing my hearing.
They just see this person, oh, you're really in pain every day?
I have NF1. I'm the only person in my family that has NF.
they like make assumptions that they're not just like every other teenager.
Because some people are different than other people.
Might see you as like someone who's different, but actually understands you and knows like a part of understanding of what you're going through.
All of my tumors are internal. I don't have any skin tumors.
One of the things people can't see about NF is the constant worry we have, knowing that a tumor could be growing anywhere in their body at any time.
I'm not too sure. I might develop tumors, I might not.
You know, I think about the future and I get scared, but then I meet people within CTF and the NF community, and it's inspiring.
Just because I have NF too, and I've had 27 surgeries, despite these challenges, I'm still pursuing my dreams.
Someone could show a scientist, and then they could figure out a cure and keep on experimenting.
Although I have NF and I went through my own struggles, the second I found out my son had it, my entire world changed. And it was my mission from then forward to find a cure or an effective treatment.
The more attention there is to it, the more research, the greater chance they're going to find a cure.
We want everyone to support her and support everyone dealing with NF in the way we've felt so supported by the CTF community and the NF community.
My name is Jesse. I want you to see me and see NF.
My name is CJ. I want you to see me and see NF.
So what we do at the foundation is focus really heavily on drug discovery. So we have been talking about being the drug discovery engine for NF. And so what that means is we'll bring all the right people into kind of one space or in collaboration. So we have access to patients, to clinicians, to researchers, to government officials. And how can we do this together? How can we bring those folks together to accelerate this process of finding new treatments for NF? And our patients are at the core of everything that we do. And our mission is simple yet complicated, and it is simply to end NF. And so that's a little bit about us. We are in the States, and we also have a headquarter in Europe. So we're in Europe and the US. So we've heard stories already kind of this morning and all throughout the session. I'm sure we'll hear more about how the patients endure more than a frightening disease, not knowing what will happen, when it will happen, how to treat it, how to handle, giving care to the children, the patients. But the system is also a little bit broken. And we won't get into every part of that system and what's broken and how for rare disease it's hard to recruit patients. You have multi-sites where you have to get patients from. You have complex protocols, but I will focus on preclinical. That's where I'm heading here. So what are some of the challenges with preclinical drug development? We know it's expensive. We know it is risky. We know that it often does not translate well from preclinical findings into the clinic. We know that some models, and I think I heard it mentioned, they're difficult to get your hands on. So even if you have a repurposed compound and you have a theory, what do you do with that? You need a model. How do you find those models? Sometimes it takes a while to negotiate those terms with institutions to use those models. That leads to very long timelines to initiate a study. Time is of the essence. So whatever we can do to shorten this and make it more cost efficient or effective, that's what we're trying to do here. And of course, standardization. If this is something we're going to use over and over again, these assays, we need to make sure they're standardized and they're used in a similar way. So in order to overcome these hurdles, something that we're working, and this is our newer initiative, it's called the NF-focused preclinical hub. And it's not a physical hub, it's a virtual collaboration kind of concept. And so it's a integrated network of experts, so various institutions, contract research organizations, and other tools. And the idea is to bring them all together into one central virtual space and make them accessible. So how do we make the models available, accessible, for drug screening. When I say models, I'm talking about in vitro cell models, in vivo mouse models. We're talking about data tools. We even have pig models we've developed that can be used for PK/PD experiments. And drug libraries, right? How do we, look through, scan the literature? Because unlike other ultra rare diseases, there's a ton of literature for around NF. So we've generated NF-focused drug libraries, and how do we source those compounds to make them available for drug screening? So this is our initiative. This is what we're working towards. But how do iPSCs, and that's kind of the focus next, how do they play a role in this? When we thought about which models to generate or build as part of the preclinical hub, we focused on clinical translation. iPSCs were one of the models. We have others. We're also working on PDX models we're trying to generate and put into the hub. But right now we're speaking about iPSCs. And I don't think I have to go into much detail, but we know that these cell types are patient specific, so we can kind of recapitulate the mutations that a patient could have. We can go ahead and differentiate them into disease-specific cell types to better understand the pathology in vitro. We can scale them, we can reproduce them. We can even use them for either medium or high throughput screening assays to screen some of the repurposed drugs that we've identified. And they have a translational potential. So here's kind of where, I think that's the button, yeah. So here's where we're fitting in here in this in vitro. We have other models that we're working on, as I mentioned here, but we're just gonna focus in on this little part. Before I talk about how we went about generating these cells, I have to give a little bit of background. So if we focus in, and this is on neurofibromatosis type 1, NF1, the gene is on chromosome 17. And this is a rather large gene, one of the largest in the human genome. It's 60 exons long. And then I think this number is actually outdated, the 2,800 different pathogenic variants. I think there are more reported at this point. And the protein product is neurofibromin, and it acts as a negative regulator of the RAS-MAP kinase pathway. So this is indeed a RASopathy. To have tumorigenesis in the NF1 patients, it's based on a two-hit theory. The first hit is a germline mutation in the NF1 gene here, followed by a somatic mutation in the second allele of the NF1 gene, and you have loss of heterozygosity. This can occur in different cell lineages, causing the complex spectrum of manifestations that I mentioned on the first slide. So it can happen on Schwann cells, astroglial melanocytes, mesenchymal progenitors, and these are just a few examples here. So you can quickly see how complicated this disease is. So how do we model this? How do we even start? What we did here with our first set of iPSCs is based on a publication that came out of University of Alabama at Brigham, UAB, we selected a germline variant based on genotype-phenotype study. The study identified 6 mutation hotspots, and we ended up focusing on this GRD region because this is important for RAS signaling. And not only did we focus on this GRD region, we also focused on a more severe phenotype. This more severe phenotype led to cognitive impairments, spinal neurofibromas. COMs are like pigmented spots on the skin and skeletal abnormalities. So that's how we went about, where do we start with these cells? And we worked with iXCells and their team here to generate our first set of cell lines that will go under this preclinical hub. We used CRISPR, so we did not start with patient-generated. There are a few hurdles there we're working through. We started with CRISPR-generated cell lines, and we used CRISPR to introduce a missense het knock-in with this arginine 1276 glycine variant, again, identified from this publication. We then used the het cells to introduce a truncating mutation on the stop codon on the second allele. And I'm sure the iXCells team can tell you all about the wonderful work they did to generate these cell lines for us. So right now we're early on in the process. So I'm afraid I don't have too much data to share. It's just a concept that I'm sharing here. So right now, we are hopeful that these cells will be available through iXCells soon, as soon as they're characterized and validated. And we have big plans for these cell lines. We would differentiate them into, for example, Schwann cells, progenitors. These are the main affected cell types in NF1. We would validate these cell lines and develop a high throughput screening platform using them and generate spheroids as well. We do want to go beyond this one specific set of cells. We have plans to look at other mutation types. There are frameshift mutations, splicing mutation, nonsense deletions, so many that we can generate. This is only the starting point. And we absolutely want to expand into schwannomatosis because for schwannomatosis, although the incidence is rather low, I think that's the area that really needs some of these new models. So we're starting here, we're moving forward to all of these other next steps. And for the drug screening approaches, we would go ahead and validate the initial using clinically relevant drugs. As I mentioned, for NF1, there are 2 right now, selumetinib and mirdametinib, that are on the market. So we would go ahead and start with those validation studies when we have the cells ready to go and generate, next gen 3D models and functional assays. These are our plans, long-term plans here. So, I'll put a quick pitch in for CTF in the preclinical hub, saying that, what we're looking for are vendors or biotech companies that want to work with us and be part of this preclinical hub, industry partners that are interested in NF or that need some of these resources. Again, we're developing these resources for the community, for the research community, for the pharma companies that need to use some of these models. That is our goal, is to make it easier to screen compounds for NF. Foundations with insights, I've already heard so many here that I jotted down, so always reach out. Happy to chat. And institutions that are eager to co-develop innovative models. I think that's about it. And I just want to thank you. We have a small but mighty team at the foundation. And here are most of them. And it really takes a village, and all of these individuals contribute quite, to these preclinical efforts and to the mission of the foundation overall. So just join us in Ending NF. I'm happy to answer questions later or talk about how we can work together and collaborate.
Thank you, Irene. As we heard there, the importance of gaining access to patient samples to improve our models is a key bottleneck and a challenge. And for our second speaker today, it's my pleasure to introduce Barbara Van Heer, who is president of patent.org, the Rare Cancer Foundation's online platform, which empowers patients to contribute tissues and data to research regardless of US treatment location. She currently leads all of the team's patient advocacy and institutional relationships, as well as manages patent.org's tissue collection. Barbara's presentation is titled From Consent to Collaboration, Practical Insights into Patient Tissue Procurement.
Thank you very much. And thank you, iXCells, for inviting me to be here today. And I am the president of Pattern.org, and Pattern.org is an initiative of the Rare Cancer Research Foundation. And the Rare Cancer Research Foundation was started in 2013 by a rare cancer patient who happened to be diagnosed with ocular melanoma, who was discouraged after his diagnosis to find that basically no treatments had changed in the previous 25 years before he got his diagnosis. And as he did some research and worked with other rare cancer groups, found out his situation was the norm, that very few of the rare cancer groups had had significant change in their diseases in terms of treatments for 20 to 25 years. So he created RCRF in order to build the infrastructure that could be used by any of those rare cancer groups and now also rare disease groups to help get rid of some of the roadblocks that might be stopping research from moving faster. And just for full disclosure, this rare cancer patient happened to be my nephew, Mark Labs. Pattern.org was created in late 2016. We did a lot of research with the different groups, the different rare cancer advocacy groups on what really were the challenges. We talked to a lot of researchers, what were their challenges? And the number one thing they came back and said is, we don't have access to patients and we don't have access to patient tissues. So we created Pattern to provide that infrastructure to be able to work with patients. Currently, it's across the US. It will probably be into Canada within the next year or so, but we are primarily US based now. So wherever they're being treated or wherever they're getting their care in the US, to be able to get tissues from them. For rare cancers, usually it's solid tumor tissue. For blood cancers, of course, it's blood, but we're able to collect virtually any type of patient tissue. And so how does pattern.org work? Patients learn about research studies for their particular disease. They then go to Pattern. They are then matched to studies. In fact, Pattern is in the process of being updated and should be available, hopefully the new version within the next 6 weeks, where if there's more than one study that a patient can contribute to, they can actually rank their choices of how they would like their tissue to be utilized. It's not guaranteed that necessarily they'll get their first choice. Let's say if there's not enough tissue or there's something about their condition that doesn't allow them, but we will let them know where their tissue actually went. So they're matched to studies, and then they can go through pattern.org online and consent to that study. If they have questions as they're going through that, they can reach out to us, or there's also a chat feature. Once they consent, we're notified literally within about a minute of that consent process. And depending on the disease that we're working with, whether we need to, if they're having a surgery, let's say it's a rare cancer, then we'll reach out to their clinical team at whatever hospital they're being treated at. It can be a large academic center, can be a regional hospital, it can be a community center. So we reach out to that patient's clinical care team and work through all of the challenges that many of you may know about trying to get tissues out of institutions to get them to release that tissue. The patient can talk to their clinician about wanting to participate in the study, not required. If they don't feel comfortable doing that, we're happy to take on that burden to get that tissue out for them. So then we coordinate the transfer of their tissue and data, because we'll also get the clinical records associated with that patient's case. So some folks are confused, like, what exactly does Pattern do and what do the researchers we work with do? So on the left side is Pattern. We handle the patient consent and the tissue donation. As I mentioned, we work with that clinical team to collect the tumor. And in case of rare cancers, we also get the germline tissue. If it's not a cancer, let's say it's a rare disease and we need whatever we need to collect for that, we will work through the details with the group that we're working with to determine what exactly we need to put in the collection kit and who we need to work through to get that. And then we handle the consent and also the clinical data associated with that particular patient. We have the full medical record, but we give de-identified information to the research groups that we're working with. Then over on the partner researcher side, they are the ones that develop the models and any genomic or other data. We get the tissue to them. If it's fresh living tissue from like a solid tumor, then we have it overnighted the day of the surgery. So it arrives by 10:30 the next morning. The only hiccups we have usually if there is a FedEx delay, like due to a snowstorm or something like that, but we will track it. We will let the research group know where it is along the way and when it arrives. And then the researchers we work with, we really, again, we were founded to help break down those roadblocks that were preventing progress. So we strongly encourage, and in many cases require, that the research groups we're working with share the data pre-publication. For biotechs, we make some exceptions, but we'll work with them on what data they might be willing to share. And then that data that is shared would go through public biorepositories. Also, any models that are generated, again, with biotechs, it may be slightly different. We require that those models go into like an ATCC or other biorepository. How do patients find out about us? We have a wonderful network of foundation partners. This is just a small list of the ones that we work with. We will work with actual foundations. We'll work with social media groups. And we're also in the process of working, setting up agreements with some smaller community hospitals where actually tissue from many of the procedures literally gets thrown away. And so we're working to try to set up a more effective system to utilize those tissues. Who are the research partners we're working with? The ones listed here are the ones that are currently on our website that patients can consent to, but there are 11 additional sites that we are currently in the process of onboarding. From the work that we've done, again, primarily it's been rare cancers, but we can help rare disease. 65 research models have been created, and they are available and in the public domain or shortly to be in the public domain. We have collected from over 120, well, it's 120, I think the last count is 124 hospitals. And again, I should stress that the primary work that we have done to date has been with fresh living tissue, not with stored tissue, but we're also capable of collecting stored tissue. We've collected from 39 different states, and 20% of our samples have come from underrepresented groups. So kind of an overview of what we're doing with Pattern and with RCRF. Patients contribute to research and empower themselves at the same time. We get those tissue donations. Historically, what we have done is we collect the tissue, whether it's a blood draw, whether it's tumor tissue, whether it's normal tissue, and then we send it directly to the research lab. We have recently been working with some groups that have asked if we could store tissue for them. Let's say they need 10 or 15 or 50 samples of a particular tumor or biological material. We now do have a biobank in conjunction with Precision for Medicine so that we can store the tissues there. We can also do different assays there. And so we'll work with the groups that want to work with us on, do they want the tissues direct or to go through the biobank? We collect the medical records. We're also encouraging, particularly for cancer patients, but for others that where it might be interested, is to get genomic sequencing. And so we're more than willing to help communicate to the doctors and try to encourage them to order the genomic test. Several of the companies that do sequencing already have committed that they're willing to share that data back with us. And so we also are in the process of creating a data commons primarily for rare cancers where that data and clinical data can be stored, but also will be tied to any tissues, any models that are out there to give kind of a fuller representation for patients and for research. Something that will be starting at the end of next month is custodial biobanking. So if a patient wants to store some of their own tissue, again, particularly in rare cancers, sometimes it's challenging to get tissue out of a hospital, even though a patient may need it for clinical care at another institution. So there will be a charge where the patients will cover our cost on that, but we will have custodial biobanking. On the right-hand side are things that our work is helping to create is models through working with various research partners, the data sets. We do have part of the organization that is also helping precision medicine for particular patients, and there'll be more information about that forthcoming in the next few months. It's been a program that we've been working on for 2 years. Quite frankly, the initial patients that did that, where we basically helped get them personalized vaccines and some other kind of one-of-a-kind therapies, was for fairly wealthy people that could afford it. We now have found a way that we can bring the price down significantly to help those patients. So we'll also be doing that. And again, all of this hopefully helps make pathways to cures. And then the learnings and data that we collect will be in open source data commons. So some of the things, whether you work through us or you work with an academic institution or you work with another group in terms of getting patient tissues, there are a variety of considerations that need to be made. Key are regulatory ones. And anything that's doing with human research studies must comply with the regulations and ensure the rights and welfare of participants. So all of these studies require an institutional review board review and approval. And in most cases, that means they need to review the protocol. There needs to be an informed consent. Recruiting materials that are used to recruit patients have to be approved, and security measures to protect any of the patient identifiable information needs to be reviewed. And you can utilize, like most hospitals, academic institutions have their own IRBs. And when we created Pattern back in late 2016, we used Dana-Farber because we were shipping samples to the Broad Institute in Cambridge. And Dana-Farber is an excellent IRB, but we couldn't change one word on a social media post without going back through them, like literally one word. If we had changed a word and not gotten their approval, technically it was a violation. So we worked with the Dana-Farber IRB for the first maybe 2 years. And then as we started expanding our reach and working with a lot of other research partners, it just got too cumbersome to try to keep track of every different IRB. So we, for our work, have switched to an independent IRB. There are a number of them. We are currently working with WCG IRB. And so we have a protocol and a consent that allows us to ship the samples to any of our research partners. We just sign them up initially with the IRB under our protocol, and then it just streamlines the process. Whenever we want to make changes to materials, whenever we want to change the website pattern.org, we're able to do that in a streamlined way. And we're also getting much smarter about the language we use, so that now every time we add a partner, we don't have to go back through the IRB to get them to approve that. Every time we change a word, we don't need for them to review that. Also, if you weren't aware, there are different approval processes for the US and Canada, like WCG IRB. You can get a US protocol and you can get a Canadian protocol, but they're two separate groups and two separate costs for that. And then if there are any protocol violations or incidents, those have to be reported back to the IRB. You have to follow federal, state, and international policies and guidance. Currently, we're not working internationally, so federal and state, but trust me, those can change. And so you need to work with an IRB that is familiar with these things. Like one area, particularly if you have pediatric patients, is the age of consent is different in different states. So you have to make sure that you're covered in the state where that person is consenting. What we do to try to make that easier is we follow one process. We make sure that anybody under the age of 21, that besides the consent online, we also verbally talk to the guardian. Usually it's a parent. It is critical if you are getting patient tissues that the legal guardian has agreed to it, particularly if the parents may not be married. There have been times, in fact, our PI for the Pattern study he's a pediatric oncologist, and if you don't get the legal guardian and the other parent isn't happy with the tissue being donated, it can cause quite a liability. So you need to make sure that there's a legal guardian, and then depending on the ages, if the child's of a certain age, you need their assent. Basically, if they're old enough to understand at least a little bit about what you're doing, they also have to sign an assent. And if research is supported by basically any federal funds, at least currently, that follow the common rule, which is basically rules that were created, I think it was in 2017 or 18, to protect patients. You must have an active SAM registration, and you must have a federal-wide assurance. Whoever you're working with that is collecting the samples, just make sure that they have that if they are getting federal funds. If they're not, then basically any tissues that were collected when they didn't have that, there could be a problem with researchers using them. Electronic signatures have different regulations that need to be followed. There may be tissue and data use agreements that need to be signed. If you're planning on exporting any of the tissues overseas, there may be laws for that. And then, of course, with AI coming on the scene, there may be special regulations for that. In terms of consent language, so the actual document that patients or their parents sign, it's really key to put into those documents cost and benefits. So if there's any cost, we don't charge patients unless they want to store their own tissue in the biobank. So you specify if they're cost or if they're getting paid to donate any tissues. But it's also important that if there isn't any cost or participation payments, that's put there in there as well. So just be very clear that in a consent that the patient or their family knows. Also, risks to patients need to be clearly spelled out. Even just collecting blood, there can be bruising, whatever, that needs to be in there. They need to know that there is the potential for a breach of protected personal information. Hopefully that is not going to happen, but we have been advised by the various IRBs that we work with that is very critical. And then genomic testing, if that is being done, and if it's being done in a CLIA lab, will any of those results be returned to the patients? In certain cases, they may not be, and that's fine, but that needs to be spelled out. Also, if you are returning that or a group you're working with is returning that, there could be major implications to that family, as most of you in the room know. And so just be aware of that. Vulnerable groups such as pregnant women, prisoners, or those with diminished intellect. We're at pattern.org, particularly for rare diseases, rare cancers, we are able to collect from those populations, but a number of studies may not want to collect from those populations. And there also may be language barriers if your consent, our consent currently is only in English. Our website is only in English. So if it's a Spanish speaking only patient, probably we shouldn't have them consent because we're not sure that they fully understand what they're consenting to. Tissue types, ensure that the consent gives the capability for whoever's collecting that tissue to collect whatever type of samples you need. And also look forward in terms of what you're planning to do with the tissue, not just today, but what may be your future needs. Because if you have a patient consent today, and let's say in 2 years, you find that you still have some of that sample, and there's something else you want to do with it that you didn't think about, you may not be able to do whatever that new work is. So try to think ahead if you can in terms of what might be coming down the pike, and using language that is broad will protect you the most so that you can be specific, but you should also have expansive language for things that you may not know about today. Think about commercialization, and if you're planning to commercialize, which I think a lot of people in this room would be, make sure that that is in the consent. And then who has intellectual property rights? We at RCRF, we aren't involved with intellectual property rights, but that is a point that can be kind of sticky with a lot of groups. Look at, as I mentioned, current and potential future research and assays that you might want to do. And really key, because we learned the hard way with our initial consent and protocol, we didn't have in there that we could recontact the patients. It's really critical, we think, with rare cancer, rare disease patients, that there's the opportunity if a patient wants to opt into this, that you can contact them in the future for additional studies that may come down the pike, either at your company or another company, and also for contacting them to make sure that their contact information is up to date, and that there is a backup contact. Again, with rare cancer patients, some of them may not be alive in a year or two, and so you need a backup contact if you have some of that tissue remaining, or there's a finding that has come out of the research. If you and your company or your foundation have decided that you want to be able to give that information back to patients, and it's, again, fine if you want to, fine if you don't, but make that decision before the patient's consenting. And as I mentioned, for research-only results, historically, you haven't been able to give those back to patients. There have been some situations where now you can. So make that decision up front if that's something you want to be able to give back to the participant. Logistical considerations for tissue collection, since we collect literally from anywhere in the US, including Alaska and Hawaii. We are technically not a covered entity, but we still follow HIPAA because we feel that's what we need to do to help protect the patients. We assign participant and case identifier codes so that the researcher receiving the tissue doesn't know who the patient is. We are starting to use GUID coding, which is a special participant code. One of the challenges with those are there are various companies that do that. That helps track what studies a participant is participating in, but with different companies using different codes, it's a little tricky. And so we're going through all the learning process of that now. And then some studies, particularly some that have gone with NIH, there was a Human Cancer Model Initiative program that we were a part of a few years ago. Models that were created from samples that we sent to the Broad Institute that are now at ATCC. We aren't able to tell which came from our samples because they mandated that information was delinked. Sample transfer requirements, depending on what type of samples you want, make sure you're clear as to how you want them sent, whether they're frozen, ambient, at refrigerated degrees. Make sure you're clear with who's ever sending those. Saturday deliveries are kind of a big thing in our world because there are a lot of surgeries that occur on Fridays, and most labs aren't open on Saturday. So you have to make sure what's going to happen with those Saturday deliveries. Contamination risk reductions. There are some labs that don't want to deal with certain diseases because of potential contamination. So just be aware of that. Post-mortem collections, we get asked about this a lot. They're doable, but they are very challenging, particularly if we're looking at solid tumors. And home collections, where blood draws are usually done. Really the only risk that we have there is if whoever is doing that blood draw does not cover up the label as we instruct and leave if the kit went directly to that patient's home and their name and address is on there, that's not good if it gets to the research lab because that's technically a violation. So you just have to make sure you cover those bases to make sure you try to reduce the chance of that. And then if required, sign material transfer agreements and use agreements. The way we get tissue to various research projects is through the use of a fairly basic material transfer agreement. And that's it on pattern. We really, just like Irene said, we put patients at the center of everything we do. And we appreciate your time listening today.
Thank you, Barbara. So we having just heard about the power of empowering patients to help assist us in the discovery process. Our third speaker today, Dr. Emil Pindon, is going to share some insights into the complexity in models and preclinical platforms in rare disease. Dr. Emil Pindon is the Chief Scientific Officer of the Myhre Syndrome Foundation. She earned a PhD in neuroscience from Paris Orsay University and has conducted research at renowned hospitals in both Paris and London, as well as having a extensive career in the pharmaceutical industry. As project and group leader in preclinical drug discovery, Emil has honed skills in cloning, molecular pharmacology, and innovative screening technologies. In the past decade, she has dedicated herself to rare disease, supporting patient-centric organizations with drug discovery strategies. Her work includes creating preclinical platforms and tools to help develop tailored treatments and options for patient needs. So welcome, Emil. Thank you.
Thank you. Thank you very much for having me at the iXCells. So I'm going to focus more today about multisystemic rare disease. That's I think something that is quite common to different diseases. Thank you. No, that's not the right one. Okay, so Myhre Syndrome Foundation is a patient advocacy and research organization that started in 2019 with 2 patients getting together. We are focused on the 2 aspects, community service, but also research. And recently, the research aspect has been really focused on drug discovery. So we want to have the right path and the right tools and how to choose the relevant drug discovery tools. Whatever the goal is to improve quality of life or decrease the life-threatening events or cure the disease, what we need is really to identify targets, models, and assays. So starting with the models, what everybody wants to say is, okay, we want to screen drugs and validate drugs in a model that recapitulates the disease. Well, that's not for us. Because unfortunately, we have multisystemic disease with a lot of heterogeneity. And this is a common issue with rare disease genetic disorders. So the molecular origin of Myhre syndrome is a mutation in the SMAD4 gene, which is also called co-SMAD. SMAD4 is a transcription factor integrating the TGF-beta pathways, and TGF-beta is a superfamily with 33 members and 12 types of receptors. So that's at the end of a lot of different pathways. Specifically, Myhre syndrome is missense mutation on only 2 localizations, the 500 and the 496. And the 90% of our patient is comprised in these 2 mutations, and the rest of the 10% is still at the site of the 500 with 3 others mutations. So the particularity of the mutation SMAD4 for Myhre syndrome is that it's a gain of function through an overstabilization of SMAD4 and the other SMAD complexes. So a gain of function of any kind of TGF-beta will act on a lot of different cellular and functional particularities. So from cell differentiation, and from people who do stem cells differentiation, you know that there is the SMAD dual inhibitor protocol, cell growth, cell proliferation, extracellular matrix production, angiogenesis, tissue repair, immune regulation, and apoptosis. So that's a lot. If you increase the function of all of that, that's a lot. In our case, we have to be very careful in the treatment we choose and the methodology we choose, because SMAD4 has an haploinsufficiency, and the loss of function is responsible for other diseases. So we need to decrease this this gain of function and overstabilization, but we need to be careful not to lose too much of the SMAD4 activity. So just to show you the complexity of the situation, the superfamily of ligands is TGF-beta, bone morphogenic proteins, BMPs, the activins and inhibins, nodal, anti-Müllerian hormone, growth differentiation factors. So that's a lot. And you have different type of receptors which are classified in two types. The type 1 is the ALK, so the activin-like kinase receptors. And the second group is the type 2, which is the BMP, anti-Müllerian hormones, or TGF-beta receptors. So you have a dimerization and then an association between the two type of receptors. And then you have the signal transduction that we call the canonical signal transduction from TGF-beta that goes to the SMADs. So some of them will go to the SMAD 2 and 3, and other will go through the SMAD 1, 5, or 8. But at the end, all of them needs the SMAD 4 to form a complex, enter the nucleus, and act as transcription factors. The thing is that this complex with SMAD is only a half-unit transcription factor, and you need the other half that can. We have now over 40 cofactors identified with the SMAD complex that goes from beta-catenin, TCF-LEF, with signal. So many different other signal transduction associated that we need to form the second half unit of the transcription. So you can imagine that we have a huge cell type and context dependent activity of the SMAD4. So from there, you can imagine that we have a multisystemic disease. We have neurodevelopmental deficits with intellectual disability, autism, speech delay, and nonverbal patients, some nonverbal patients, skeletal abnormalities, neuromuscular, also, it's not only skeletal, it's it's musculoskeletal, sorry. We have fibrosis, whoops, that was the wrong one. We have fibrosis and stenosis that trigger life-threatening, and in fact, the last 6 months, we had 3 deaths in kids because of tracheal stenosis and aortic stenosis. And we have widespread fibrosis in various organs, even in liver fibrosis. We have hearing loss, cardiovascular condition, respiratory condition, gastrointestinal condition, immune dysregulation, endocrine, reproductive system abnormalities, and even solid tumors. So from there, you say, well, what system am I going to take? And on what model should I focus on? But that's not all. If you find a system and you say, okay, I'm going to that system, we have heterogeneity in the system. So we have, okay, we go for neuro, we have almost 80% of seizures, but that's cumulative. You can have several of those conditions. But not all of them. Every patient is going to be different. The developmental delay seems to be something quite common. So if you go to other type of system, for skeletal or hearing, the same, you are going to see, again, a lot of heterogeneity of the phenotype that you can have. And in hearing loss, even if you have 53% of the patients that have hearing loss, we have half of them that are conductive, but we can even have some rare cases of vestibular schwannoma, like in NF2. So that's really a big issue on the model selection. Same for cardiovascular, respiratory. I'm not going to show you go through all of them, but a lot of heterogeneity. So what can you do? Where do we go? Well, we decided to go to the patient and look at the patient priorities. So through surveys, we identified that the cardiac manifestation and respiratory were the main two issues where the patients wanted to have some drugs. So I put in 4 fibrosis and stenosis because in fact the cardiac manifestation and respiratory are mainly due to fibrosis and stenosis. And the third aspect that the patient really wanted to focus on was behavioral. And if you go to the one of the patients that was mentioning what altered in adolescence was a shift in his cognition, and he developed psychosis. This is difficult to manage as a family, much more challenging than heart surgery, which he's had. So the behavior was really a big issue, even bigger than some cardiovascular. So we decided that that would be our focus: fibrosis, stenosis, and behavioral. To have the model, so we wanted targetability and modelization. The model has to have a continuity and a correlation at every level, from biochemical, cellular, organ, system, or whole organism. So the 3 questions that we ask ourselves is what is the pathway in those fibrosis and behavioral? The mechanism responsible for the phenotypes we have. Is the model sensitive to this pathway and relevant to the application? And do we have reversibility in the model we choose of the phenotype of the disease? Is it possible to have reversibility? Because we have also adult patients. So we applied some model selection criteria, which was we wanted a limited engineering intervention. So we went for the patient-derived iPSCs. We wanted feasibility and timeline, not a model that is going to take 1.5 years to get ready. Reproducibility and standardization for the screening. A good throughput, so that it won't take 6 months to test 100 compounds. We wanted an assay available or easy to further develop or validate in our models. And we wanted a relevance to the identified targets. So behavioral and fibrosis, that's still very large. Could be, is there, so we ask ourselves, is there a coherent link between behavior and fibrosis? And in fact, we started from fibrosis. And what's fibrosis? It's wound healing that goes wrong, that goes over what it should be. So we thought, okay, that's peripheral system response to injury, that's fibrosis. What's the CNS response to injury and stress? And what's the link with the TGF-beta? So for the peripheral system, if we go for fibrosis, you have the choice of different cellular model. You have fibroblast, of course, that's the first thing you think about. But you have many other cell types, such as endothelial cells, epithelial cells, that all go through reactivity and transformation that we call the epithelial mesenchymal transformation. And this transformation leads to the creation of myofibroblasts, which is the issue in fibrosis. What about the CNS? So what's the response to injury? Well, that's not neurons. That's microglia that is modulating and responding to injury and or diseases. And the other one is astrocytes that also goes through reactivity and scar formation. So we were thinking, okay, so for the CNS, let's focus on CNS. It seems that microglia and astrocytes for our behavioral model might be more appropriate than neurons. So we checked with the TGF-beta and CNS, and we see that TGF-beta is a regulator of microglial function, inhibits phagocytosis and myelin clearance. And we thought, oh, okay, myelin clearance. In our patients, we have, in the MRIs, we have some white matter hypersensitivity, which can be a sign of demyelination and myelin issue. So that was one more point for microglia. TGF-beta is also involved in the astrocytes reactivity and synaptic plasticity. It's involved in different pathogenesis of neurodegenerative disease, but that's not our goal here. We have also a pre- and postnatal macroglial cells that is regulated by SMAD4. SMAD4 is critical for driving the transcriptional signature of the microglial cells. So that was, we thought we were going in the right direction, but something that was really important for us was, what about the adults? What about reversibility of the model? Can we have a model that can show reversibility of the phenotype we have? And there is a publication on molecular diversity of the diencephalic astrocytes, reveals that adult astrogenesis is regulated by SMAD4. So that was one more point to go in that direction, but we were asking, in fact, okay, diencephalic astrocytes, are we really, do we really want us to have that? Is it relevant for our disease? And in fact, if you look at the diencephalon, which is really at the center here, you see that you have the hypothalamus, which is involved in endocrine system. And those hormones from the hypothalamus are the hormones that are involved in Myhre syndrome disease. The thalamus, where you have motor function that we have issue with in Myhre syndrome, and sleep and sensory perception. So sleep is a huge issue with Myhre syndrome patients. Again, the epithalamus is involved in the regulation of sleep, which is one of the big issues. And we have a certain amount of our patients that are already treated with melatonin, and so some improvement in the sleep. So, and the last part is the subthalamus that is involved in movement, and there are movement issue also in Myhre syndrome. So that was really to reinforce us in the astrocytes. We said, okay, astrocytes too. What about the fibrosis? So fibrosis, I told you, there's a lot of different origin of the myofibroblast. The first thing you think about is the fibroblast. Okay, let's go for fibroblast. And then you see, again, we go from a multisystemic heterogenic disease to a huge heterogeneity between fibroblasts, intra and extra organ heterogeneity, and a big difference between fibroblasts from human to mouse. So we said, well, could we avoid fibroblasts? So we went back and looked at the different cells in the lineage of the cells, and mainly we are affected by all the different MSCs, differentiated cells. So we could go for all those different cells that would, in fact, at the end, give some fibrosis. So we went back again, asking, what about the patients? What's the burden, and what's the big issue for the patient in fibrosis? And the main one is vascular fibrosis that trigger stenosis. So we decided to go for vascular endothelial cells, and the assay will be the endothelial mesenchymal transition and wound healing assay. So we finally had our preclinical platform designed. So we sent the blood immediately to iXCells and got the iPSCs. The in vitro parts of the platform comprise the iPSCs, reprogram vascular endothelial cells, reprogram microglial cells, and organoids. And we are going to work with Alison Morty, hopefully. So again, this name coming up for brain organoids. We have also other organoids for adapted more for fibrosis. The in vitro cellular assay we have binding assays and more biochemical assays with measuring the stabilization or destabilization of SMAD4 SMAD complex here. And then we have the functional assays from the cells, which is, as I said, fibrosis, wound healing, and glial reactivity. We hope that the astrocytes will come when we have more funding. The in vivo models are conditional knock-in mouse model. One is already available at Jackson Lab, and the other one will be on the way. The in vivo assay will be on fibrosis and stenosis, but we are starting deep phenotyping with psychogenics to really get the right screening assay specific to Myhre syndrome markers in behavioral and other characteristics. So this preclinical platform is really at the center of our pipeline, and it's really fundamental for us. So we have our treatment portfolio for now is composed of small molecule repurposing and new chemical entities, around a bit more than 100 that came from the IRB in Barcelona. Sorry, in Barcelona, the IRB Institute. They were selected, the hits were selected on the destabilization between SMAD4 and SMAD complexes. We have peptides from a company in South Korea. Our big effort is in immunocell therapy, so FAP CAR-T therapy to reduce the fibrosis. And the gene therapy editing had the first good results and is going into the iPSCs now. And the more translational research is on the neoprotein-protein interaction between the mutated SMAD4 and new cofactors, and the new brain organoid with Alison Murty and new therapy in development. And hopefully going through the preclinical platform will allow us to jump into our clinical platform that we are working on. So this preclinical platform, we hope, could be expanded to other TGF-beta related diseases, just that have fibrosis, vascular condition, neurodegeneration, or glioblastoma, or others, where you have, in fact, with Myhre syndrome cells, a kind of natural overexpression. It's not overexpression, but gain of function. So on over-stabilization and over-signal of all the TGF-beta. So that's, I think, a good way to screen and read any treatment that could decrease that. So that's, so I just talk about the first efforts. That's our preclinical, but we are working also on clinical readiness, and those are the treatments, our pipeline of treatment that we are working on now. And we work also, of course, on the regulatory and industry. Okay, thank you.
Thank you, Amel. Wow. We talk about the complexity of getting the cells, and then when we get them, the amount of work to come up with a relevant model there, it just really hit me. So thank you for that. As we move into the final speaker in this session, it is my pleasure to introduce Dr. Kitchener-Wilson. Kitch is the CEO and co-founder of Rosebud Biosciences. He is a physician scientist with 16 years of experience merging human iPSCs with genomic and molecular technologies, particularly in the cardiovascular applications. As a Stanford-trained molecular pathologist, Kitch was an attending physician at Stanford for half a decade before founding Rosebud. He holds a BS in mechanical engineering, a PhD in bioengineering from Stanford, and an MD from Duke. It's my pleasure to introduce Kitch and his presentation on scaling stem cell-derived organoids for disease modeling and drug discovery. Thank you.
Great. Can you all hear me? All right. So I am a co-founder CEO of Rosebud Biosciences. We work with iPS cell-derived organoids This comes from many, many years that I did in academia. I jumped ship to the detriment of my colleagues there to start a company around this. And I do truly think that this has a place in drug discovery. It's at early days for organoids. We've seen a few people mention them. What we really focus on is scaling them and homogenization, precision, quality control in terms of organoids. And it's organoids broadly. And I'll show you what I mean by that. So I show this slide to all the investors that I pitch to. I think this is really what we do. And if you take anything away from my talk, this is it. We have organoids that are mimicking fetal development. Remember that these are iPS cells. Anything derived from an iPS cell is fetal-like. These are not adult tissues. This is not a 6-year-old person's cells in a dish. However, with genetic diseases, you can frequently find a phenotype in these organoids that mimic what happens either in utero in terms of development or shortly after birth. So we introduce mutations into iPS cells. We have our own secret sauce for deriving them in a dish. I'll show you a little bit about that. And then we do longitudinal monitoring every single day. I would love to do it real-time monitoring someday, but we have longitudinal monitoring every day looking for a disease phenotype. This is what they look like in a dish. So this is a 14-day protocol for deriving cardiac organoids. I'll show you a little bit more of that later. These are just bright field images once a day of a developing cardiac organoid. There's this, as far as organoids, I think that there's a lot of, people say organoids. It can mean a lot of things. It could be cancer organoids, maybe little microbiopsies of tissues. For us, these are actually attached on matrigel, so they're relatively flat. So they're not necessarily 3D, they're 2.5D, if you will. So there's a structure to them. However, the reason we like these, and this was a 2014 publication that really kind of set the field ablaze, these are attached circular micropatterns of iPS cells, 2 millimeters, 3 millimeters across, all on standard tissue culture plates. They go through embryogenesis. So you can actually see concentric circles. Something laser. No, that's laser. Anyway, that's which one? Oh, that little one. Okay. You can see right here. So you can get endoderm, ectoderm, mesoderm, all in concentric circles here, and these are regular. So that means the thing is not just a homogeneous group of cells, but they're actually going through embryogenesis. So we like this because you can scale these much more easily than the spheroids that everyone thinks about with organoids. The other thing, before I move on to this, the other thing about this, because they're relatively flat, makes imaging much easier. They are static. They're not floating up around. They're not spheroids, so you're not getting necrotic cores with them. Diffusion of nutrients, drugs are through the entire thickness of these 2.5D organoids. They make them a lot easier to work with. So these are some cardiac organoids. I come from a cardiac background, so you will see a little bit more of a cardiac bent to this talk, but we have more tissues that we do. Single cell RNA-seq. You get, because of that, it's going through embryogenesis, all the multiple germ layers. You're getting cardiomyocytes, you're getting fibroblasts, you're getting endothelial cells. There's even some neural progenitors, which are that rudimentary conduction system with them. So they are very diverse cell type, tissue types. So I think of these as tissues, not as homogeneous cell cultures. There's a little bit of a busy slide. The take-home point for this is rodents, 2D cell cultures have been the mainstay for decades, for probably half a century for drug discovery. You go from 2D to rodents. You do your preliminary tests in 2D, then you go to rodents. What I think is that these organoids are going to bring any complexity, and in the near future, they're going to be that gap between the 2D and rodents. In the future, I'm a maximalist on these things. I think that they are going to replace all of those, and organoids in the 21st century are going to be the primary drug discovery platform. That may not happen in the next 2 years, but I think in the next 10, 15 years, you're going to start to see these being the mainstay for drug discovery. These are rich, heterogeneous, macroscopic as opposed to microscopic tissues in a dish that have mutations of the patient. They show disease beautifully. I think that as long as we can standardize them, agree on particular quality controls as we do with our 2D cultures, that they really are going to be the mainstay. So this is a little cool video. They lit up our laboratory with some fluorescent lighting to make it look like a nightclub. We're very excited about this. This is what we have. We have a robotic arm. We grow our organoids in 96 well plates. And you can see our robotic arm. We have a microscope in the background that's moving them around each day. So there's a liquid handler. They're getting fresh media. They're getting bright field images every single day. Once we put a plate of micro-patterned iPS cells in these plates into this system, it's hands off. Humans are the biggest source of error in terms of culturing these iPS cell-derived organoids. So we've tried our best to remove the human component from actually producing these organoids. So this is automated. It works on weekends, it works on vacations. So it's wonderful. We're a team of 3. That's the entire team there. We can produce these in the thousands. We have a capacity for about 10,000 organoids that we can do simultaneously. And that includes longitudinal imaging every single day, media changes, you name it. So the real idea is to use this as a drug discovery platform. And of course, my co-founder, who is big in AI, we are training AI. What we want to do is train it on bright field images of organoids. This is similar to what Recursion is now doing, and get away from the need for immunostaining, biomarker staining, that sort of thing. So just training on bright fields. That's our long-term aspiration. We're not quite there yet, but I think that's where we're going to go with it. I'm going to give you 3 examples. So this is dilated cardiomyopathy, which is my background from my academic days. Hearts get enlarged. They're not able to pump blood. There's probably 50 genes or more involved in dilated cardiomyopathy. It can also be environmental, all sorts of issues. These folks generally need transplants within a year or 2 of getting diagnosed. Mortality rates are atrocious. About 50% die. There are no targeted therapies for this disease whatsoever. So this goes back into my academic days. I found a gene that's associated with DCM. It was upregulated in DCM, dilated cardiomyopathy. This was now 10 years ago. And this led to the whole saga of trying to figure out what it does. The challenge with this particular gene is called Banker, which is a long non-coding RNA. It's primate specific, so it's not found in mouse. You can't do a mouse model with this. It's also fetal specific. It's only expressed during fetal heart development. So how do we get around actually studying what it did, since it seemed to be associated with DCM? Well, HESC, this is human embryonic stem cells, iPS cells, are the same thing. Derived cardiomyocytes also express that because remember that they're fetal-like. So I thought, after a lot of work, it looked like migration. It caused cardiomyocytes to migrate based on a lot of RNA-seq data and some other reports that we had seen. So I did a swoon scratch assay, which is usually you do with endothelial cells and fibroblasts. Very unusual to do that with cardiomyocytes because you don't think of them migrating that much. And I found a little bit of a difference. It wasn't very satisfying, however. You can see just a little bit. There's a p-value. I think it was a 0.01. That was probably on a good day. I didn't really think that that was the true phenotype, and I needed something better. So what we did is we moved on to organoids. I had a colleague that was working on these micropattern organoids at the time. And this is where you saw this beautiful phenotype in a dish that you could see with your eye. And this is actually the source of the name of the company, because they look like flowers to me. So what's happening here is the green are the cardiomyocytes, all derived from, in this case, HESCs. They've been micropatterned in these arrays. This is using an older technology, stencils, but this is something we've come around with later to make it more high throughput. And we're overexpressing this particular gene. It causes migration of those cardiomyocytes, which you can see kind of like a blossoming flower. And here's our control with the scramble control. This, to me, is what sold the deal for organoids and their ability to show a phenotype far superior to what you would see with single 2D cell cultures. And this is what tells me that organoids have that macro ability to show disease, which you could almost see with an eye, with a beautiful recapitulation of what a disease of in vivo disease. So we've scaled this up. Let's see if this works. Do you see that? So we work in 96 well plates. These are cardioids, differentiating them. Homogeneity is a big deal for us. I'll show you a little bit more about what that means. We've introduced mutations for DCM. So on the left, you can see are our beating disease ones. On the right, I'm sorry, let me see if I can show that again. On our right is the disease lines. On their left lines are wild type. You can see they're kind of squirrely looking. So we're really trying to get high throughput homogeneity between batch to batch with these things. High N is really what we're going for here. This is, we'll look at the single cell level, single organoid level. You can get beating rates, you can get magnitude of beating, you can get percent rating, and then you can see the disease at the bottom is the disease isn't beating at all. So beautiful data that you can really get with these organoids. We moved on. You know, starting the company, I did not want it to be just a cardiac company. We're like, there's protocols for liver, there's protocols for kidney, there's protocols for brain as well. This is a brain, I'm sorry, a liver. This is for a disease called Alagille syndrome. You get a paucity of bile ducts in these kids. 60% of these kids ultimately need a liver transplant. There is no targeted therapy for these whatsoever. It's a well-defined genetic disease. However, JAG1 is the actual mutated gene here. So here's a hepatic organoids. I'll just point out, you know, they have vimentin is our fibroblasts. Here is our bile ducts. CD31 is our endothelial cells. Albumin are hepatocytes. So there's a diversity of cell types in these. You can also see secretion of albumin by these. So this is actually the media of these organoids that you can sample and run ELISAs on those. They also metabolize urea, and you can detect urea in the media of these. So they really are functional little units of microtissues. So here is where we introduced a JAG1 mutation that causes Alagille syndrome. And I told you that there's the bile ducts. There's a paucity of bile ducts, meaning these kids aren't able to produce bile ducts and move bile through their livers. And so here is wild type, where you see actually normal ring-like bile duct epithelium. These are a few millimeters across, but you can see that regularly. This is a stain for actual bile ducts. So that's wild type. Down here, you see our mutated line. Virtually gone. There might be a few little hints of bile duct here, but for the most part, they're gone. This is something you would not see at the single hepatocyte level or the single bile duct level. This is something you see at the macrostructural level. And so this is where organoids really come into play. Third and final example, Fabry disease. So this is a disease, there's actually a lot of interest in this disease in pharma. There's quite a few companies working on this. There's shortened lifespans for these folks, about 50 years for men and 70 years for women untreated. What it is there's a it's a lysosomal storage disease, multisystemic. You have a healthy GLA gene. It produces alpha-gal enzyme. It metabolizes this sphingolipid called GB3. When it's mutated as GLA gene, you have low amounts of this alpha-gal gene, and you get buildup of GB3. This could be multisystemic. Typically, what these folks die of is either heart failure or kidney failure, but it's multisystemic, so it can occur in lungs and elsewhere. A lot of interest in this. So we were approached by a pharma company that wanted to model this. Apparently, the mouse models do not recapitulate the disease pathology to their satisfaction. And so we said, okay, we have human organoids. Let's try that. We knew nothing about kidney at the time. We developed a protocol for developing kidney organoids in a dish. We introduced 3 different clinical mutations in the GLA gene that are known to cause Fabry disease. We also have a knockout line. GB3 is our marker for the buildup of that sphingolipid, which causes disease in Fabry disease. And then there's a variety of kidney markers. You can actually get little proximal tubules in here and podocytes too, which is pretty cool. So this is what they look like. On the right is what a healthy kidney organoid with a functioning GLA gene that metabolizes its GB3. This is a stain for GB3. On the left here, see all these red speckles? There's a bit of red background here, but you can see those bright red speckles. That's that buildup of the sphingolipids. And so we could get this in this was about a 3 to 4 week protocol from start to finish, and we could produce these in the thousands. So that's been exciting for us. We can now do drug screening and all kinds of things with this. These are human, remember, these are not mouse models. So this is just an overview of what we are focused on as a company. I've shown you heart, I've shown you kidney that we've done. We have this liver program, we have an SBIR funding that. although we'll see if that continues these days. And we do have some interest in Huntington's disease in the brain. We do have some preliminary data for brain organoids too, although it's kind of early days for that. Ultimately, I want to do all of these tissues. If there's a protocol for differentiation, you can do modify it for a protocol for organoids. We are not spiking in cells. We're not creating 3 different cell types and mixing them together to create these. These are all assembling by themselves as iPS in our 96 well plates using our different factors that we add to the media. So relatively simple. If you ask AI, what does this is a Grok actually, what does an automated organoid factory look like? And it gives you this. It looks a little dystopian, I'll be perfectly honest. And I think my father had one of these in the 1970s for playing music. But there's some things about this that I do like that captures where we're going for Rosebud, our company. As a pathologist, you think about precision. And precision is consistent between repeated measurements. And that's always been an issue with biology, and particularly when you're having humans culture these cells. If you remove humans from, I love humans, but if you remove them from the actual work of producing these organoids, you get better batch to batch and repeated measurements. Accuracy, are they close to the true value? There's a very low signal to noise in a lot of biological research. We're trying to increase that delta between signal and noise. And so this automation helps that. High throughput, of course, pharma wants high throughput. They don't want little 6 well plates of single organoids. They want thousands, preferably tens of thousands, to really produce high-end statistical value. So that's what we're going through here. Modular. Technology changes. Microscopes get better, liquid handlers get better, robotic arms get better. I don't want a $4 million box that I have to buy from a company that has everything that breaks down. By the way, I have to send a technician to come fix it, but that is stuck with the same microscope, stuck with the same liquid handler, and I'm never able to upgrade. So I like the modularity that this actually image shows, and that's what we're going for as well as company. Interrogatable, meaning you can change the conditions, change the media, change the factors, change the environment of these organoids to produce different data to create disease organoids. And then real-time monitoring. I really think that it's important to detect disease as these things are developing in a dish. And when is the moment when you can first see that detection of disease and that diverges from health? And a lot of AI is going to be involved in this real-time monitoring. So I think that's where we're going. I'll wrap it up here. I don't think this audience needs any introduction to rare diseases. I tend to use genetic disease for iPS disease modeling. There needs to be a genetic component, in my opinion. If there's no known genetic mutation or gene involved in the disease process, I would be hesitant to get into it because it could be environmental, and it's tough to decide what is that environmental perturbation going to be to elicit the phenotype that you want. So I call it genetic diseases. And rare diseases, I think it's about 80% are known to be genetic. So that's my preference, to call them genetic diseases. Our goal, high throughput, precise, accurate phenotyping of the human genome for target drug discovery. And our working hypothesis for this company is that 3D tissue is greater than 2D homogeneous cell types for disease modeling. And human data is better or equal to rodent models. I think ultimately they will be better, and that's our competition, our rodent models, to be quite honest. What these do lack, however, is immune system. There is no immune system in these organoids, and that's true of any 2D culture. So we have to find creative ways to get around that in terms of modeling the immune system. So we're sticking with genetic diseases of actual organs for now. This is my team, myself, Evan, and Aditya. We are a Y Combinator company from 2 years ago. We have a few investors in it. We're based in San Francisco. If any of you are interested in organoids, please reach out. Thank you.
All right, thank you, Kitch. We have a few minutes for questions. We'll take questions in the room first. If I could just ask the speakers just take a seat at the front, just makes it a lot easier with questions. Okay. Wow. Okay. Just take that one over. I'll pass this microphone over to the panel. Thank you.
Thanks. That's great talks, everyone. I've got one really sciencey question for Kitch, and then a broader question for this group. Think about standardization. Kitch, it was great seeing the single cell RNA-seq data with the cardiac tissues, especially. Thinking about that and the interplay between different cell types driving disease, have you guys considered omics-based or CRISPR pooled screens to look at what individual subtypes are driving phenotype? I was especially thinking about your cardiomyocyte single cell type not showing phenotype, but when you mix 9 cell types, you see a very striking phenotype.
You're thinking about Perturb-seq and
Perturb-seq.
Yeah, we'd love to do it. It's big money for a small startup, but yes, we'd love to do it. High throughput CRISPR knockouts of 25,000 genes, seeing what each one does in the different subcellular types. My ultimate goal, like a pathologist, was to have single cell RNA-seq as our disease readout for a lot of these organoids. It just comes down to cost right now.
There are some newer ways you could think about the inducible Cas system in your IPS that you then turn on once you're in your organoid stage.
Absolutely.
Throw your pool in, do a crop seq and look at.
We'd love to do that. Yeah, absolutely.
And then broader, everyone mentioned standardization, scalability, reproducibility. Leveraging AI and thinking about all these machine learning techniques, we need to have some sort of commonality to start pooling data together from multiple different platforms. What do you think about the scientific industry needing to do differently so that we have shareable, combinable data that we can start to identify patterns? And rare is no longer rare. Rare is just a different way to get to 5, 6, 8, 10 phenotypes.
I think you put the finger on the words sharing data. I think that will be the main issue. That is already a big issue to share data inside a single community of rare disease. So I have difficulty imagining how we are going to do it in between different community. Maybe there will be less competition between different rare disease, so the will to share will be bigger. But that's it's not only from the scientists that we have issue sharing data. It's more on the side of the IP and all the legal aspect and the legal team in academia, university, or I think it's also an aspect in CRO about sharing data. Then you decrease your IP. So that's I think the next challenge, and I have no idea how to solve it.
I can add a little bit because at the Children's Tumor Foundation, we actually have a data portal hosted by Synapse. And so over the last, I guess, 10 years or so, we've been first encouraging researchers to share their data, funded data, back into one central repository, but more recently we're demanding it. So in recent years, it's part of our funding kind of award letter where researchers need to share their data back, and we have it in a portal. But to take it one step further is, now we need to share it outside of CTF, and so we have initiatives where we're identifying complete data sets that are shareable and putting them on public platforms like CBioPortal is one of them we're working on, maybe Geo at some point. So we first have to identify which data sets are well annotated and formatted and go through all of that work, and then identify which public platforms to share them on. And we're actively doing that now. So.
I'll just second that, making it mandatory. Like, I've worked with a number of research groups that said they didn't want to share the data. And I said, that's great, but then you're not going to get tissues through us. So that changed their mind, and every single one of them said, okay, we'll work with you. I think also patients are key here. Patients understanding the value of their data, patients getting the data if you can't get it directly from the researcher and patients starting to talk to people about, I want my data shared, I think is really key.
Yeah, second all that. In academics, you have to, there's Geo and all the repositories for seek data, but industry side, it's a mess to keep all this data proprietary. And then, our business model is partnering with pharma who themselves don't want to share that data because also the IPS cells themselves, there's incredible royalties and fee structures and proprietary. You can't even screen for molecules without paying Japanese academia for the IPS academia for these things. It's onerous, the amount of boundaries that they place around IPS cells. So what we've got a workaround solution. We're able to create our own disease lines and then generate data from those. I would love to put that data somewhere in the cloud that's accessible to all. Right now, it's accessible to only our partners, which is unfortunate. But yeah, there does need to be some sort of industry-wide paradigm for addressing that.
Okay, I have a question, Kitch. So you talked a little bit about this, your 2.5 versus 3D organoids. But did you see any differences? Have you ever done experiments that shows really the outcome or phenotypes? Is what you see only possible with the 2D versus the 3D?
2.5D versus 3D. I can only tell the practical differences we've seen. They're relatively flat and they're attached. The 3D, they are floating in media. They are not able to get oxygen and nutrients into that central core for those spheroid type things. And so many of them get hollowed out, which can be a challenge with those. And then imaging, meaning that, you're taking serial imaging of these things, because those spheroids are floating in media, each day that you're taking an image of them, it could be in this orientation, it could be in that orientation. So comparing it to between days is also a challenge.
Yeah, no, I get it, but there are some people that kind of optimize, standardized to a little bit at least, organoids when they're free floating, right? So did anybody do comparative studies between what you do and what?
I don't know that. I'm not aware of anybody who's done actual comparison of the 2D versus 3D, other than the diversity of cell types seem to be the same in the 2.5D versus the 3D. You have smooth muscle cells, endothelial cells, fibroblasts in relatively the same quantities as what you'd see in 3D. They all beat. I showed you a lot of the beating. So beyond those metrics, are they more or less mature? How do you define maturity? We can let these culture for 2-3 months if we had to. I'm quite confident they would be similar to the spheroids themselves, which can be matured as well. I honestly don't see a lot of difference, but if there's been a statistical quantitative study of the difference between the two, I'm not aware of one yet.
So I have a Okay, I wanted to ask 2 questions. I guess I'm going to ask one only. Okay, I'll ask the second one later then. I have a question for the NF1 and the Armile, no, that you show that there is a lot of heterogeneity on both those diseases, no? In the case of the NF1, you show that there is so much phenotypic, no? I mean, in the symptoms of the people and also for the SMAD one, for the TGF-beta. So do you think that's going to be make more difficult to find like a common therapy? Or do you think that we're going to need to find different therapies, I mean, kind of subcategorize inside of the disease, no? Different symptoms, and then do you think that we are going to need to find different therapies like small molecules or whatever for these categories, or do you think that it should be able to find something that encompasses the whole disease?
I think you're absolutely right. It's very heterogeneous with a broad spectrum of manifestations, so it will take multiple therapies and mechanisms. We're just talking about NF1 today, but there's a whole other schwannomatosis type that I didn't have time to get into today, but absolutely, it's going to take more than one kind of therapy and more than one mechanism. Ultimately, combination therapies are something that are being looked at. So I think a combination therapy is on the horizon as well, to be able to hit multiple targets of the disease.
Yeah, I think it depends what you target. For us, if you target the SMAD4 or SMAD complex, then you can imagine that whatever the cofactor is next to it, you are going to act on all the different phenotypes. In our pipeline, we foresee that maybe we won't be able to do that. So we are also developing with the Geneton Fab CAR-T, especially for the fibrosis, in case we cannot get to the development of the small molecules. But since we start our first screening on really the destabilization of the complex, then the only limitation would be the delivery of the molecules, if it goes through the BBB or not. Or but if we would go for other like ALK inhibitors, for example, ALK5 inhibitors, that we also want to test, that would be only a part of the phenotypes. But going really at the origin of the issue, I think will allow us to go larger in the targeting of the phenotype. It depends where you go in the signal transduction. And since SMAD4 is really at the end of everything, then if we go for that, we hope that we will be able to tackle many different aspects.
Okay, thank you. I'm just gonna, one quick question from the online team, and then I'll get you all to lunch. Kitch, there was a question in regards to cost. How does the cost compare for the 2.5D versus typical cell and animal models?
Did you say cast?
Cost.
Oh, cost. And between that and animal models? Oh, that's an easy one. They're far cheaper. They're each organoid is less than $5, excuse me, to produce, and you can produce these, in the thousands. So you can imagine the scale that would be impossible with these sort of things. So yeah, they're quite a bit cheaper.
Wonderful. That's great news, because drives the economics of the screen.
Indeed.
So again, I just want to thank all 4 presenters for a wonderful session. Thank you all for your attention and questions. Again, round of applause for our presenters.
Knock, knock. Is it on? Yeah? No, is it on? Is it on? Okay, the halftime show comes to an end. Counting down. Halftime show comes to an end. Okay, you can continue to eat. That's not the problem, but we have to continue with the program. And it's not even fully halftime, because we have, I think, 9 more presentations to come before the sun goes down. So let's try to do it now. And let me just a few words before we go into the next session. Oh yeah, I don't know why I put it down here, but as you immediately figure, there's an accent here, but we had before, right? So like Chinese accent, a British accent, we had Spanish accent, a lot of questions from a Spanish accent, and of course, many others. So this is also a little bit of a reflection of what you find in iXCells. So very international team, and we're looking forward to further expand that too. I think I heard from the speakers some Polish accent, a French accent somewhere, I think. So it's very international, and that's what we want to be, right? Because rare diseases are not just in this country. What I also like, I have to see a few observations I made when I see the numbers, right? I think Kitch had on his slide, 360 plus million people that are affected by rare diseases, right? If you just imagine 260 million people, that is kind of the US, right? People that live in the US almost. It's a little less than the people living in Europe, where I'm from. It's just a huge number. And still, I agree with Noah, the word rare disease, rare is kind of, so how I understood what he said is this is why you called it uncommon, right? So let's continue to do what we do until these rare or better uncommon diseases show commonalities, so that more people show attention to what we're trying to accomplish here altogether, right? So, so just to introduce myself, I'm Helge Bastian. I'm the CEO of iXCells since a year. And also everybody knows Nian Wei, so I don't have to introduce Nian Wei. But I also want to, I think Steve didn't identify who he is. He's, did you? Huh? What's that? So Steve is also an iXCell, of course, and he is our Chief Commercial Officer and helping to do the external communication and in improving this for the company, also with us for some time. And we have some people have an identifier, which is the little pin we have on our reverse. So if you run into those people, you can be sure they're with us, and very precious little stickers, because we couldn't find more than the ones we have that you see today. Right, so that was a long introduction to, the next session. So the third session, as I said, it's not just halftime, but almost. And the next session brings us from what we heard earlier, the real life stories, right? Which is, I think we did it last year too, starting with the real life stories. So that people really get grounded a little bit, right? In the journeys of the families, and then rare disease foundations, I think is always a good start to really ground us and say, well, this is why we're here, right? This is why even this company exists. Modeling rare disease with patient-derived cells is what we heard in the last session. And now we're moving on to talking about building bridges to collaborations with industry and otherwise to make really progress in this area. So as the first speaker in this area, I think, do I have to push this forward? We're welcoming Kent Rogers. And let me just introduce Kent a little bit. So Kent is currently the CEO of Everyone Medicines. And, but this is, I think he will explain to us what it really is. So I just don't want to get even on some eyes here to misinterpret this. But at the same time, you're also partnered at Arch Venture Partners, right? But beside that, there is a long career already, 30 years in the healthcare industry. And I just call out a few: EQ Therapeutics, Accorda Therapeutics, Merck Sharing, right? So a number of really, heavyweights in the industry. But also, as I realized, you have been on the other side when you worked for one of, I guess, what people say the payer side of the industry. So at your time at Optum, right? So really, I mean, a huge experience. And we are looking forward to hear more about this with regard to scalable and sustainable solutions for individualized ASO therapies. So we heard about ASO earlier. As you know, iXCells provides ASO services too. So let's listen to your wisdom. Very happy to have you here, and thank you very much. Do you need this?
So thank you for having me. As mentioned, my name is Kent Rogers. I'm the CEO of a company called Everyone Medicines. And if you haven't heard of Everyone Medicines, I will provide just a little bit of feedback and background on our start. You will see the founder of our company, actually the two founders of our company, and their picture are on the wall right here, just around the corner. So there is an established relationship with iXCells already. I know I don't need to tell this audience the statistics that you see up here, but I present this deck quite a bit. This is actually part of my pitch deck right now. We're in a Series A raise, which I'll explain that as well. But what's important to understand about what we're doing is that we're committed to the smallest space within already a very small space within rare disease. And I think you're right. Uncommon is a better word. I think we all know rare disease is anything but rare. When you consider the totality of numbers, it's just the occurrence is so infrequent. So we are focused in a space that was really inspired by the fact, and this is something that I've now seen coming into this part of our ecosystem with healthcare. I've been in this industry 30 years, as it was mentioned, and I have never seen anything that has been so unfortunately organized in its disorganization, if that makes sense. Parents that are raising money for their children all over the country, and this is being repeated over and over again, companies starting foundations, and this is an audience that knows this all too well, and that's not a sustainable situation. As mentioned, our company was inspired by the journey that Mila took with her mom, Julia Vitarello. She was the first patient that ever had an ASO designed for her and her unique mutation. They did this within about a year. Now, that doesn't take into account the years-long odyssey that she had to go through to find out what her daughter had. And then when she did, that there is no company researching and or developing these products. And so Tim Yu at Boston Children's actually determined that ASOs was going to be a great modality for her because of her kind of mutation. The treatment actually did help her, but she was 6 when she was starting to become symptomatic, 7 when she was diagnosed. So you can imagine a little body like that had already ravaged her. And so she unfortunately died at the age of 10. But the inspiration that came from that experience motivated Julia to start a company called Everyone Medicines to give everyone, or at least every child, the same chance at life that her daughter Mila had. And so that's what we're going to do. We are going to design, manufacture, and deliver individualized medicines. Our current modality is ASOs. We will expand into other modalities in the future. But I want to underscore this, that while there are a lot of approaches that are going about this, one of the reasons that they asked me to come in is that I don't even know how many drugs I've launched in my career, 3 dozen. And I've been on the buy side as a payer. I've been on the sell side with both large and small pharmaceutical companies. So I understand how the supply chain works. And it's not a fun supply chain, and it's an expensive supply chain. And it's one that doesn't always make a lot of sense, as you guys all know. But I do know how to navigate it. And so I intend to navigate it with these kinds of medications. The good thing, too, is that when I came in, of the companies that are in this space, I'm already got wind at my back. I've got a very experienced team in the rare space, but we also have top-notch investors. If you're familiar at all with the VC world, which I have been in the VC world for the last 2 years, Khosla, GV, and Baker Brothers are top tier. And so they're actually supporting the company and have been for the last 2 or 3 years. They are committed to the current raise, which is good. But we also have both Tim Yu and Julia involved in our company. Julia is a co-founder. She sits on our board, and Tim is a co-founder and is one of our key strategic advisors. If you're not familiar with ASOs, I'm not going to drain the science because I think a lot of folks are, but they're rather elegant in their simplicity. It's a single strand RNA that approaches basically any of the challenging mutations that are giving either proteins the wrong direction, not enough direction, or just direction in general that they shouldn't be. And so we're going to approach this with a couple of different modalities. The RNA is age dependent, which is basically our way of being able to cleave the RNA when there is an opportunity for us to actually knock down what's happening from a protein perspective that's a wrong signal, or splice switching, which is what Millicin, they named it after her, was. That really is where we actually design it to enable the expression of protein to be appropriate if you're lacking that expression. This as a design is not uncommon. Spinraza is a splice switching ASO, as you guys are probably aware. And then obviously in the Duchenne's area, splice switching has been a very popular approach. Now, when I first came in, we actually were, sometimes things just happen serendipitously and you just go with it, right? It's just luck. We had 2 patients that had been identified with the same Niemann-Pick C1 mutation. And they had been struggling to try to find treatment, or anybody committed to Europe as an area for treating patients. It just so happened as I was coming in that they were asking if we could help them. And we had already started down the path of developing an ASO to treat that same mutation because Tim Yu had 2 kids in Boston that had the exact same mutation. So we were already creating it to see if we could do it. And as we were completing that, we made an agreement with Gosh that if you want to do this with us, then you'll be our first treatment center and you'll be our first kids. So that's when I had the fun to be able to go to my investors and say, well, hey, you've been talking about doing this for a while. We're actually going to do it. So we need to go out and raise money, and we're going to build a clinical trial. I'm not going to drain this because it's actually pretty small anyway, but this just gives you a general idea that the way we're going about developing the ASO is over-indexing to try to determine safety as well as, if we can, efficacy. Is there a biomarker against cholesterol and Niemann-Pick? That's one. But the reason that we're doing this is that when you consider a regulator and the way a regulator thinks, which is not the typical way to think, and they certainly don't have empathy or sympathy, I should say, for rare disease, we need to be as thorough as possible. So we're kind of walking through the front door with the requirements, knowing that we're going to have side door arguments to why they need to be able to approve products that we will not be able to study and that will never have been in a human before we're asking for them to approve it. So how do we go about that? So the idea here is that in the US, and this does not have to do only with the Trump administration and the fire sale that is going on at the FDA and HHS right now, which arguably that's going to be distracting anyway. But the FDA is a little slower than other countries in terms of embracing rare disease. Now, while the Rare Disease Innovation Hub was formed, and that brings two sides of the FDA together to try to address this, the science is way ahead of the regulators. And I know I don't need to tell this room that, but the issue within the US is they're not only distracted, but most of the guidance that they're giving is for research. It's not for treatment. And while the ASO guidance they gave for a 3-month GLP tox, that is a huge savings in the drug development phase. That's for research. It's not for treatment. And so that guidance wasn't really useful beyond that. They've also given guidance for platform designation. Seems or stands to reason that an antisense oligonucleotide, it's basically 16 to 21 polymers. We're not doing a lot of difference in terms of the backbone. That is right for a platform designation, but they gave guidance that was vague. And their response to our initial conversations is, well, you've got to bring us data. Agreed. So where do we get data the quickest and the easiest? You do that in Europe. You can get a clinical trial authorization in Europe up and running. I would say breakneck pace compared to the US. It's still a hurdle, but that's why we're approaching it. And it just so happens that these two children that were being treated by the Great Ormond Street Hospital, that's one of the premier neurological centers in the world. Things were just kind of coming together almost too well for us to say, we have to move, and we have to move as quickly as possible. So we started to put together a master protocol, and this is a variation on a theme you might have seen in oncology, where they'll have one drug to treat multiple indications. And the idea is that they're not spending the next 5 or 10 years building out the medication. They're actually convincing the regulator that the chemistry is what you focused on. It's less about the condition, it's about the chemistry. And if we can show safety with the chemistry, if it's a severe and life-threatening illness, as you might see in so many different cancers, does it matter what the efficacy parameter is to the extent that you need to find a primary and secondary endpoint? No. It's that it's safe and it actually can show activity post-approval. So we're doing that, but we're flipping it on its head. And so instead of one drug, well, 8, 9, 10 ASOs. We don't know how many kids that's going to treat. And I can't tell you how many times investors are like, well, how many patients are going to be in your trial? I don't know. Well, what do you mean you don't know? Well, each mutation is treating one child, but for instance, with the first one, they're treating 2 kids. Well, how many kids have this same mutation? I don't know. Well, how many in that country? I don't know. How many in the world? I don't know. We're going to find that out. But we do know that the more people do whole genome sequencing, the more we're going to find of these mutations that may be more common. The real trick is that if you have more than 5 or 10 or 15, like ataxia in Turkey, we're investigating that with Tim Yu, it's about 60 patients. I'm sorry to say that is too many. That is too many because I have to design a traditional clinical trial and go through the process to get that approved. That's not breaking the door open in the corner to show the regulators, focus on the chemistry, not on the indication, and not on the primary and secondary endpoints. So that's one of the reasons why we are sticking to a very, very small population. So the other idea is that because these are such small populations, they're not all going to occur in one country. So we have started the process of working with, GOSH is the acronym for Great Ormond Street Hospital, we've been working with Paul Gisson and Gosh to find patients that have mutations that are amenable. We've already found 2 kids in Italy, same mutation, and there are several in Germany, and we've actually now visited the Middle East, and we were at Arab Health a couple of weeks ago. They are very proactive in this space, by the way, and have done genome sequencing on almost 1 million people in the Middle East. They didn't necessarily do it for medical reasons, but it doesn't matter why they did it. They have that information now, and so being able to run our mutations against their database is part of what the future part of our business model would be. If we have drugs in a library that can treat a mutation, and you find a child that has that mutation, then they need to be in touch with us as soon as possible. So the clinical trial will be across multiple countries. It'll be across multiple mutations. And so far, the regulatory feedback has actually been positive. Now, in the context of a regulator, the reason I say positive is if they don't shut you down, then you're saying I got a chance. So then if they'll say, well, what about the idea that we cannot, and frankly, probably should not have to do a GLP tox for every single ASO that we bring to you, that's a pretty crazy strategy, right? Well, that's the reason that we're going to have 8 of them. Repeatability over and over and over and over again, and talking to them about the second one, the third one, the fourth one, the fifth one. By the 5th, the 6th one, we've been in a very iterative process with these regulators. We're showing them every one of the assays we're doing, which is pretty much the same overlap with all of them. There may or may not be a biomarker, but we'll be able to explain why there is or isn't. And we will go to the greatest lengths, whether it's wearables, hopefully not burdensome surveys that parents have to complete, but any kind of baseline and post to show that there is evidence of effect. I'm not saying efficacy, I'm saying effect. Because I don't know that you could ever state that the efficacy that a parent sees is the efficacy that a regulator cares about, if that makes sense. So with this, our trial design is to basically start as we will have our first 2 patients in trial in the 4th quarter of this year. We are already starting the design of 4 additional ASOs, and then we would hope to get up to 8. You know, we may have more, we may have a little bit less, but that's the target right now. And then as we progress over the next year to 2 years, we'll get CTA approval in multiple countries. This gives us a wealth of data to negotiate a corridor with regulators for a new pathway. Now, is this a guaranteed? It's not, no. When I talk to investors about what we're doing, this gives us the best shots on goal to create a brand new pathway with regulators. But by 2027, 2028, we should have enough to go for approval through EMA as well as other countries in Europe. This strategy is really basically to convince Europe so that we can then, sorry, I'm in the wrong spot, aren't I? If we convince Europe, then that means that we convince the US. But this is the vision of where we want to be. Very much like Mila, when her symptoms showed up, they were because her mom noticed, but the symptoms had been there since birth. Newborn screening isn't here today, certainly not in this country. At some point it will. And someone asked me at one point, well, what happens when you get a newborn screen that has an NPC1 mutation or a Batten's disease mutation? I mean, are we going to treat our child at that point? That's a good problem to have at this point, because we don't even know what newborn screening is going to look like in the future, or much less how it's going to be reimbursed, but other countries are doing it. So this is what would be realistic today, as the symptoms would begin, and that means the clock starts. The best case is that a neurologist would do a knee-jerk reaction. If you've got rare, random neurological symptoms in an otherwise healthy 5-year-old, run a whole genome sequence. Just run it and see what you come up with. And if you can get any kind of cellular data from the patient, swab, you know, I don't think urine and blood were there yet, but skin samples, then you can characterize that mutation and know what modality will work. In our case, an ASO, we could sequence an ASO within hours. And if it works, or we know that it's amenable, we can design it within 6 to 9 months. That means that a child with a previously unknown mutation, and I don't say disease state, right? Because there may not be a name for the disease, but an unknown mutation, we should be able to design, develop, sequence, manufacture, and deliver within 9 months. And that just means we need a truncated regulatory review in order to get it to the patient as quickly as possible. So our business model is all inclusive: diagnostic networks, treatment centers, obviously that we're on the drug and the manufacturing side of it, and then working with regulators and payers constantly throughout the process. you guys can read this, and I'm sure it's probably something that you already know anyway, but as I'd mentioned before, the science is way ahead of the regulators, and we know that. They know it too. And I think they're starting to respond and react, but our approach, and we hope others will take the same kind of approach, walk in their shoes with them, give them a reason to support you. And instead of theory, we have to bring them clinical evidence to have the conversation. And so while this is a very costly approach, and that's what the Series A is going to pay for, we feel like that this is the best possible way to find a brand new pathway that others can follow. CRISPR-Cas9, siRNA, I mean, there's a lot of great modalities out there, but Everyone Medicines, I think, is going to be at the bleeding edge, so to speak, of making sure that new pathway exists. If not ex-US, we can certainly try to bring it back to US if they agree. That's it.
Thank you. Thank you very much. Okay, we have a little bit of change in schedule because the next speaker on the agenda is still on the road, unless I see him here. No, I don't think so. So therefore, we move on with the next. You see it here, right? Konstantina is the next speaker here, and you're here. I know, hiding. And let me just introduce you with a little bit more really good kind of background here, right? So today you are, as it says here, at NLOREM, and you are the Senior Director of ASO Discovery and Research at the foundation. And of course, as it shows on your CV, you have been working on RNA research for some time, right? Before that, you led the RNA and technology team at Amina Therapeutics. That was also about double-stranded RNA and so forth. Before that, you also did work on rare diseases, so it's not a new area for you. But also before you went to these positions, you had a significant career in academia. And when I was the people that prepared this for me, when I was reading through this, it's quite something here, right? So we're talking about the PhD came from the University of Oxford, but then there were some prestigious awards. One is called the Sir Henry Wellcome Reward for Postdoctoral Fellowship. And this was done in Edinburgh and in my home country in Berlin, in Germany, in Berlin at Max Delbrück Center. So quite amazing. But if this would not be enough, you also got a major award, which I think is called the Women Science Award, which was, I think, probably sponsored by L'Oreal and UNESCO for this. So, and I'm sure there's more to find about you. So thank you very much joining us for this meeting here. Really welcome this. We heard earlier today that there were a few foundations that mentioned, and NLOREM, we are known anyway, the company, right? So, but we had this in Piotr's talk, and I heard it from Michelle earlier. So there's already collaborations going on with people that are here at the meeting too. Today we hear about what you guys do when, again, about personalized ASOs in, you call it nano-rare patients, right? So we have rare, nano-rare, uncommon, and I still pledge that at some point we all call this commonality somewhere, and we're talking the same thing. Welcome, and the floor is yours.
Thank you very much. An amazing introduction. Thank you. I'm extremely happy to be here, and yeah, what a wonderful event. And for the videos, would I just pause? Okay, that's wonderful. All right. Great. Yeah. So feel free to call me Nadina, everybody. I have an extremely long name. I'm Greek originally. So I'm leading all the research activities at the NLORM Foundation. So essentially from ASO design, discovery, and also all our core mechanistic research approaches to really reach more patients with more efficacious ASOs and in a safer manner. First of all, I would just like to take a few moments to introduce NLORM and just tell you all what we're doing and what is really our mission. NLORM's mission is to really apply the efficiency, versatility, and also specificity of the antisense technology to charitably provide ASO medicines to treat patients with nano-rare diseases, and that means less than 30 patients worldwide for free for life. And essentially the reason that we can do that, and I'm very glad that I'm hearing ASOs here since this morning, is that really we leverage knowledge and expertise that has been around for about 35 years. And our founder and CEO is Stan Crook, who is the former CEO of Ionis. So we have really leveraged all this expertise from Stan Crook, his colleagues, and everybody else on the antisense space. That has been amazing. And at the same time, while being in the shoulders of these giants, we really want to build continuous research innovation. This is where breakthrough will happen, right? And our ultimate goal is one and only one. We really want to find the best ASO possible for each patient. And this is what my team and I are really working on. First, I would just like to take you through what is our NLORM pathway to treatment. So the first part is essentially, I believe this is a pointer, maybe. The first part is the research, and then we go into preclinical development, the regulatory stages, and also the clinical development. So essentially my team is initially involved in also what we call the feasibility review, where we have the application evaluation and feasibility. So the physician sends us an application on behalf of the patient. And what we essentially evaluate, and this is, we also do it initially internally, but also we have a committee of really experts that really provide either ethics or clinical development, antisense technology, RNA, all of that, to really evaluate whether the ASO technology is the optimal therapeutic strategy for this patient. And after we have this committee and have these discussions, then we really take the decision if that application will be accepted or declined. If the application then is accepted and really we think that NASO is a suitable therapeutic approach for this patient, then this is where really we get to work. And this is where the ASO discovery and optimization takes place. So the very first thing that we do is we take the whole genome sequencing. This is one of the most important things for us, especially for allele selective programs, which I will go on into a minute. And also we obtain the patient cells. So everything that we do, and because the FDA, as I will say in a moment, we have this unique guidance for ultra-rare or nano-rare indications. We don't have specific steps, but we essentially have a very extensive in vitro discovery step, which we really use the patient cells for. And then we go into ASO design with our in-house AI-driven algorithm. We design the ASOs, we synthesize them, and then we really start the discovery. After that, we enter for about 20 top leads in vivo tox studies. And this is essentially the rat tolerability studies where we screen for tox. And then after that, this is essentially where really the preclinical development starts, where we have GLP toxicology, and this is where the GMP manufacturing and sterile fill and finish start. After that, and this is, we hold the IND, so we draft this massive document, which is called IND, and we essentially submit it to the FDA. And as you can see, actually, I didn't mention that in every one of these steps, you have around all the numbers of how many programs we have currently in each of these stages. And the very last thing is, of course, the clinical study and treatment. And at the moment, we have more than 30 patients that we are currently treating. And we have 16 patients that are actively on treatment, and we have more than 20 INDs submitted. And the important thing is that we have this in all 4 divisions of the FDA. So just all in all, and we started really, really small. So I'm really proud to stand in here and say that we have about 330 applications submitted today. We have more than 160 patient-directed drug discovery programs. Today, 25 INDs submitted, and the four divisions of the FDA are supportive right now. So just to give you a little bit of numbers and where we stand at the moment and how we're treating patients right this day, this is the NLORM regulatory clinical pipeline. So as I said, the number of INDs that we have submitted and all that is more than 20. The number of patients covered by the INDs is more than 30. And the reason for that is because some INDs cover more than one patient. The number of patients treated is more than 20 right now, and we're going to duplicate soon because actually my team is doing a great work in discovering a lot of ASO, so we have a lot of programs. The number of ASO doses that we have administered are more than 90, and the maximum duration of treatment is more than 2 years. And the most important thing of all is, of course, that we have pristine safety and tolerability profile. And this, of course, is one of them, is the most important thing. So really the current status of the clinical programs is that we have a very pristine safety and tolerability profile. 8 out of 9 evaluable patients show evidence of clinically important benefit. And I will show you in videos in a moment what that means. Sometimes a video is more than 1,000 words, which means that we are already seeing really important improvements in multiple domains in CNS, continued benefit with continued treatment. And very importantly, this is something that it was quite surprising to us because we start very low and very modest, and then we dose escalate. We really see evidence of CNS benefit at very low doses. We also see, in terms of kidney function, because we have some kidney targets as well, target reduction and stabilization of renal function, recovery of functions lost, demonstrating really plasticity of the CNS. So for some indications that we actually thought, oh, this person might be too old to actually treat, we find out that that is really not the case. That we think that the CNS is much more plastic than what we initially thought. And in terms of ALS, because we have quite a lot of ALS patients, we're really seeing halting of ALS disease progression. The important thing is, as I said, we have programs in all the 4 routes of administration: CNS, liver, kidney, and eye, and we're seeing important benefits in all of them. Now, what brings us to pristine safety profiles? It's really a big balance between what happens in research, what happens in discovery, and what happens in clinical development. So you really want a rigorous discovery, and this is exactly what we do. We screen thousands and thousands of ASOs to really find the one that is really the most optimal and best. So we really want to be very rigorous in that and really screen a lot in a very high throughput, high quality manner. But at the same time, on the other end, we want to be very conservative in our dosing and monitoring in terms of clinical. So what allows us to do that is a very deep and broad relevant experience in both ASO technology, drug discovery and development, clinical development and management of patients in clinical trials, and also our regulatory processes. We have an excellent relationship with the FDA. Our systems and processes are actually can be really scalable almost indefinitely. So we have really industrialized system and processes. We have multiple external committees for the preclinical data package review and clinical safety. As I was saying, this even starts from when an application comes to NLORM. It's not that we're just deciding internally. We have a lot of external people where we do a peer review. We really have a very strong commitment to caution. And also our research physicians are really outstanding. Some of them here at Rare Disease and all over the US. We have a very close collaboration with them. And now I would just like to show you, this is one of our patients. This is a KIF1A. This is an allele selective ASO, which I will talk about in a moment. And I would just like to, this girl had a really severe disease with multiple seizures, neuropathic pain, couldn't walk, nonverbal. So I would just like to show you.
I'm ready, sis. Oh, wow. That was... Yeah, and then what?
And then when...
Oh, See?
Look at that.
Get up. You know.
Oh.
You.
That was amazing.
I like that step though, but again, boom, yeah, ooh.
Dad, come on, I'll teach you how
Okay.
to stand
You
up.
teach me how to stand.
Throw an alley-oop. That's an alley-oop. Easy peasy, lemon squeezy. Oh.
Yeah, it's pretty incredible. So this is just boring words to just summarize what you just saw. This girl was, yeah, she was in a very, very bad state. This is a KIF1A disorder. As I said, very, very frequent and very severe seizures. We're seeing a significant reduction in seizure frequency and severity, significant reduction in neuropathic pain. She just wouldn't sleep because she would just cry because of pain the whole day. She's regaining, as you saw, the ability to stand up and work social distances independently. She couldn't do that before. She has a significant reduction in tremors, significantly improvement in communication with family. This is something that has been communicated to us by from her caregivers, and general significant improvement in the quality of her life. She even attended a sleepaway camp for the first time in her life. And of course, we have actually just published that in 2 publications. The second one that I would just like to show you is an ion channel mutation. This is SN2A. This is a boy that has actually, the ASO that we discovered and developed was actually administered here in Rare Disease. This is a boy that has never walked in his life and had very, very severe GI dysfunction as well. This is a very severe disease, extreme seizures. This is an ion channel, so you can imagine this is very, very severe. This is an allele selective ASO as well. And here's he's walking independently for the first time. Yeah, so this again summarizes what I was just saying. Reduction in seizures, he regains the ability to walk independently, decreased ataxia and choreoathetosis, and improvement in autonomic dysfunction, normalization of bowel movements, which is amazing for him and also for his caregivers as well. So all in all, I just talked to you before about what is happening with the clinical development. I would just like to show you as a whole what we do at Enlorem. And this is really our process. This is our ASO discovery and development process. It's an individualized approach and infinitely scalable. We really use, as I was saying, the efficiency of ASO technology in parallel with the FDA guidance for individualized ASOs to really provide a rapid, effective, and affordable treatments to nanorare patients. Everything starts with the whole genome sequencing and the patient cells. This is really the most important thing. This is where the ASO discovery begins, concludes that we enter the GLP toxicology. In parallel, we have to accelerate timelines, manufacturing, and sterile fill and finish. In parallel, our regulatory people at Enlorem, they start drafting the INDs and submit. We have the IND submission, IRB approval, which comes from where the physician is. And then, of course, we have the patient treatment. For the last part of the talk, what I would like to do is to take you through what my team and I are doing, and how do we end up with that optimal ASO? What does this look like? So this is essentially our process. But before that, I would just like to take a step back. And as I was saying, I'm extremely happy today that I heard ASOs multiple times. And I think the reason for that, the reason I think that this modality is really an ideal one for nanorare patients, and also importantly, why we do what we do at Enlorem and why we do it so well, is essentially because of this particular modality. For example, if you were to take, the really big guys at drug discovery, which is essentially historically, right, the small molecules and the monoclonal antibodies, you would know that the rules of engagement are not as well understood. Whereas with ASOs, with RNA targeted, we know exactly what are the rules of engagement. We know that in this particular case, the receptor, if you like, is the RNA. So you know exactly what you're going to get. After you design an ASO, you know what it will do. It will target that mRNA, it will hybridize like a zipper, and then it will degrade or it will block something. So since then, the rules of engagement are very well understood and easy to use. It really means that we can also learn from previous drugs. Not everything is just like, you know, sometimes with small molecules, we say change the method, change the drug. In this particular case, we have a broad transferability. The cost of drug discovery here is modest compared to other routes of drug discovery. The rate can be months. It is very, very selective. As I said, this is an RNA. This is just based on base pairing and hybridization. Very versatile. A lot of different mechanisms here that we can do. We can alter the RNA in many different ways. And also the important thing, and this is what matters, right? When you want to do breakthroughs and when you want to do things faster and better and safer, is really to advance the technology. And with ASOs, there is an extensive rapid advancement. And this is what we strive to do. Very briefly, and I don't want to bore you too much with all the science stuff, but a lot of people talk about gapmer ASOs, right? This is the RNase H1 ASOs. This is literally what is the drug. This is literally the ASO. And a gapmer ASO, this is what is used when you want to reduce or knock down an mRNA target. And essentially, we have what we call the 5-10-5 modality. So this is where you have the 5 bases at the wings at the very beginning there with the 2 black, which is RNA bases that are essentially or modified with methoxyethyl. And then you have in the middle there that orange part, which is the DNA. This is essentially where all the magic happens, because this is exactly what provides the substrate for a beautiful enzyme, very, very selective, which is called RNase H1, where when this orange binds your mRNA target, this is where the cleavage happens. And this is how you reduce the target. Now, you can do that in what we call a non-allele selective way. So you can reduce both your wild type, the good copy, but also the bad copy, the mutant, and that is what is not an allele selective. But the beauty of this enzyme also allows you to do it in an allele selective way, which basically means that you can leave your wild type untouched, but you can only target the mutant. And I will show you in a minute how we do that. Now, this is what we use when we want to degrade, when we want to reduce the target. We also have the other class of ASOs, which is called the steric blocking ASOs. This is an 18-mer, and as you can see, there is no orange here, everything is black. And the reason for that is because you essentially have only RNA bases, that they're all modified, the 2' methoxyethyl. And here the backbone is phosphorothioate. And something that I forgot to mention before is actually you might ask, why do you need all of these weird chemistries? Why do you need to chemically modify the backbone or the RNA bases or whatnot? The reason for that is because this allows you to have a longer duration of action of the drug, less immunogenicity, and also higher binding affinity with the RNA. This is the reasons why we chemically modify ASOs. So you have different classes. So this would actually, you would sit back. So for example, when we receive an application for a patient, we sit back, we see the disease, and we're thinking, what is the best strategy for that patient? Do we want to reduce something? Do we want to reduce both the wild type and the mutant? Do we only want to reduce the mutant? Or do we want to block something? Do we want to use a different isoform? Do we want to alter splicing? And the reason that we can do that is because really we have, the ASOs are very versatile. So as I said, we can do an RNA degradation. These are the first 2 in here, either with RNase H1, ASOs, or you can have the siRNAs with AGO2, albeit a little bit more of a promiscuous molecule, not as specific. But also you can have mRNA modulation. So the splicing ASOs that I just showed you before in here can be used to either alter splicing or affect mRNA stability or switch between different isoforms. So this brings me to the allele selective ASO approach. And this is both of the ASOs that I showed you in videos before, they were using these kind of ASOs, and we use them a lot in a lot of diseases for the CNS. because essentially you don't want to completely reduce the protein, but you really want to keep one good copy, but get rid of the bad one. So the way you do that, and this really leverages the specificity of the RNase H1 enzyme, is you really try to guide the RNase H1 to target only the pathogenic mRNA with leaving intact the wild type. How do you do that? We have these little signs in a gene that they're called SNPs. You might have heard of them, single nucleotide polymorphisms. And this is why the whole genome sequencing is so important to us, because we take the entire gene and we really see where these SNPs are. SNPs are basically positions where your wild type and your pathogenic differ. And this is what we need, because we need to find these little differences so that we guide the ASOs in these positions. So the ASO can specifically, the RNase H1 can specifically degrade the pathogenic with leaving the wild type untouched. Now, we've done that. So what I've shown you is we've decided on the strategy. What do we want to do? Do we want to reduce? Yes, no. Do we want to block something? So after we have done that, then we are ready to essentially design the ASOs. And in order to design, we have our own in-house ASO design algorithm, which essentially really is the one that makes our ASO discovery highly efficient. And AI is a buzzword right now. It's amazing. The thing that is important with AI is what kind of data you bring in. And the kind of data that we bring in our design software is essentially, we're taking all of these millions of ASOs that both us and also ASOs at Ionis we've been dealing with, all relevant cell and animal models, hundreds of thousands of patients, continuous learnings from the whole genome sequencing, because it's essentially what we are striving to do, to design better and better allele selective ASOs, and designing ASOs to reach more patients. And also at the same time, of course, there's so much information out there that is amazing with the technology, and there are a lot of multi-omics data sets that we're essentially putting in our algorithm to really make it better and better. And I'll just finish the talk with just showing you how does it really look like? You know, when you screen, how does it look like? What kind of graphs are we seeing? So our process is essentially the very first part, and this is where it takes some time, is the primary ASO screen, because we screen about 500 to 1,000 ASOs. And for an allele selective approach, as you're going to see here, is the blue is the wild type allele where you want it almost untouched, and the red is the mutant allele where you want it reduced. So you can already see that there are some patches there. Oops. There are some patches there that we have some really, really good ASOs. And we have very important and very strict minimum criteria. So things that do not pass our criteria, for example, of more than 80% of target reduction, it won't pass to the next step. The next step is really the epitome, as I would call it, of drug discovery for ASOs, which is essentially the dose response evaluation, where we do a 5.4-fold dilution to really scrutinize the efficacy, potency, and also allele selectivity of the ASO. And here what we take in this step is we take about a 50 to 75, so the best of the previous step, and we essentially scrutinize what is the IC50. This is what defines the potency. What is the IC50 of the mutant? What is the IC50 of the wild type? What is the reduction of the mutant? Do we have the wild type untouched? What is the selectivity that we see? And here you can see here, they are in the red box, some ASOs that really stand out in this particular case. And finally, as we move down through the discovery path, we also do an in vitro, of course, of target analysis. This is initially done in silico, but then, of course, we also do that in wet lab. And we do that as many as necessary to make sure that we eliminate any off targets. And finally, of course, we're going to do the in vivo talks later on, but you really want to check, you wouldn't put ASOs that you know are not safe into animals. So there is an assay that has been established for years now, which is called the BJAB assay, which is essentially is a very good indicator, is done in B cells, which is a good indicator of innate immune activation. So the ones that really activate immunogenicity are, of course, thrown away. So you have essentially surrogates for that in a qPCR. And finally, you can see these really fancy robots that we have in the lab. And this is what allow us to really handle about 90 patient programs at the moment, even though we don't have a very big team. So this is all liquid handlers for high throughput screening. So as I come to the end of my talk, these are the conclusions. So we have a very highly experienced and functioning team in place to discover and develop optimal ASOs. We have the capabilities to do this rapidly in a cost-effective and high throughput manner. Our clinical experience is outstanding at the moment. The ASOs given via multiple routes of administration are safe. Our clinical benefits are seen across all organs, and very importantly, at low doses and really early in treatment in CNS. And we're really treating these patients today, and we are extremely proud and happy about it. These are all the publications to date. We try to publish as much as we can to have all of this out there. And really, thank you.
Okay. What can I say? This is Constant. Where is she? There she is. So Konstantina, this is just amazing, right? I mean, what else can I say? You called it outstanding. It's amazing on all levels, I think. And to see kids walk that have never walked is so funny, right? So we again are reminded why we're doing what we're doing, right? So you said you also like to talk about ASOS, generally speaking. You like the other people talk about ASOS too. And I think we can promise that we will probably hear more about at least RNA modalities in general. And he arrived safely. Hi, Jeff. So are you also blown away? Still to breathe, right? Because now it's a little bit of a competition here, it seems like, right? So many people know Jeff Milton, right? Jeff is not just the founder of La Jolla Labs, but also run it as a CEO. And if I saw the news correctly, now you're switching with your co-founder and focus on technology, and she does the CEO and has to deal with all the HR issues and so on. So I think it's a relief, is it? So yeah, so for those that don't know La Jolla Labs, and he will talk more about it, a company that also thinks about RNA therapeutics and modalities. So, but it's not the first time he does it, right? So you have been working with, as a head of data science with Arcturus Therapeutics. You have been also working, of course, as a director of functional genomics and drug discovery at Ionis, right? And then before that, you also had time with Genentech. And so we know you, right? We work with you. Neon Way has worked with you before, and we're looking forward. What are the news? And yes, so Jeff, actually, I just also have this on my list here. You are a graduate from Mellon College of Science at Carnegie Mellon University. Yes, okay. So welcome again. Glad you made it safely, and here we are.
Great. Thanks. Yeah, so it's going to be a continued talk on ASOs, but this is actually a talk I've wanted to give for a long time, and it's a work in progress, because what I wanted to actually talk about was actually more about the modalities that are used. One of the things that I experience a lot at La Jolla Labs. So a little bit of history. We started La Jolla Labs. My co-founder and I, Tamar Grossman, started La Jolla Labs about 3 years ago. We're both from Ionis Pharmaceuticals. We were at Ionis when Spinraza was made during Milasen. So there's a lot of overlap. In fact, you're going to see some slides that I have that Stan Crook gave me that were just shown in the preview. But that's fine. That's good. But in any case, there's a lot of overlap. And so I'm going to come at this from a point of view of a company. How do you run a business doing rare disease? It's very difficult. And so every time when I get into this thinking about drug discovery, I think about it from the point of view of how can we make a business out of it? That breeds innovation that hopefully kind of perpetuates and makes it sustainable. So that's kind of the angle. This particular talk is really because I've always wanted to talk about the different modalities of drug discovery and the context of rare disease, because rare disease, you have to think of it from the point of view of rare disease and not be sort of stuck with a technology trying to fit it to the disease. So this is part one, because I'm going to talk about it in the context of chronic dosing versus gene therapy. So I'm not going to talk about gene therapy, the one and done type scenario, because that would be part two. But you've seen this before, rare diseases, it's a long tail. And what that means is that from the population, from a patient perspective, there are a lot of different individual patients that you have to focus. So you have to be more individualized. And that creates a very different landscape. The conventional approach to therapeutics, development, and even clinical trials is totally different. You have to interact with patients. You have to talk to families and kids, and there's no Hippocratic oath when you're in business. So you have to ride this very fine line between what's right and what's wrong, because you have to answer to the patients. Ultimately, that's what you have to do. But you also have to make a business. So that's the struggle, is how do you really do that? And when you think about the fact that 90% of drugs fail in sort of the common approaches to clinical trials, 90% fail, that doesn't work for the business that we're in. So there's a lot that has to be taken into consideration. So I get parents talk to me all the time, and they say, we want to do an ASO. And because it's a rare disease, and they've heard about it, and they think about the technology, and I say, I don't know if that's what you want to do. It's probably about $700,000 from start to finish, if you're lucky. And so what I say is, try to look at repurposing. So is there a way to repurpose a drug that's already out there? So there are companies that do this. One of them is, I'm blanking on the name, but it's Unravel, if you're familiar with, anybody familiar with Unravel? So they'll for like $15,000, they'll do a screen for a particular disease, and then they'll find other particular indications that might be linked to it. They have a business model that somewhat works. I say, go to Unravel first. It's cheaper and probably faster. The tox profile, the preclinical work is super, super expensive. And a lot of people who do rare disease don't even think about that part of the work. They kind of do proof of concept, they get excited, and then they get into the preclinical side of things, and the money runs out. And so you end up with a lot of dead-end projects. So let's talk about small molecules. So small molecules, I started this industry when rational drug design was kind of the approach. It was kind of the, Gleevec was the idea of taking a protein from the PDB and finding the pocket that you want to get it into, and designing the small molecule into that little pocket, and rationally design the drug to fit into the target that you want and make it do what you want it to do. But ultimately, is it really rational? There's a lot of space you're playing around with there. Maybe AI can help, but as we're finding out, there isn't enough data to really figure out, to really kind of make that happen in a super efficient way. It's still 10 years to do a drug and about $1 billion. So that's kind of the small molecule approach. Can it be selective? Well, here's one that's actually pretty selective. So this is a drug by Merck, and it interfaces or interacts with this MET kinase inhibitor, so mesenchymal epithelial transition layer, and it's very, very selective. I think it's like 10,000-fold more selective than any other kinase. So it's very selective to that particular target. So that's still a very costly enterprise if you go down that path. So what does that look like? So for a small molecule world, this is kind of what a typical small molecule approach would look like. You'd have a big or a large library of 1 million different compounds. In this particular case, the screening library is what's called a fragment-based drug discovery. There are other types of small molecule libraries, diversity libraries, things like that, that you can do these millions of compounds of screens. And then you find 500 hits for the particular target you're looking at. First of all, the target has to be extremely well characterized. And then maybe you can do crystallography to figure out how that fits in there. And then you can do some rational drug design, some med chem to figure out, I've got to make it look more like a drug and have the properties of a drug so that it sits around and does the actual, what it's supposed to do without being cleared too quickly, that type of stuff. But again, you're still looking at, in this particular case, I think this was about, yeah, so this is under 10 years, so that's quick. So this is kind of giving you an idea of the benchmark that we're dealing with a small molecule. And then this one is actually, this will be the last one. So this is from Vertex. And this is the ETI drug for cystic fibrosis, or I'm sorry, yeah, CFTR. Yeah, so cystic fibrosis. And what's cool about this is actually, this one is actually, it binds to a lot of different, it's a folder enhancer and potentiator. So you can kind of look at the phenotype of this and screen for that. So that's kind of cool. And this was actually pretty quick. I think it was less than 10 years, but they have these nice cryo-EM structures over here that show the structure. So that's nice. And oh yeah, so the one thing I want to say about this is that there are a lot of off-target effects with this. So there's liver tox, there's kids get cataracts with this. So that's the other problem with small molecules, is that you can get very promiscuous hits in a sense. So now we go to antibodies. So this is a separate modality. This is much bigger structure. You have the variable region, the antigen binding region, and then you have this very conserved region here. So this is the part that you're going to bind to the actual, whatever it is, the molecule you want to bind to. And then there are all these different modalities that you can chop up and use separately. So for example, there are some enzyme replacement therapies that use just this Fab fragment as a delivery vehicle. So they're very versatile, but very complex. And this is one of those structures that gets into sort of downstream costly effects with synthesis and characterizing structures. So in any case, this gets very complicated. I'm not going to go through all the details, but they're very versatile in the sense that they can be used as delivery vehicles. So in this particular case, you've got an antibody with a linker and then a payload. And so for cancer purposes, that's great because you can have the antibody bind to the tissue, the specific cancerous tissue, internalize, and then the payload is the toxic part for the tissue. And then you can have something like an antibody bound to an siRNA or an ASO. And so these are structures from Avidity. I think I took that from their website, but that shows how you can go even more selective. So you can be tissue selective and then target selective, RNA selective. So that's kind of gives you some of the idea that antibodies go a little bit further as far as selectivity than small molecules, but there is a very big downside to antibodies, and that is the cost of some of the synthesis and some of the downstream preclinical studies. So when you scale up these reactions, they can be very expensive. I mean, so I was at Genentech for many years, and I used to go into the scale-up facility and talk to some of the guys there, and they were saying, I'm about to run $1 million reaction, so don't talk to me. Because if they mess up, that's it. So in any case, there's a lot of complexity that goes into this. And when you're doing things like making or manufacturing this, you have to characterize what they look like. If it's an antibody drug conjugate, these things are actually in solution in ratios, and you have to kind of find ways to develop the assays so that you can actually characterize what those structures are. so you know what you're delivering. So that's an extremely expensive downstream enterprise, but it's very selective. And so, of course, you know, this is kind of a dump of all the different things that you need to do for taking a drug, antibody from beginning to end, from cells, from sort of research all the way through to the IND filing. But this is the type of stuff that gets very costly that people don't see. So part of the reason why I love RNA is because it's targeting the genetic cause of the disease, and it's very simple as far as the modality itself. So I'll talk for the rest of the talk. I'll talk about the RNA sort of landscape, and this kind of gives the history of it. So Ionis started in 1989. And so right about here, it was started with $7 million right here in Sorrento Valley by Stan Crook, the founder of Lorem. And they went through a lot of ups and downs. It was not an easy road by any means. But in any case, Stan persisted and really worked this out. And you can see that really by the number of publications. He's very he publishes quite a bit on this, and so it's you can really kind of follow the whole path that he did for this. But the first drug was actually Formotersen. It's off market now, but this was for it was for HIV patients who had a viral infection in their eye, and so that was the first drug. And I think that was a straight DNA. I don't think that was chemically modified. It might have been chemically modified, but very little. And in any case, and then we go, yeah. So anyway, I'm gonna skip through some of that rest of that stuff. I don't think they have Spinraza up there, but Spinraza was 2016. And that was the first blockbuster ASO drug. So that was, and that is a steric blocking drug. I'm not going to go through the mechanism of that, but that was it wasn't a gapmer. It was basically just a molecule well, I'm getting too deep into the science, but basically it's a steric blocking ASO. So it was fully modified ASO. And so these are the two RNA targeting drug modalities that are typically they're talked about. So this is the Ionis approach, and this would be the Alnylam approach. So the siRNA versus the ASO, single-stranded ASO. And so the mechanism of action is basically for the gapmer ASO is it binds to the target RNA. The RNase H enzyme recognizes this as basically not supposed to be there, and it cleaves it. And then you have the RNA degradation. And then the siRNA is a little bit different, but it's the same basic mechanism. And they can be viewed as catalytic in the sense that these molecules actually don't get cleaved right away, but they can go and bind to other RNA targets. So, and the big difference here is that this happens in the nucleus, whereas this is in the cytoplasm. So the siRNA doesn't actually happen in the nucleus. And so that's part of the reason why, so there's some advantages to the RNase H approach when it comes to cleavage, because you can cleave in the intronic space versus the siRNA has to be on the mRNA. And so the siRNA, and in this particular case, actually this is the steric blocking approach. So for the steric blocking approach, it's the same type of molecule, but it can actually get into the nucleus. So for steric blocking, for things like exon skipping, you might have seen with DMD, this all happens in the nucleus. And so you can see that it can bind to various locations in the intronic location and cause various splicing factors that would either enhance an exon or skip out the exon. And that's the mechanism of action of Spinraza. And so here's the stand slide. So there's basically a lot of different approaches to RNA, this particular modality. And this is really the reason why we really like it. So you can do the RNase-H approach, which is the gapmer approach, the siRNA targeting RNA, and then nonsense-mediated decay, which in a second I want to show you a little an experiment that we did that we really, one of the reasons we like the nonsense-mediated decay is because we can use a fully modified ASO. It's not a gapmer, has a longer half-life in the CNS, and it's much more durable compound. So we could potentially use it systemically or in muscle. And then there's no-go decay, which is basically pausing the ribosome so that the body says, okay, this is not working out, so we'll degrade this. And then translation inhibition and alternative polyadenylation. I'm not going to get into that, but what we have done at La Jolla Labs, and then there's microRNA strategies for increasing expression. This is kind of like this is essentially what we're going toward, is trying to build a palette of experience for being able to do any of this work. So we've done upregulation with microRNA inhibition and enhancing canonical splicing. And don't think I'll show that one, but we have not been able to reproduce this, although it has been published at Ionis. And then we have downregulation. We've done RNase-H. Turns out that's a much easier process than our favorite, which is inducing NMD, which I will show in a second. And then we have done a pretty good allele selective knockdown, which is the n-lorem approach. And then we have done exon skipping and cryptic exon modulation. Protein engineering, I don't think I'll show that, but what we're able to do is essentially move a splice acceptor site so that we add amino acids into the active site of an enzyme. And we think that would attenuate that enzyme and essentially knock it down. So that's the way we would do protein engineering. So this is an example of an NMD. One of the reasons we started La Jolla Labs was because of a disease called Lafora. I've talked about this before, but this is a glycogen storage disease. These are kids that are high functioning into their young adolescence and then start a cognitive decline and end up dying in a vegetative state. It's a terrible, terrible disease, very rare disease. So what we have, and this is our software, our La Jolla Lab software on the top here. This is an AI model that we have that shows us how to splice out an exon, essentially. So what you see with this dot right here is, this is an exon, this line. The bars going up are attributes, we call cis-regulatory attributes that are associated with the inclusion of that exon. The bars going down are for the exclusion of the exon. So we would put our ASOs, which are steric blocking ASOs, on these blue dots. And those are hitting the bars that are going up. So we want to cut the exon out. So we know where to place them. And so I think we had probably about 30 ASOs for this one because we knew where to put them. We cut out the exon. That caused a frame shift, and that induced nonsense-mediated decay. And so we did this in a dose-responsive way. And then we showed it with protein. And then we also did a phenotypic assay where we're actually knocking down glycogen the way we wanted to. So what we did was we created a molecule that was fully modified so that it wasn't consumed by nucleases. So we didn't want the gapmer approach because that has a shorter half-life in the CNS. So this, theoretically, the patients would be dosed once, twice a year. And it has that durability with this particular approach. So it's the same molecule as Spinraza, just a different sequence. The same chemistry as Spinraza, different sequence. And here's an example of what that looks like. So this was really the reason we decided to go this route, was because Spinraza has such a long duration of action. So we wanted that chemistry, just a different biology. So the last part of the modality talk is really where RNA is going, which I'm kind of excited about. I mean, there are these A-to-I-mers, which are the RNA editing. For the first time just recently, we went into the clinic with that. Wave went into the clinic for this particular target. We had, I think I've had probably over the course of the last year, 2 families come to me with single nucleotide mutations in acceptor sites of splicing acceptor sites, which could be done with, they could have fixed it with this type of approach. But it was so new that we're not prepared for it. We're not ready for it. But I think that's coming. So I think this is kind of an exciting thing to see. But the other thing is, does this, we're kind of waiting to see what Wave kind of reads out and how this goes. But the other thing is also, it still has the same delivery problems that every other RNA modality has. And we'll have to work through that. But in any case, they have this PRISM chemistry that they talk about that I guess makes it very durable. It is a larger molecule, so it has a sequence space that targets the actual RNA, but it also has a part of the molecule that recruits the enzymes that does the base switching from an A to an I or A to G. And so they actually show some pretty compelling data that shows that they're actually getting this in vivo. And then they have some pretty good clinical data coming out here pretty soon. And so I think there's a lot more that's going to be happening in the RNA space that I'm really excited about. And so this is what the RNA editing space looks like right now. So there's a lot of stuff in preclinical, but Wave now has that first clinical in phase 1 right now. And I think they did read out some positive data on that. But ultimately, when we're talking about rare disease and the approach, it comes down to a combination of how much money, how much time, efficacy, safety, money and time are basically the same thing. But you've got to build that therapeutic window in a short amount of time with a certain amount of money. If you can get a situation where you have multiple shots on goal, that's the best. When I see these families come to me and say, we want to do an ASO, I think, that's one shot on goal, and it's probably not going to work out. It's going to take a long time to get to that point. And they're putting their own money into it. I try to kind of manage that process. Because ultimately, when you think about just the manufacturing costs, anytime you go from, I like to look at this from the point of view of chemistry and biology. If your manufacturing process involves biology, it's going to be expensive, no matter what. That's just the way it is. And in chemistry, it just has a little bit more of you can characterize the compounds a little bit better and you can manage it a little bit better. But this is the landscape of what things look like. I did my best at kind of figuring out these numbers, but for the most part, if you can find a small molecule approach, that's the best. And then ASOs are next, and then goes from there. But this is part of the reason why you see, if you look at a lot of the companies that are really in distress, you'll see a lot of these are in the cell gene therapy area because that late stage price tag is astronomical. And with that, thank you very much.
Yes. Okay. Thank you, Jeff. I think it shows one more time how sophisticated these RNA modalities are, right? And why there's more than one company involved in this, because there's a lot to find out and to gain too, right? So we're coming to our next speaker, and is Yael on? Is she on yet? Are you Yael? Dr. Weiss, are you on right now?
On, and I'm having trouble sharing my slides. It says someone else is sharing.
Okay, you have we have a few, one more minute because I'm going to introduce you first, and it says on the slide anyway, coming soon, so we are good.
Okay. You set me up. Thank you.
Yes, so let me introduce our next speaker. So where do we start? Because there's a long career already, and maybe the latest, we start with the latest, and while Dr. Weiss is speaking to us. So Dr. Weiss founded Masi Therapeutics, a venture that is dedicated to delivering therapeutics to patients with underdiagnosed, and here's the new term, ultra rare genetic neurodevelopmental disorders. And I point this out because we had rare disease, we had uncommon diseases, we had nano rare, now we're talking about ultra rare. And I still hope in a few years we find, as I said many times now, there's commonalities between all the different diseases. Anyway, so we will also learn that later on what Masi Therapeutics, their approach, how to address all this. But before we come there, so I just want to mention also that Dr. Weiss has been having roles at Genzyme, Merck, Ultragenics. So this goes over 20 years back. So a long career here already. I also don't want to miss to say that she is a distinguished member of the NIH-driven Bespoke Gene Therapy Consortium, the ASGCT Translational Committee, N equals 1 collaborative, and holds the prestigious title of, and this is from 2022, Termere Fellow. The activity serves, she also serves as a board member for a number of organizations like ADNP and FOXG1 Foundation. The long career started with graduating from Hadassah, and I'm sorry for the pronunciation, medical school at the Hebrew University in Jerusalem, and she holds an MD and a PhD. And this will all lead us into her talk, revealing a little bit how Masi Therapeutics goes about developing gene-based therapeutics for genetic disorders. And I understand this will be, you will talk about your joint efforts that bring biotech, academia, and patient groups together. So looking forward to your talk, and the IT people here will make it happen that your slides will run properly. We can see you now, that's good news. And hopefully you can see us, and your slides should be up.
I'm just transitioning to slideshow.
Okay.
Great. So can you see the slideshow?
Yeah, it's not on presentation mode, but maybe there's, if you press it on presentation mode, we should be all good. So the right on the bottom. Here we.
No, just a sec.
Oh, this is you.
I'm there to do it. Just a second.
We have you on the screen right now, and your slides are behind you. Can we help?
It's not moving to presentation.
Don't worry.
Let me try again.
We hear you well, we see you well.
Okay, tell me what you see now.
Maybe it's loading. It was there before.
I know. Here we go.
Here we go. Yep.
Great. So, hello, everyone. I apologize for not being there in person. One of the reasons that I'm not there in person is that this morning I was part of a meeting with the FDA, with the American Society of Cell and Gene Therapy, which we actually thought would not happen, which would have allowed me to come to San Diego in person, but it did happen. And even though Kent says that the regulators are slow and not in tune with what we're trying to do for uncommon or rare diseases, we actually had a really good meeting with them on the topic of gene therapies, and a lot of openness to creative trial designs, to creative approaches to endpoints. And basically what the team that was on the phone told us was that we should talk to them as much as possible. So come to the FDA when you have ideas, ask for a type D meeting or a type B meeting or whatever meeting is appropriate for the question that you're planning to ask. And they will do as much as they can to accept your ideas and at least have a discussion on them. So that was really a good way to start the day. With all the news that we're hearing about what's happening in the FDA, it was actually a very positive meeting. So Jeff set me up for a lot of the slides that I have, so I will go through them quickly, some of them quickly, not to repeat what Jeff's already said, but I do want to talk a little bit about the Mozzie perspective and how we approach working on therapies for, I know I'm adopting your terminology, uncommon diseases, and I would say very uncommon diseases. And a little bit about the lessons learned from what we've been doing over the last few years since Mozzie was started. So the key messages that I'd like to convey is that the more genes are being associated with genetic disorders. Obviously, we started with 7,000 a few years ago, and now we're over 10,000. The more genes there are, the more patient-specific and gene-specific organizations there are. And drug development in general, as Jeff said, there's a lot of failure. It's not for the faint of heart. I have a lot more white hair on my head now than I did when I started out on this journey. But we really, in the case of rare genetic diseases, I would argue that we have an advantage if we fully understand the biology, but it's not sufficient or not mandatory. And you can choose a modality that allows you to go ahead with the therapy, even if you don't fully understand the biology of the disease. For example, gene therapy, you need to know where you need to replace the protein, but you don't necessarily need to know exactly what the protein does. Proof of concept is only the beginning, and I'll talk a little bit about what we consider as proof of concept, and then why is it so important to work with advocacy groups, and why is this triangle of industry, advocacy groups, and academia so important. So the topics that I'll cover are a little bit about advances in genetic diagnostics. What is a target product profile? So when we go to investors, when we go to regulators, when we go to payers, we need to show what the drug that we're planning to develop is going to look like. What is it going to do? How many times a day is it going to be administered? What the toxicology profile of it is. There's actually a whole list of things that we need to address before we even start thinking about going into clinical trials. So I'll talk a little bit about that and then about the teamwork that's required to get there. So I talked about the number of genes. We work in the neurology and the neurogenetic space. So a lot of the examples that I'll be giving you are from this space. And for example, when you look at the number of genes that were known for epilepsy 15 years ago, there were very few genes that were strictly associated with a specific epilepsy clinical diagnosis. As you fast forward to today, there are hundreds of genes that are associated with epilepsies and very specific genes. And these could be channels, they could be secreted proteins, there could be nuclear proteins, transcription factors. There are many different types of proteins that actually, when they're mutated, lead to seizure disorders. And the genetic testing is a key for many of these disorders that are just being associated with a gene, that it's key for commercial interests. So many companies are sitting back and saying, okay, we will wait till there are 10,000 patients and then we'll start developing a therapy. Other companies like Mazi, we say, okay, we're going to start to developing a therapy, and hopefully by the time we get to the clinic and by the time we get a drug approved, there will be enough patients to make it commercially viable. So this is Mazi in a nutshell. We're a small team, we're only 15 people, but all of us have a lot of expertise and experience with rare disease drug development. And the four bubbles, the four green bubbles exemplify our key partners in the journey to develop therapies. So we start with the advocacy groups. And this is where Mazi started, when I realized that a lot of advocacy groups, and some of them you heard from this morning, are developing therapies, spending money on getting proof of concept in cells or in an animal, and then kind of getting stuck because, as Jeff said, it's really expensive and it requires a lot of expertise. So we decided to start Mazi in order to be able to take programs that achieved proof of concept in an animal or in cell lines or in organoids and take these further into the clinic, take them into tox studies and into clinical studies. We work with the academics that actually conducted the work for the advocacy groups, because most advocacy groups look for the person that's an expert in their gene or in their protein and go to that person and ask them to start developing a therapy. So this is where the actual data will come from. And then we work with diagnostics companies because we're constantly looking for more patients for our disorders. And we work with CROs and CDMOs that execute a lot of the work that we're planning in order to get into the clinic. I'm giving SMA as an example. Jeff gave it too, but I'm trying to give it from a little bit of a different perspective. You heard about SMA from Jeff. It is a muscular atrophy, a spinal muscular atrophy. There's a gene mutation that's been known for a while. And until 10 years ago, there weren't any disease-modifying therapies for SMA. And currently, there's not only a small molecule, there's also an antisense oligonucleotide and a gene therapy. And these currently live together pretty well in harmony to give options to patients. And I think this should be the goal for every group and every gene, to be able to have a diverse set of tools to treat the disorder, because not everything, as Jeff said, will work for everyone. And kind of following up on what Jeff was showing, I have here the 3 different drugs that are available for SMA. The small molecule that was actually the last to be approved, the antisense that was approved in 2016, and the AAV gene therapy that was approved in 2019. So everything happened in the last 6 to 7 years. And you can see that there are big differences between them, and the mechanism complements each other, because the AAV gene therapy restores the protein by giving the missing gene. The ASO makes an alternative gene make protein, and the small molecule is a splicing modifier that also works on the genetic basis of the disorder. So all 3 of them work well. Price-wise, as Jeff indicated, the small molecule is likely the cheapest, and the AAV gene therapy is likely the most durable therapy of the 3. So continuing on this line of comparing the small molecules to gene therapies and to ASOs, I highlighted a few things. I won't go through the whole table here, but I highlighted a few things that I thought were important. Small molecules, again, and Jeff, I'll constantly quote you. I hope it's okay. As Jeff said, they're the lowest hanging fruit if you're repurposing. They're a little bit more complicated if you want a brand new chemical entity that will do something that other compounds have not done before. But in general, they're very rarely disease modifying. It's very hard to find something that targets exactly where the mutation is and changes the origin of the disorder. If you're screening for and trying to repurpose, you don't always know how the small molecule is working, depending on how you're testing to look for the molecule. And it has on and off targets effects. So there are a lot of side effects that are associated with small molecules. The gene therapy advantage is it's a one-time therapy, but still we cannot redose, and people that have pre-existing antibodies to the virus cannot be dosed at all. So there's a barrier to treat certain patients, and definitely a big barrier to treat again in case you need a booster of the therapy. And antisense you heard a lot about, so I'm just going to skip, and if you have any questions to Jeff or me at the end, then feel free to ask us. So we know the target. What else do we need to know? So here I tried to give a little bit of a flavor of your child got a diagnosis, you got a diagnosis, you Google your gene, you look it up, you want to see what the protein's doing, and what do you need to know in order to go to someone and ask them to develop a therapy, or what does that someone need to know? You need to know, for gene therapy, it's really important to know what the size of the mRNA is, because that's what you're going to be putting into the virus, and that's what will make the protein when you're trying to replace it in your body. And not every mRNA fits in a cassette that goes into a virus. For example, in Duchenne muscular dystrophy, the gene is huge. So the trick there was to make a mini gene. And this is what raises the question whether if you cut and truncate the protein and it's not the original protein, is it really acting the same as the original protein? And this will be the question for many of the genes that were truncated and put in as a mini gene. You need to know what the introns and exons are for the gene. You need to know how the expression is regulated, especially if you want to look at upregulation and you want to know how your gene is modulated. You need to look at microRNAs or long non-coding RNAs. Jeff mentioned NMD and exon skipping. So all of these things you probably can just feed into a computer, and that will tell you a lot of the regulatory elements that are associated with the gene that you want to restore its function. And you want to know homology across species, and we learned the hard way when you have a gene that's very homologous between mouse and human and monkey, and you express it in the mouse and you express it in the monkey, and you want to see if you restored expression. If the gene is very, very similar among species, it's very hard to tease out the new expression versus what the animal is expressing in the background. So a lot of times we actually recommend to groups to optimize the human sequence so it can be detectable over the animal background. What type, and you look at the mutated proteins, how is it mutated? How is it inherited? Is it a de novo mutation? Is it brand new in the family? No one knew before that anyone else suffers in the family from this mutation or carries the gene. That's important because if it is inherited, you want to go back and look at the mode of inheritance and find other patients that are potentially affected in the family. You want to know if it's a loss of function or a gain of function. That's really important when you're designing a gene therapy or an antisense oligonucleotide. You want to know is there a genotype-phenotype correlation? If it's just a missense mutation, does it behave differently from a nonsense mutation when there's no protein at all? Or are the mutations all the same and they give the same clinical phenotype? This is really important when you start thinking about enrolling in a clinical trial. What are you looking for? What patients will you enroll in the trial based on the mechanism of therapy that you have decided to pursue for this indication? And then the last one I mentioned already, do you really need to know the protein function? As I said, I would argue that you don't have to know exactly what it does, but it's nice to know it if obviously there is information about it. What is proof of concept? And this is something that we're asked a lot by patient advocacy groups who want to partner with industry. What does industry want to see? So obviously it's not a one size fits all. Ideally, you want to see in vitro data, and it could be from 1 dimension, 2 dimension, 2.5 dimensions, or 3 dimensions. And if there is a good mouse model, we'd like to see data from a mouse model. Many groups have several different mouse models. It has to be the one, obviously, that's the most suitable for the modality that you've chosen. Cells could be either patients or isogenic cells, and organoids are now becoming more and more common, and especially for certain disorders and certain tissues, the data from the organoids is becoming really a key for advancing things further into the clinic. And then the mouse model, a lot of times we have questions about, oh, we have a mouse model, but it doesn't behave like the humans. Is that okay? Well, obviously, ideally, we'd like to see some behaviors that are shared, but we want to know that the drug is working. So if your child has seizures, but the mouse does not seize, but the mouse has a different behavioral abnormality, if you can restore that, that's good enough. Or if you can show that the gene therapy or the ASO actually restore the mRNA and the protein, that's good enough. In the right place, that's good enough. So don't get stuck on the fact that the mouse doesn't behave like the humans. And there's always a question of when do you treat the mice? Everybody does P0 because it's really, P0 is immediately after the mice are born and the babies are tiny, tiny, and their skull is very, very soft. So it's easy to inject into the skull. And people tend to do that, but a P0 mouse does not represent a newborn or a patient post-birth, a human post-birth. So a lot of times people get start insisting on seeing data from a 3-week-old mouse or a 6-week-old mouse. Showing in P0 is a good start. If your drug does not work in P0, it's likely not going to work in a later age as well. And of course, we need biomarkers, and I encourage every group and every company that's working on disorders to start looking for biomarkers, because having an early indication that the drug is working is really helpful. So I wanted to give this example quickly of what we had when we in-licensed the program for a disorder called worry. It's a very, very rare disorder. I think there are 200 patients known in the world, plus minus. The kids, a lot of them die. So the number probably is around 200 around steady state. It's inherited, it's an autosomal recessive. And the key feature of this disorder is very, very severe seizures, up to hundreds of seizures a day. And the kids end up passing away because of complications of the seizures. So when the PI came to us with work that was funded by the advocacy group, what he had at the time, and it's all been published since then, was a good mouse model that replicated some of the human features, an AV9 gene therapy under the SYN promoter. So the protein is expressed in neurons, which is where we need it to be expressed. And he was able to show that the gene therapy reverses all the symptoms that are seen in the mouse. So early mortality, failure to thrive, hypoglycemia, seizures, other things that were observed in the mouse were reversed by the gene therapy. And we thought to ourselves, you know, there are only 200 patients in the world. That's true. However, this gene therapy looks very compelling and we have to get it to patients. The proof of concept package was very robust and we decided to bring the program into MODSI. Quickly, a few words about patient groups. You heard from some really great groups in the morning, and obviously great examples for what patient advocacy groups are doing these days and how advanced and how sophisticated the work is. On the left, you can see Cure Duchenne, the Cystic Fibrosis Foundation, and more recently FAST, the Angelman organization, that have been very successful by funding therapies. They're able to see a return on their investment and become very, very rich and very affluent and very influential advocacy groups. I just learned last week at a dinner that the Cystic Fibrosis Foundation has more than $3 billion under management, which is crazy, right? Everyone's dream to have so much money to be able to spend on your disorder. On the right-hand side is just a sample of the new CNS disorder groups that we're working with, and there are tens of them. And they're all hoping to be as successful as the ones on the left. So on the sorry if I confused my right and left, but on the left is FAST, Cystic Fibrosis and Cure Duchenne, and on the right is are the new groups, and they're just aspiring to be as successful, but they don't have the resources, and this is where MODSI comes in and hopes to support these groups and help them get into clinical studies and prove that the drugs that they're making are actually working and could be commercialized. So our mouse showed a really great slide on the tools that their foundation has. And I built this pyramid of all the information that advocacy groups can actually generate to, first of all, have a strong basis for for clinical development, and also to attract industry, because all these tools are already available and the industry partner does not have to spend money on registries or natural history studies or whatnot. So having all of this is really, really valuable. It costs money, true. So what would be the priorities? The priorities would be things that are maybe started with things that are less expensive, which is serving the community, understanding the disorder better, putting in place a disease concept model, which says what the priorities of the community are, versus spending a lot of money on looking for biomarkers, for example. But if you have the resources and you are able to do it, then everything on the bottom of the pyramid here could be executed by patient advocacy groups. As I said, registries, natural history studies, disease concept models, starting to think about clinical endpoints and the clinical endpoints that are meaningful in case your disorder has never been in clinical trials before, and in case you have some unique features and unique clinical symptoms that haven't been measured before. Helping the industry partners and regulators understand what these endpoints are and how they're measures is really important. And I think that patient advocacy groups are best equipped to actually design those clinical endpoints. Looking for biomarkers, is there anything in the blood, in the CSF, in urine that could be any imaging or EEG biomarker that could be adopted for early response to therapy? That's very helpful. And then if you go up the pyramid, looking at what Armel showed in her slide earlier today, having cells, having organoids, having a biobank that also came up this morning, and having animal models, the more the ones that could be used for drug development, the better. And then on the right-hand side, I would say that's where industry comes in. We can guide proof of concept. What should it look like in vivo, in vitro? What are the IND enabling studies that are needed? What toxicology studies are needed? What would a clinical trial look like? How many patients will we enroll? What would be the endpoints? How many visits does the patient need to have in order to get the therapy and to measure the response to therapy? And then the choice of the modality will be based on the expertise of the company. So you'll go to Everyone or in Lorem or La Jolla Labs if you want an ASO, or you go to other companies for a gene therapy and discuss with them what is the most suitable for your gene and your genetic mutations. Clinical development assumes that many of the disorders that we're working on have had limited previous clinical trial exposure before, and that we would like ideally to work on disease-modifying therapies. So, as I said before, small molecules not likely to be disease-modifying. ASOs, gene therapies, gene editing, of course, will likely be disease-modifying, and an intervention will change the fate of the patient and the trajectory of the disorder. The challenges are to validate novel endpoints, or to modify known endpoints to be suitable for your disorder. The endpoint has to be sensitive enough to measure change. And sometimes when a population is very diverse, it's hard to find something that could measure across a population. The studies are small. In most cases, especially in gene therapy, we do not want a placebo or a sham control, because it just doesn't make sense to bring a very fragile patient in and have them go through the whole procedure of injecting something into their brain or something into their liver, and then they're not getting anything, and then bringing them in again when they are eligible for treatment. So actually the regulatory agencies are more open today to running especially gene therapy or viral vector studies without a sham control or without a placebo. And then sometimes you have advisory boards and you hear from the patients one thing, from the clinicians a second thing, and from the regulatory agencies a third thing. How do you collate everything and make sure that you're going into the clinic with something that will satisfy the needs of the patients on one hand, but actually could be approved on the other hand? And again, biomarkers, that my head of regulatory brainwashed me with a biomarker part. quickly on disease concept models, we start with caregiver surveys, interview families to understand what they would like to see changed. We go through a medical chart review to see what information is already available without needing to go into a prospective trial. We then interview the families again in a more qualitative manner. So go deep into interviews, understanding what a day in their life looks like. And based on all this information, we design a natural history study. And all this is put together to get a conceptual model that will guide us on what the clinical trial would look like and what the endpoints are. And all this is done in parallel. So nothing should be done isolated. All this should be parallel tracked, of course. So whatever you can do and jumpstart in the beginning, you're better off later on when you want to get into the clinic. And just going back to the Mazi slide, I wanted to show that the interaction between academics and family groups is critical. And I think it will become even more critical if less grants will be available from the NIH, or there will be restrictions on federal money. A lot of academics will be relying on patient advocacy group money to do work in their labs. And I hope this won't create more conflict of academics needing to publish and to get tenure while families really want to get drugs into the clinic and treat their loved ones. It's a joint effort. There should be alignment on goals and the same sense of urgency. And it's a process, and especially for young investigators that never worked with patient advocacy groups or families before, it is a shift in thinking. And families need to understand that usually when somebody went to academia to head a lab, developing a therapy was not the first thing that they had in mind when they signed up for the academic career. And the collaboration is based on one hand, the scientific expertise of the academics and everything that they bring to the table, with the acknowledgement of the fact that the advocacy groups will bring a lot of expertise that otherwise they will not have access to, the academics will not have access to if they don't work hand in hand with the advocacy groups. So in summary, it takes a village to develop a therapy, and alignment, collaboration, synergy, however you want to call it, it's critical for this whole process. And we need solid biology, very smooth handovers between academics into biotech, with working hand in hand with the advocacy groups. So imagine a 4 by 100 race where somebody drops the baton and loses the race because of that. We don't want that to happen. We need to parallel track, so run everything as smoothly as we can at the same time, and collaborate. So thank you, and happy to answer any questions you may have.
I have a microphone that helps too. And any questions? There's one.
I have two questions. So my first question is about the ASO modification, and both Jeff and Nadina sorry, mentioned in your presentations. So there are various types of modification, including like LNA and MOA, or MOE, or gamma. So I want to hear your perspective or rationales, how to choose the modification, pros and cons for each different type. So that's my first question, ASO modification. The second question is more for Nadina. So would you mind to elaborate a little bit more regarding how Lauren's team access the off-target effect for your ASO hits?
the regulatory experience, at least the feedback that we're getting from some of the ex-US regulators, is the commonality of backbone is something that they would actually really like to see if they're going to approach this more like a platform approval. It doesn't mean every drug just gets automatically approved. It just means they can truncate the amount of data that's needed for the review. And they've at least signaled early on that things like LNAs, which does create better stability, they actually mentioned that in their feedback to us, that they would rather not have different backbones, that would require a different level. So we're somewhat erring on the side of let's just get through what we can, even though I think we would really enjoy the opportunity to put an LNA in there. But that's just kind of one example where their feedback is that the commonality of backbone gives them more of a predilection to say, yeah, this is a truncated review based on the amount of repeatability you can show.
Yeah, I'm just going to add to that, that essentially there are a lot of years of experience now where we kind of have nailed, well, I'm going to use the word nailed, but I'm not sure if that's the correct word, the chemistry for gapmers and also for splice switching ASO. So for example, essentially for that, we know that for research purposes, so if you're in a lab and you want to use LNAs, that's completely fine. For clinic, we do know that these modalities are a little bit more toxic. So in the terms of the backbone, phosphothiorate is something that we routinely use, and especially in a gapmer, I'm going to go technical here, in a gapmer for CNS, a mixture of phosphothiorate with a phosphodiester is the one that we have the most knowledge on and the most results on. 2'-methoxyethyl modified on the RNA. This is also something that we have a lot on. And, also the splice switching, and Jeff can elaborate more on that, that is fully modifying an 18-mer, and essentially the backbone is mainly phosphothiorate. This is also something that has been now well established. So I think that they are, and the reason, as I was saying, that we use the chemistries is you want a drug, an oligo or whatever, to last long, to have less immunogenicity, right? To have high affinity for your RNA, which is essentially your target. And lastly, you want the best pharmacodynamic properties. So all of this together, this is basically what is on your tick list in order to define the best chemistry. And these chemistries are pretty much now defined at least for the ASOs.
Yeah, I mean, 250,000 people dosed with that chemistry. So that's a platform that has been tried and true. It's tested in many ways in the clinic. So that's basically it. That's where we focus. And at Ionis, we did a lot of work around, understanding the biology first and then teasing out the chemistry if we have to. So the idea was to find the target space, and then if you have to do a chemical SAR around a particular spot to tease out toxicity or improve potency, then that's kind of the strategy we took. I mean, for rare disease, it's just the clinical proof of concept. What's the fastest or the cheapest way to kind of get through the path? And if you change that chemistry, you throw a big wrench into the equation.
Okay. More questions here?
The question for Adina or Jeff. My understanding is that this, for ASOs, the delayed tox is something that's, it has a lot of time to the development, but is also a little bit hard to predict from the sequence. Do you have any idea, what may be leading to that? And are we getting towards, some predictiveness for that?
It's a great question. It is a really good question. So I think and I'm going to share my perspective, there are twofold to this question. There are aspects of it that we can predict, and there are aspects of it of like some motifs in sequence that, you can build into your algorithms and predict that some of the things could potentially cause toxicity. And especially you would see that in the tolerability, which is, a smaller scale tox study before going to GLP tox. So that is something that, the more learnings we have, yes, we can fish some out and just, either cytotoxicity or immunogenicity. These are the two main things. But there are also some other aspects that at the moment we cannot predict them. So at the moment there is a molecule that can go to tolerability. And that's why I think from my perspective, these toxicology studies are very, very important. Yes, they're costly and they're lengthy, but this is why they're important, because at this moment in time, there are some that we can predict, but there are some that we think that is not immunogenic, is super safe, not cytotoxic. I can't see anything wrong with it. Cells tolerated just fine. And then we go to rats, and then they are either, acute FOB or delayed FOB. So there are things that we cannot predict. There are things that, and at least at Lorem, we really try to look at the data and see for any commonalities, because obviously having less of these leads into tolerability, we don't want to lose any good drug. But at the moment, this is a work in progress. So some we can predict, some not.
Yeah, and I'll just add to that it comes down to the data. I mean, there's some really good models that Ionis has developed, and but having delayed tox data is harder and longer and more expensive, and that's really what it comes down to. Once that data is there, then yeah.
Okay, we have two questions from the outside. The one is a more general one. Is there a centralized database that collects all the genetic data from rare disease patients around the world?
Unfortunately, no. We don't even have a centralized database in this country, but there are a number of countries across the world that are putting in, I would say, yeoman's work to try to create databases that are largely newborn screening. The Genomics England database is, I think, well over 100,000 at this point. Emirati and the Qatar genome projects, actually, I think Emirati alone is almost 1 million full. In Qatar, I think, is somewhere around 250,000. There's about 4 or 5 states, I know California is one of the states, where there is upwards of tens of thousands of newborn screening with whole genome sequencing, but there's not even a database in our country to do that. And I think, as I maybe alluded in my talk, wow, that is a fantastic effort for us to do for so many different reasons. And I think it opens up an entirely new ability for us to target disease. There's going to be issues with that too. I mean, one of the things that I noticed at Arab Health just a few weeks ago in Dubai is that they are incorporating newborn screening with genetic counseling and premarital counseling as well. And that's a concept that is going to be something to tackle as we get closer and closer towards having this evidence of gene and the number of mutations people may carry.
So if I can comment, there are, is that okay if I comment? I don't know if it's.
Yeah, absolutely. Go ahead.
There are, the issue is with data privacy, obviously, and different countries handle data privacy very differently. The Scandinavian countries have really good genetic databases, as the countries that Kent mentioned. But for example, if a company wants to use them, they have a very hard time accessing this information. If only mutation information is needed, then ClinVar is probably the best source for genetic information that's totally de-identified and just has information about mutations in genes.
Okay, thank you. I have one more question online, and then maybe one more in the room, and then we have to move on. Oh, there are two maybe. So one that came in online is, are there any challenges for ASO administration that goes intrathecal?
Yeah, I mean, there's always going to be challenges anytime you're in that space. I'll just say, from a research perspective, it's challenging from the perspective that on the one hand, it's hard to find people that can actually do it well and consistently. So if you think about just actually doing that in a research setting, and the consistency is a challenge, think about it in an actual treatment setting as well. And that's always going to be the concern with any kind of intrathecal administration. So the idea of accredited treatment centers, places that actually are defined in being able to do this, there's multiple disciplinary aspects of those treatment centers. What we see in research is what we're going to see in the real world settings as well. But it's not an ideal space. That's where, for me, I mean, we're intrathecal administration, of course, but I'm really hoping that there are companies that are going to come along with different modalities of delivery. I think it's going to take a while, though, unfortunately.
Yeah, and just to add to that, that's why I was saying that having really, really good physicians and really good treatment centers, as you were saying, that they're really equipped. I mean, of course, you require anesthesia a lot of times because of the specific challenges of these patients and dosing them intrathecally. However, having said that, it is a route of administration that is well established, and unfortunately at the moment, until we have another, exactly, and a safe one, and until we have another delivery, this is.
We have one more question.
Yes. Thank you. Well, first of all, thank you all for the session. It was fantastic. So my question is on behalf of my husband, Bernie Ziprich. So Kent, you talked about the need for a better regulatory pathway, but NLORM seems to be finding success with the current FDA framework. Can you help us understand the nuances better and some of the changes you're hoping to achieve?
Yeah, it's a great question, because I think in some respects, folks may see as NLORM and Everyone Medicines and others as sort of in competition. We don't see it as competition. I mean, I think the one stark difference between what NLORM's doing versus Everyone Medicines is that we're actually just going down the route within Europe to create a brand new pathway. And I think NLORM is actually going with the existing pathway, but using that totality of data to also negotiate a new pathway while going down the existing one. Our attempt is to really try to create as much data as we can in this sort of compendium approach. And the thinking behind it is that the timing right now, at least in Europe, seems to be very favorable. They clearly understand you can't do a clinical study. The question of GLP tox, quite honestly, even if they require that, if it's a GLP tox that's reasonable and that we can figure out a way to make it even somewhat affordable, that's okay. But right now, there's not a pathway that says it's not only approved for marketing authorization, but then it'll get reimbursed as a result. I think we're all kind of trying to bring as much as we can to bear to give them the experience to say, yeah, this is how to do it. And this is the way to go about it from a research perspective. And it's not just for research, it's for treatment and approval. Reimbursement's the key here.
All right, thank you for all the speakers for this section. Hello, can I ask everybody to take the seats? We will have the last speaker is starting from a different country. We better keep our time. So we already hear a lot of great talks from patient advocates, also from researchers and biotech thought leaders. So our last section of today's event is advancing ALS research. ALS is a neurodegenerative disease. The full name is amyotrophic lateral sclerosis. So it's affecting the motor neurons in the human bodies. It hits usually in the late stage in people's life. And today we have a few influencers in the field. We have 5 influencers in the field to discuss the breakthroughs in mechanism and therapies. So before starting the section, I'd like to play a short video from a friend of our company, and also who is one of my most admired ALS fighter. Close the light. So his name is Jaime Lafita. So a native of Bilbao in Spain. Jaime was diagnosed with ALS in 2016. In response, he founded the Della Candela Foundation in 2019 to raise awareness and funds for ALS research. The Della Candela is the Spanish phrase with the meaning of fighting hard against ALS. Demonstrating the remarkable resilience, Jaime has undertaken significant challenges, including a 600-mile bike ride from Spain to Brussels, Belgium in 2021, where he was received by the President of the European Parliament on World ALS Day. In April 2024, he completed a 9-day, 400-mile tandem bicycle journey across Death Valley in the United States. I was honored to join his Death Valley ride, leading a Chinese cyclist team. After the grueling 400 journey, and we ended our ride in front of El Capitan, which is the symbol for, conquer the most impossible task. And then we celebrated our accomplishment with the leadership team from ALS Network, and where I met the leadership. Sherry and Cheryl is today here as joining us. So I consider this a small reunion. And through this endeavor, Jaime continues to inspire and mobilize the effort towards finding a cure for ALS. And this is a video he shot for us.
Hi, everyone. Hi, Wei. Hi, iXCell Biotech team, and thank you for thinking about us for this video. This is Jaime from Dale Candela in Spain, fighting for a world without ALS. We had the pleasure to visit your company and Don Cleveland's lab when we joined forces for the Death Valley Challenge, as a metaphor for the valley of death that research projects need to cross to become a drug in the market. We wanted to raise awareness about this valley and also to show that it can be defeated. And we wanted to encourage and pay tribute to you, the researchers, and the painstaking work that you do, persevering even when frustration creeps in and results resist to show. We need you. In our case, the challenge helped start ALS Research program in our Basque Country, for which we feel proud and filled with hope, because research is the only way to beat ALS and any other disease. So the more research, the better. Awareness is very much needed too, and that's what those of us who are not researchers need to do. From the Death Valley Challenge, a documentary was filmed and we screened it 2 weeks ago in our city of Bilbao. We hope to show it over there once we finish the English version. I want to end by thanking you again and expressing once again that we will never give up. Hi.
All right, without further delay, let's continue, because we have another ALS fighter today with us, which is our next speaker, Bernie Zibrich. Bernie is also an ALS patient. He is an accomplished healthcare innovator, strategist, and entrepreneur. He was diagnosed with sporadic ALS in 2020 at age 32. He founded Project Mosaic, a program aiming at bringing a new class of innovative ALS disease models and an AI-based platform for conducting phase 0 technical trials in a dish to market. Bernie and his team are partnering with researchers, industry partners, including iXCells, and ALS nonprofits, including ALS Network, during this initiative. Beyond Project Mosaic, Bernie is also a NEOS certified ALS research ambassador, a member of the ALS Association Board of Trustees, and is a recognized thought leader on ALS scientific strategy. Bernie lives in New York with his wife, Marie Zibrich, and their young son. We are honored to have him and his wife joining us today, and as well as his Project Mosaic team members, Eugene and Tusha. So because of Bernie's condition, Eugene Brandon is going to be present the work and the mission about their initiatives from lab to market, decoding the ALS mosaic to enable precision medicine at scale. Eugene. Oh, really? Oh, sorry. Sure.
I just want to say that it is such an honor to be here, and I've so loved hearing all of the other presentations today, and 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 maybe as a way of introducing what we are hoping to do with Project Mosaic. One thing that is incredibly exciting about about ASOs and the precision medicine techniques that we heard about before is that they allow us to target rare diseases at at the source, and 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 as many of you probably know, and as we will discuss in a moment, one of the big challenges with not only ALS but other neurodegenerative disorders like Alzheimer's, Parkinson's, and 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 FTD is that many cases don't have an obvious genetic target to treatment. So a big thing that we are trying to do is develop ways of being able to identify both targets and promising drugs that might work for subsets of patients with these diseases. And the last thing I'd like to say before I hand it off to Eugene is that one thing that I love about working in this field is mission driven and collaborative. So many people are, and I'm so excited that we will also be hearing from Dr. Donna Cleveland and Dr. Justin Cheetah, because a lot of the science that we are now trying to build on and scale. We are a big debt of gratitude to their research as well. Eugene.
Thank you. Thank you, Bernie. I want to thank iXCells for inviting Project Mosaic to present today and for having a session on ALS. As Bernie was just pointing out, it's pretty interesting that the rarest diseases, the gene is often known, and then you have to address it in that way. And here we're almost coming in as one of the least rare diseases. meaning there's a prevalence of 30,000 in the US, but an incidence that's much higher, equivalent to multiple sclerosis, for example. And in the vast majority of cases, we don't know what the gene is, or if there even was a gene that was involved in the source of the ALS incidence or progression. So here's the team for this is a early stage in this operation, the Project Mosaic, but we're launching fast, as you'll see in the presentation, and want to acknowledge Bernie for all the work he's done to get this to where it is today. I first met Bernie a few years ago when he joined the board of the ALS Association, and he started telling me about this idea of neurobiopsies for patients. And at the time I was a little skeptical that this could be something that would be sufficiently effective and sufficiently cost effective to really attract investigators and investors. But now things have changed. There's the technologies have evolved, and the landscape is different, and there's just things are accelerating. It feels like the whole field of ALS has been accelerating over the last few years, and I think now is the time that a Project Mosaic is actually really needed. So we'll talk about that right now. So because these are Bernie's slides, and I just joined the group, I'm going to read my talk, which I admitted to Don, it's a little bit embarrassing. It's a little unorthodox for scientists to read a talk, but I think that's what I need to do this justice. So what is the vision? What is the motivation? And neurodegenerative diseases are tragic, and everyone is different. Everyone is different. And that's kind of the recurring theme here. What if we could cure devastating diseases like Alzheimer's, Parkinson's, or ALS with treatments tailored to each patient's unique biology, unlocking a future where precision medicine is what transforms lives? So this is obviously a highly ambitious goal, yet we see a path which firstly involves creating an in vitro avatar for each person, which we'll talk about now. We're extremely excited to be embarking on this journey. So where are we today? Let's consider the current state of affairs for neurodegenerative disorders. There are millions of people. This is not rare to have a neurodegenerative disorder. If you consider Alzheimer's with nearly 7 million people in the US, Parkinson's over 1 million, and the other neurodegenerative disorders combined, this is 10 million people in the United States, costing approximately, by some estimate, $650 billion. And this is largely carried by the Medicare, Medicaid, and insurance, but also importantly, families. This is one thing that we come across a lot with the ALS Association, is that families are really taxed financially dealing with their disease. These neurodegenerative diseases, adult onset neurodegenerative diseases, are for the most part sporadic, with no particular genetic cause or even environmental cause identified. So this has been plaguing the field, in particular ALS, for forever. And it's still something that we think we can start to ferret out, as research continues to unfold. Tragically, there are no effective drugs to slow the progression of these diseases for sporadic ALS. And Don will talk a little bit in a minute, I'm sure, about the antisense oligonucleotide for SOD1 ALS. And on top of that, there's a high failure rate. So the rate of drugs not making it to market for neurodegenerative diseases is 10 times greater than for other CNS disorders. So this is really a situation that's screaming for a new approach. So let's consider why drugs are failing. Firstly, there are numerous potential causes of disease, and in many cases, there may be several causes interacting to initiate and propagate the condition. Thus, testing a drug addressing one specific molecular target may be insufficient. Even identifying a target may be difficult. And importantly, the course of disease and the nature of disease can be very different between patients. This heterogeneity is seen in all of the neurodegenerative disorders, but ALS is particularly notorious for it. Typically, it's said that the prognosis is 3 to 5 years with ALS, but from what I have observed with my ALS friends and colleagues, admittedly, this is anecdotal, there seems to be at least as many exceptions to this prognosis, with some pals dying in less than a year from symptom onset and others living a decade or more. Clearly, a one-size-fits-all drug development program is not working for neurodegenerative diseases, and given the heterogeneity, maybe we shouldn't even expect it to work. Despite the heterogeneity with neurodegenerative conditions, research has revealed a common feature across these diseases, and especially prevalent in ALS, which is loss of function of a protein known as TDP-43. TDP-43 is an RNA handling and processing protein, which generally operates in the nucleus of the cell, but in many cases, including over 95% of cases of ALS, TDP-43 protein is found mislocalized to the cytoplasm. And in the cytoplasm, it is prone to aggregation, becoming insoluble and dysfunctional. So as we consider the platform we are building at Project Mosaic, we will pay special attention to TDP-43 loss of function as an important feature. So what is Project Mosaic? Project Mosaic aims to transform the way we view neurodegenerative disorders and revolutionize treatment development, driving more precise and effective therapies. What do we mean by this? Well, let's consider cancer by analogy. 100 years ago, cancer was considered as a single disease which can affect different organs and systems, but which was really best treated with blunt tools such as radiation and surgery, with obviously limited efficacy. Nowadays, we know so much more. For example, within breast cancer alone, there are at least 4 different forms, which can have very different prognoses and susceptibility to treatments. Thus, you will not be surprised that in cancer research, drug screening can include testing on hundreds of different patient cell lines at a time. The PRISM lab at the Broad Institute is a great example of the kind of initiative and represents an effort which Project Mosaic may seek to emulate. The PRISM lab has over 900 cell lines from different patients and cancer covering different cancer types, with extensive clinical and biological information associated with the lines. Hundreds of collaborators are partnering with PRISM, including over 30 biotech and pharmaceutical companies, reflecting the incredible opportunity to gain critical knowledge using such a powerful system. Project Mosaic aims to do the same for neurodegenerative disease. So why ALS? Obviously, Bernie and I and others have a personal interest in ALS, but at the same time, it represents a great starting point for this approach. That is, a great deal of scientific effort has been invested in ALS, and we already have industry-ready sporadic disease models and commercially ready iPSC cell lines with ex vivo data. As well, ALS pathology is concentrated in known distinct cell types, and the potential for precision medicine to be effective has already been demonstrated with the recently approved ASO to SOD1 known as tofersen or Qalsody. Importantly, recent advances in iPSC-based models suggest that a personalized ex vivo approach is possible. Scientific reports from the Achita Lab at USC and the Coyne Lab at Johns Hopkins have shown disease phenotypes in vitro, including TDP-43 loss of function and the potential for therapeutic intervention. Yet some barriers to adoption remain. These include demonstration of reproducibility and reliability in the industry setting, questions as to the volume of data that will be needed for regulatory acceptance, and correlations to patient disease characteristics, including responsiveness to approved therapeutics or therapeutic candidates. And reports have shown that, like the heterogeneity of the disease itself, there is heterogeneity in how well ALS patients' cells respond to drugs in vitro. These data suggest that there is an opportunity to stratify patients in other ways than just the stage of their disease or the course of their disease, as we seek to tease the subtypes of ALS apart and improve clinical trials. Project Mosaic seeks to move the approach from academic research to commercial utility. And in November 2024, Project Mosaic had a meeting of the minds to explore the possibilities, consider what issues might need to be addressed, and make a plan to get the project rolling most efficiently and effectively. You can see there, there was both drug makers, thought leaders, academic labs, government and nonprofit groups, as well as CROs, including iXCells at this meeting. And here you can see Project Mosaic was fortunate enough to attract some of the best in ALS from academics to industry for this meeting in Washington, D.C. last November. So the moonshot outcome of the meeting is a goal to make standardized sporadic ALS disease models and a PRISM-style AI drug testing platform, and to have these available commercially within the next 3 years. We don't consider this just an aspirational goal. We believe that with sufficient resourcing, it can be achieved. So how are we approaching this ambitious goal on a practical level? We need to start somewhere. So we take the crawl, walk, run style ramp up. Learning to crawl in stage 1 involves getting the cells up and running from a relatively small number of donors and establishing the endpoints and the protocols for this foundation of the project. This will culminate when the systems are internally validated and reliable and reproducible with a track record of success. Stage 2 will involve so-called external validation, establishing algorithms to mine the data and assessing the connections between results in vitro and patients' characteristics. Stage 3 will be the scaled clinical trial in a dish, whereby we have a system of sporadic ALS in vitro, which can be used commercially for research on ALS, for drug screening and drug development. Ultimately, we would like to have a system which can be used to predict which course of treatment is going to be most effective for a given patient. We are grateful to iXCells for partnering with us on this. As well, we have fantastic tissue and data partners lined up in Answer ALS, ALS TDI, and Genius, the data company. So we are on track for beginning this work now. And I'm very excited for Neon Way, and we'll be having a meeting tomorrow to keep this rolling. Indeed, we can't afford to wait. The Project Mosaic system is needed as soon as we can get it up and running. And there are clinical trials in ALS today which could benefit from Project Mosaic's inclusion. So, Bernie, let's finally make neurodegenerative disease treatable. Are you in? Thank you.
Thank you, Eugene. Thank you, Bernie. Yes, we are in. Yeah, so I'm very excited to get something done. So next speaker. So we're gonna welcome a prestigious professor, Don Cleveland, who is a distinguished cancer biologist and neurobiologist, renowned for his groundbreaking research in neurodegenerative disease and cancer. And among his long list of titles and awards, he is an elective member of the National Academy of Science, American Academy of Arts and Sciences, National Academy of Medicine, American Academy of Microbiology. Dr. Cleveland is the 2018 Breakthrough Prize recipient in Life Sciences for his pioneering research in neurodegenerative diseases. Don's major contribution includes the studies on tau pathology in diseases like Alzheimer's and other tauopathies. His team made a major contribution in identifying key mechanisms of ALS, as well as optimizing technology for delivering ASOs to the brain and spinal cord, overcoming the challenge of the blood-brain barrier. These findings directly result in several clinical stage ASO drugs developed by Ionis Pharmaceuticals. This includes Spinraza, the first FDA-approved treatment for spinal muscular atrophy, or SMA, as well as tofersen, one of the first targeted therapy for genetic form of ALS, SOD1. So I think Ionis owe you tons of money. So last summer, I had an opportunity to travel with Don and spend a few days with him visiting several ALS patients and laboratories and biotech companies in China. I'm extremely touched by his uncompromising scientific rigor, genuine care and compassion of patients, and deeply committed to mentoring the young scientists. So I've been discussed a lot about whether iPSC model is a really suitable model for modeling ALS. We have a lot of different opinions, which I like. So with the greatest respect, Don.
Yeah, so thank you for that. And what I thought I would do today, I saw the program and I thought for some of the young folks, even the ones at the corporate positions, I wanted the birth of the use of antisense oligonucleotides, or as I call them, designer DNA drugs, in the nervous system. So from beginning to the next ones. Okay, so that's what I hope I'm going to do today. So, and we started in 2003 with the then ISIS, not Ionis, now Ionis, and recognizing that actually for all of the major diseases of the aging nervous system, all the big diseases, Alzheimer's, Parkinson's, ALS, Huntington's, where there was a gene involved known to be causative or at least contributory, they were all widely expressed, some ubiquitously expressed in every cell in the nervous system. And indeed, it was then this two discoveries. The first one by Andy Singleton and his colleagues at NIH, who discovered that if you inherit 3 copies instead of 2, 3 copies of a completely normal gene that encodes alpha-synuclein, you will develop early onset Parkinson's disease. And that was followed by a similar discovery that if you, because of a recombination error, you inherit 3 perfectly normal copies of amyloid precursor protein, a gene encoding that, you will develop early onset Alzheimer's disease. So too much of a good thing. And indeed, so And a therapy that would be directly on disease mechanism would be just to, in those instances, would be to lower the level of production, because 150% is going to be disease causative. So we set out how can we do that? And of course, for this audience, probably everybody realizes, well, ASOs are going to be a way to do it. But if you have, and I apologize to you, an even younger audience, and they're young scientists, they're going to tell you use an siRNA or an shRNA, and you'll tell them, no, 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 we're going to use something older. Older, the idea that if you can make a single-stranded piece of DNA, easy to make, and then if you can get it inside the target cell so that it can pair with a target RNA inside the nucleus of that target cell to make an RNA-DNA hybrid, we can then exploit the presence in almost all of our cells of a nuclear enzyme, RNase H, which cleaves the RNA, but not the DNA in this hybrid. And therefore, it's a catalytic RNA destruction mechanism that relies upon a single endogenous enzyme that is present in almost all of our cells anyway. And over the years, the medicinal chemists, we heard a discussion of that earlier this afternoon, have modified both the DNA backbone and the 2' and 5' positions of the ribose sugar to produce the kinds of ASOs that are now being used in human trial. But it was actually a very sobering problem, was that if you dose in the periphery, by any mechanism that we know today, effectively 0% gets in the nervous system. So what we're going to do is to deliver it into the cerebral spinal fluid near the bottom of the spinal cord. And that's what you heard this afternoon, intrathecal administration. And we did that after we had failed to use pumps for continuous flow. We went to Medtronic, and their conditions for using their pumps were so unreasonable that we actually thought the whole project was dead. And then I got my big team together, 3 of us. You'll see them in a minute. The 3 of us, and one of us said, well, why don't we just inject it in and see how long it lasts? Because we should have done that right from the beginning, but we hadn't. And that's when we learned that these modified DNAs, that you can, the best of them that you can get from single dose injections, you can get 3 to 4 months worth of efficacy, and that we didn't need pumps after all, that we would just use periodic dosing. Okay, so, and we discovered here in that if you do that intrathecal dosing, and this is work from, first part of it was from my team at UCSD, and now it's Holly Cortesewicz, who is the head of neuroscience at Ionis now, and a second publication from Ionis. Yes, if you dose intrathecally, you can hit almost every region of the non-human primate brain effectively, near the site of injection, but you can see all the way across, although you miss some very deep structures. So you can't get everywhere, but you can get almost everywhere. Okay, so, and the disease, yes, that we started with was ALS. Of course, you've just heard about that. And the founding example here in this paper from 1993, now, wow, 30 years ago, the original proposal, there were mutations in a gene called superoxide dismutase, and those mutations were proposed right here in the News and Views article accompanying the original publication. They were going to be loss of function because mutations usually lead to inacting or partially inactivating the gene. And and we tested that. We made, of the original 11 mutations, we made 6 and tested them. And one of them, this one, with this amino acid modification, has 0 activity as a dismutase. And then we asked, does it cause disease in mice that have 2 endogenous genes? They have normal dismutase activity. And the answer is they develop age-dependent fatal motor neuron disease. And then we removed one of the endogenous genes. Oh, and then we removed both of the endogenous genes. So we have mice with normal dismutase activity, half 0 in the presence of an inactive mutant, and it doesn't change the disease course at all. And that led, after a contentious scientific time, that led to what is now a consensus view that these mutations provoke disease through acquisition of 1 or more toxic properties, not through loss of enzymatic activity, although it actually is quite sobering that in 2025, we do not agree on what the key toxicity is. But if you could just turn the gene down, you'd be directly on disease mechanism. And so we set out to do that. And this is the founding use of trying to use an antisense oligonucleotide in a therapeutic way, taking a transgenic rat that expresses an ALS-causing mutation in superoxide dismutase, and now dosing it, and here near the site of injection into the cerebrospinal fluid, yes, you can, in a dose and time-dependent manner, lower the level of the target RNA from blue to yellow. And oh, look, throughout the entirety of the rodent the rat nervous system. And if you dose these animals, it's a hyper-aggressive model of ALS-like disease from mutation in SOD1. But here's the founding experiment. And if it hadn't been successful, there wouldn't have been a second experiment. But this is it. And we took animals that were either mock dosed or dosed with a saline, and we dosed them at disease onset. And I said it was hyper-aggressive model. They only live about 25 days before you have to euthanize them for humane care. But look, we doubled survival after onset in this animal by dosing at onset. So that, and we thought, hey, we've actually done something. We've really been able to target the actual cause of the gene. And indeed, just in 2023, got FDA approval. Here was the trial. You can see loss of muscle strength in the green curve are the dosed patients. The dashed blue curve is were the placebo patients. And now you can see, oh, look, in the open label extension, 6 months, the original trial was for 6 months, but in 6 months afterward, 25% of the patients got stronger. They got stronger. This was a demonstration that ALS is a treatable disease. And even the placebo patients, when they were converted to drug in the open label extension, you can see they are also following the flattening of disease course. And we now need to follow these even more carefully, but there are anecdotal examples of individuals with mutations that really cause very rapidly advancing disease who have lived much longer than had been recorded by any of the clinicians I know, any of the neurologists I know. So and indeed, 2023 was a watershed year, in my view, for ALS, because there's evidence for SOD1-mediated ALS, and even in a second inherited cause of ALS, mutations in a gene called FUS, that work led by Neil Schneider at Columbia with a series of colleagues, including Sandrine de Cruz, who was here in San Diego at the time. Okay, so now I'm just going to take a further view of what's happened in the ASO space in neurodegenerative disease, and then come back to how can we use that in ALS, okay? So the most frequent genetic cause of the second most frequent disease of the aging nervous system, Parkinson's disease, is mutation in a gene called LRRK2. And LRRK2 encodes a kinase, an enzyme, but these mutations are not inactivating mutations, they actually hyperactivate the kinase. So if you could just turn it, and if you can just turn the gene down, you would be directly on mechanism. And with Ionis and Biogen, that has now gone to trial. And here, thank you for the introduction. Yes, I did get the breakthrough prize. It was handed to me by Sergey Brin, the founder of Google, who, this is very public knowledge, so there's no HIPAA violation here, who is the most famous LRRK2 mutation carrier. His mother is a homozygote. And actually, I missed a huge moment. I did not know he had LRRK2 mutation at the time I met him, but we have gone to trial for his mutation. And indeed, I opened with the idea that if you inherit 3 copies of a completely normal gene encoding alpha-synuclein, you will develop Parkinson's disease. And alpha-synuclein misaccumulation is found almost universally in sporadic Parkinson's patients. And so the idea of turning down using an antisense oligonucleotide to turn down alpha-synuclein, that too has gone to trial with Biogen and Ionis. And I can't wait for the outcome. So I have no affiliation with Biogen And I have been a consultant for Ionis since 2003-ish. So I should have pointed that out on the first. I own not one share of Ionis stock. Okay, so, and then there was one other disease, the biggest disease, Alzheimer's disease. And this is the 46th anniversary of the discovery of a protein called tau. You can see there was this young graduate student and the tau misaccumulates inside the affected neurons in Alzheimer's disease. And actually, it's the best predictor today of cognitive decline. So you probably are aware that there are two major pathologic events in Alzheimer's disease. There's accumulation of a piece of one protein, A beta, accumulating outside the neurons, and there's tau tangles accumulating inside the neurons. And you know, it's like if there's a garbage strike and the garbage is piling up outside your house, that's bad. But if there's a sewage problem and it's backing up inside the house, don't you think that's worse? And and indeed, it went to trial to try to suppress tau. Let's just try to suppress tau. And you can see this is 2023. And indeed, with these ASO drugs, you can you can lower the level in a dose and time dependent manner, lower the level of total tau or phospho-tau for those who think that's the more toxic form. And indeed, but the most exciting part is here. There is imaging, positron emission tomography, PET imaging that can identify the tau deposits, at least some of the tau deposits. And look, here's two individuals who were entered into the trial in the placebo arm. And what happened is that, oh, look, they have these deposits, the reddish yellow stuff, these deposits. And the deposits got bigger in the first 6 months of placebo treatment. In both cases, they got bigger. And then COVID disrupted much of the imaging, but then they were imaged 1 year later after converting, being placed in the drug arm to suppress the synthesis of tau. And look what happened. So the deposits almost disappeared. So that was true for 11 of 12 patients. Again, it's not the imaging was really disrupted by COVID, but the idea 11 out of 12, it's actually really quite good. I could hardly have drawn it better. But Biogen released no information about cognition, but they have pledged to do so this year. So I can't tell you. I don't know either. But anyway, it will be. And now, okay, so we now know we can do this. What about ALS and sporadic ALS? And you heard it before that there's this protein that's typically intranuclear, the brown stuff here inside this nucleus, but it accumulates. Oh, look, it cleared out of the nucleus here and accumulated in this big deposit over here in that cell and another one over here. And so this is the hallmark of actually multiple neurodegenerative diseases, but it's absolutely the hallmark of sporadic ALS. And also the second most frequent the most frequent genetic cause of ALS. That has these TDP43 abnormalities. Now, so we tested, well, what happens if you lose TDP43 from nuclei? And so we suppressed it. We suppressed TDP43 to about 25% of its original level, and we saw that there was an abundant protein that was suppressed even more, and it's a protein called Stathmin-2. We know this is actually an abundant RNA in human motor neurons, and it had been proposed to be required for recovery after axonal injury. And I'm going to show you, it's absolutely required for recovery after axonal injury. So, and we know why it gets lost, but when TDP43 binds to its pre-mRNA and blocks the use of a cryptic splice site. And so that if you lower TDP43 here to about 25% of its original level, Stathmin just disappears as a protein, completely disappears. And we know it's because it gets cryptically spliced and polyadenylated. And indeed, if you ask what happens if you look at the Stathmin RNAs, here you go, exon 5, exon 4, exon 3, exon 2, in a typical RNA-seq experiment, Yeah, there they are. But if you suppress TDP43, looks what happens. You miss the protein coding exons, and you now use this cryptic splice site, which truncates the RNA, making a completely non-functional RNA. So we know that works. And even more so, if you go to sporadic ALS patients and recover the surviving motor neurons from an autopsy samples, you recover them and ask what happens. Well, if you take individuals who died from non-neurologic disease and you look for Stathman, you'll see exon 5, exon 4, exon 3, just what you saw before in cell culture, but look in the sporadic ALS patients, they are all truncated. They're almost completely truncated because of loss of function of TDP43, and that was true in 13 out of 13 of the sporadic ALS patients that were tested. Okay, so, and even more so, we have identified in 2023 that binding of TDP43 at that locus in the Stathman pre-mRNA is absolutely, that TDP43 acts just to inhibit the use, the 3' splice site finding machinery from identifying that cryptic site. Okay, so does the loss of Stathman, is it really a key component of driving ALS-like disease? And so what we did, was Yoni Lopez-Araskin right there. What she did was to suppress Stathman in completely normal mice, really aging normal mice. And when she did that transiently using an antisense oligonucleotide, look what happened. The neuromuscular junctions where the motor neuron and its axon connects to the muscle. And most of us think that in ALS, that's the disconnection is the first step. And here, oh yeah, you drive disconnection by transient suppression of Stathman. And if you suppress it even longer here for 8 months, oh yeah, you have more than half of the neuromuscular junctions are now disconnected. So loss of Stathman is sufficient to cause the first steps of ALS-like disease. So, okay, so we argued that. And now you heard about this again earlier this afternoon, and I put this slide back in just to remind everybody. In in spinal muscular atrophy, it's a disease. It's a disease completely of the motor neurons. It affects, and the affected kids have inherited 2 inactivated copies of this gene, the SMN1 gene. But we, almost alone among primates, have a second gene, and it encodes exactly the perfect protein. Oh, but its RNA gets misspliced to make a non-functional RNA, and therefore these kids have both genes are basically inactive. And now, so the strategy was now, can we use an antisense oligonucleotide, which is modified so when paired with an RNA is not recognized by RNase H. And so it's not cleaved, it's simply blocking actually encouraging the use of the correct splice site. And here, if you take cells from one of these individuals, the RNA is almost completely missing as an essential exon. But if you use this in ASO, you can now restore it almost fully. That's the basis for the Breakthrough Prize in 2019 to Adrian Craner and Frank Bennett. Adrian Craner at Cold Spring Harbor and Frank Bennett, the chief scientific officer of Ionis, because for the first approval of an ASO in the human nervous system, a true disease-modifying therapy. Okay, and we're going to try to use that strategy in sporadic ALS. And what we're going to do is to make cells in which carrying, actually this is an ALS causing mutation in TDP43. We put it incorporated into both copies of the TDP43 genes in human cells, and then ask here, by tiling with Ionis across this region, can we find ASOs which will block the use of the cryptic splice site when TDP43 is absent? And the answer is, well, you first ask, can you suppress the use of the cryptic splice site? Yep, you can suppress it in a dose-dependent manner, as you'd expect, with both the blue ASO and the red ASO. You can see they're positioned here. And even more importantly, they restore the correct level of the Stathman encoding RNAs here, starting from 50% of the normal level, bringing it back almost to 100% with both the blue or red ASO. Okay, so we think we have a drug that can restore Stathman, even in the absence of TDP43, is that functionally important? And so to do that, what Yoni and her colleagues did was to grow human motor neurons in these compartmented chambers where there's cell bodies are over here. They have their axons and processes into the distal compartment, and now you can intentionally damage them. You can axotomize them, cut the axons off mechanically, and now ask, can they recover? And the answer is, yeah, they can recover. If they're normal human motor neurons, they just recover beautifully within 24 hours. They regrow those damaged axons almost perfectly. Now, can you now? And here's another example of that using the control ASO. Yes, after axotomy, they recover. Look, the processes recover, looking at either the presence of Stathman or a neurofilament. Now, if you now suppress TDP43, and ask, what happens? Can the axons recover? And the answer is, you can see, no. Actually, the neurons are still there, but they don't recover at all, at least not in the short term. Now the question is, can we use a rescuing ASO to restore the synthesis of Stathman, even though we're not restoring the other products, the other RNAs that are misprocessed when you don't lose TDP43. So we're only putting back one of the misprocessed products, and it's almost completely restores it. So we argue that we have proof of principle here in human neurons in culture that restoring Stathman, even in the continued absence of TDP43, can really make a benefit. Now, one last thing. So it was my group and Kevin Egan's group then at Harvard who in back-to-back papers identified Stathman as a target in ALS since 2019. We achieved last year, we humanized the Stathman allele in mice so that we could test, do we have an ASO that can restore in the mouse nervous system, can restore Stathman synthesis even when TDP43 is not bound to the Stathman pre-mRNA, and we identified that in 2023. We hope with Ionis, we hope to get to clinical trial this year. And I should mention that a small startup, ALS, went to clinical trial with an ASO already 2 years ago in 2023, and we're I want to make it really clear, I wish them the best. I hope their ASO really does in the actual nervous system restore Stathman, and nothing would make any of us, I bet anybody of us in the room, happier than to see a true effective therapy by anybody. Of course, we're even happier if it's us. But okay, so all right, so here's the scorecard of where we are. We know we can use these ASOs in broad and deliver them broadly throughout the nervous system of rodents, non-human primates. We can use them for catalytic mRNA degradation or altered splicing. The best of them have very long efficacies. We know they're certainly commercially feasible, and they can be safe. You have to screen for the safe ones, but you can be. The first approval was in spinal muscular atrophy, 2016. Second approval from SOD1 mutant-mediated ALS in 2023. There are ongoing trials as I indicated, in Alzheimer's disease, lower tau, 2 in Parkinson's disease, one of which we'll hear about. There'll be a readout from the trial later this year. There were also 2 failures, and there were 2 failures in ALS. There was a target, the most frequent genetic cause is a hexanucleotide expansion in the gene C9orf72, and that failed despite lowering C9orf72 as expected. No clinical benefit, and the team, there's an intra-team disagreement about what is the source of the failure. All right, so there was another one in Huntington's disease, 100% genetic disease from dominant inheritance in the same gene. That too lowered mutant Huntington, absolutely, and absolutely had no clinical benefit. In fact, possibly And again, the overall collective effort, we do not agree on what was the source of the failure. Roche ran a trial, a 700-patient trial. They're running a second 350-patient trial, now using even a lower dose of that ASO and dosing only patients that were much earlier in disease. They're fearing that they just started too late, lowering the power to show at real efficacy. Okay, so that's where we are today. And I just have, yes, okay, so who was the we that I described today? And this is the founding quintet of the use of an ASO in the human nervous system. This local neurologist, Richard Smith, was absolutely adamant. Some of you have met him, you know that he's absolutely adamant and he without this, it would not have started. Tim Miller, who was a postdoc with me at the time, co-ran the clinical trial for SOD1. My animal coordinator who did the surgeries to introduce these ASOs, and Frank Bennett, the chief scientific officer of Ionis, and the effort to identify Stathman as a target. A pair of postdoc fellows with me, Yoni Lopez Oraskin, who's here, Zaya Melibin, now on faculty in Israel, and a pair of former graduate students now graduated, and my long-term collaborator, Clotilde Lagier, and her team at MGH. Okay, so thanks very much.
Thank you, Don. It's always inspiring to hear your. So actually, I learned quite a bit about the TDP-43, the proteinopathy when I talked to Project Mosaic team. That's my cheat sheet from their team. So I really learned quite a bit. So our next speaker, so we will have our next speaker, Justin Ichida. He is a prominent stem cell biologist and neuroscientist dedicated to advancing treatment for neurodegenerative diseases. He's an associate professor of stem cell biology and regenerative medicine at the University of Southern California. In 2022, Dr. Ichida joined BioMarin Pharmaceutical as a VP and head of neuroscience research. He's also a co-founder of Acura STEM, a biotechnology company focused on developing therapies for neurodegenerative diseases. His research primarily involves creating in vitro models of human neurodegenerative diseases by converting patients' skin fibroblasts into disease-affected neural cells, facilitating the discovery of potential therapies. Without further ado, Justin.
Thanks, Yahweh. I think my conclusion from that is I have too many jobs, but it's really a pleasure to be here in what feels like the epicenter of ASO technology in the CNS, and I've certainly drank the Kool-Aid for this modality as well. So I'm going to talk about some, as you mentioned, I am the head of neuroscience research at BioMarin, whose mission really is to develop therapies for rare disease. Today, I'm going to talk about some research from our lab at USC, much along the lines of the Project Mosaic talk earlier, in seeing how we could use iPSCs and leverage them to maybe identify points of convergence in sporadic ALS, and also to better leverage the genetic information that might be out there to point us in the right direction. So this was a perfect setup the last several talks, but we definitely think of ALS as a landscape with many different etiologies of the diseases. We do have, some known genetic forms. They might be clustered around mutations that cause instability or misfolding in proteins like TAR-DBP or TDP-43 that are already prone to misfold. On the other hand, you might have mutations in genes that affect the protein clearance mechanisms within neurons, which could also lead to aggregation of things like TDP-43. And then even some cytoskeletal genes, which may encode proteins that underlie the the protein clearance machinery and their ability to function. And I think it's a perfectly good and sound strategy, and maybe the best strategy, to go after these known causes of the disease, maybe with designer ASOs. I think there are many people, or some people, going after that, and I think that has a high chance of success in some cases. One of the challenges is that we have this unknown or sporadic sector of patients where, as you can see, it's the bulk of cases, and we don't yet know all of the genetic causes that might underlie those diseases. In fact, we don't even know that there are genetic causes of those diseases. And therefore, it's been really difficult to try to understand if things that we're developing for some of these rare forms, or testing on models of the rare forms, really would translate to a mixed population like you would see in a sporadic ALS clinical trial. And this is what we're trying to potentially use iPSCs for, is to understand what therapeutic targets might be out there. Are there any therapeutic targets out there that might target a point of convergence with all these forms of ALS? As I mentioned, the genetics is not well understood for 90% of ALS patients, but we know that we can make iPSCs from these patients, and if there are genetic drivers within those patients' DNA, they will be preserved in the iPSCs and the motor neurons we make from them. And if we turn them into motor neurons, they may drive disease. I think that was like the overall hypothesis. And if so, we might be able to learn about what are the differences in different forms of ALS, but also what proteins or pathways could we target to reverse some of the pathology that affects all patients. What we did to start, it was pretty unknown, of course, that iPSCs or motor neurons in a dish that are maybe a month old would ever be able to mimic what happens in a patient's motor neurons when they get ALS in midlife. But we nevertheless tried it, and this is a big graph bar chart here, just showing the rate of death of neurons made from iPSCs from these different individuals. So on the left, the far left, there are about 9 white bars. Those are all motor neurons made from unaffected control individuals, and they had a hazard rate or rate of death that we sort of averaged out to be 1. And then the next set of bars, the brown bars, that's the hazard rate, the rate of death of neurons made from C9orf72 ALS FTD patients. And you can see they're uniformly higher than the average of the controls, which goes along the lines of their genetic lesion in C9orf72 potentially driving their faster motor neuron death. And then the next set of bars are motor neuron survival times from different sporadic or unknown ALS cases. And I think what was very surprising to us was that all of the lines that we tested showed a faster rate of death compared to controls. And then on the far right, there are more sort of rare genetic forms of ALS, including mutations in TDP43, also having the same sort of phenotype in a dish. And so you could also see on the left that if you look at the TDP43 within those neurons, in the control samples, they're all predominantly nuclear. In the C9 and sporadic ALS cases, you see a lot of that TDP43 leaking out and being sort of deposited in the cytoplasm and leaving the nucleus. So this is essentially what one sees to some extent in postmortem tissue of ALS patients. And even though we didn't know that there would be genetic causes of any of the sporadic ALS cases, I think one of the simplest interpretations of this data is that there are likely genetic drivers of motor neuron disease in many of these, in all the ones that we tested. Maybe it's polygenic, and so it's been difficult to really pin down all of the variants that contribute. But this suggests that we may be able to use these lines to understand how motor neurons from different types of ALS patients could react to the same type of therapeutic perturbation. Not to beat a dead horse, I'm definitely preaching to the choir here, but one of the convergence points we may be able to target would be the TDP43 pathology. We like that as a target because, you know, TDP43, the gene encoding TDP43 can harbor mutations that have been shown to cause ALS. And this pathology forms in, you know, 95 to 98% of patients, but it's not, as Don, you know, eloquently introduced, it's not that simple to target the pathology because you don't want to knock down the gene because it has an important function in splicing. And if you try to kind of overexpress it to kind of restore nuclear function, what's been seen in the literature is that slight overexpression can actually lead to more pathology. You sort of get the aggregation that you see here in the cytoplasm. So it's a tough nut to crack, I would say. But I think it's something that we're game to try. And so how do we sort of go after things that might address TDP43 pathology? It might act upstream of it. We decided to do a screen, a genome-wide screen, using lines from a TDP43 patient. So this is an ALS FTD family where they harbor a single mutation in TDP43. As you can see by this schematic, I think one of the theories in the field is that many of the mutations that cause ALS in this TDP43 protein occur in this prion-like domain that causes it to aggregate more than it normally does. And that could lead to the pathology that one sees. But even in cases where there aren't mutations in that gene, it's thought that things that lead to higher concentrations of TDP43, like, you know, deficiencies in protein clearance or other types of stress, could also lead to this type of aggregation. And we thought we could model this potentially in the iPSC neurons as I showed you previously. And so Jasper, Ruben Sigler, and Sharon Tu in our lab started collaborating with Ken Kosick and Juliana Acosta Uribe, who had in Colombia found what I think is one of the largest TDP43 ALS FTD families known to date. So they have a spectrum of different ALS FTD phenotypes, but they all have the same mutation in TDP43. And we made iPSCs from that patient line. And what we did was we generated these spheroids of neurons from that patient or from controls. As you can see on the left, if we look at the known genetic form of ALS, C9orf72 patient neurons, they have a fast loss of their motor neurons in a dish compared to controls. And similarly, the neurons from this TDP43 ALS FTD patient have also exhibited fast decline. So this was kind of a phenotype that we thought we could search for genes on where, if we, you know, with an eye on maybe ASO, antisense therapies in the future, could we find genes that, when suppressed, could rescue this type of degeneration. And so Jasper set up a genome-wide screen using CRISPR technology. And so I think we had done chemical screens like this, 6 or 7 years ago. It probably took us, 5 months to screen 2,000 compounds and pathways. This one took us like 2 weeks to screen all of the genes in the genome. And so essentially what we were looking for was, every neuron got a different guide RNA that suppressed one gene. And then we were just looking for the surviving neurons at the end of the survival assay and to see which guide RNAs were most enriched in those. And that would suggest that the suppression of those genes promoted the survival of these neurons. So we found a lot of things that we had seen before, like PIC5. We had seen things that other people have found in the literature, like nuclear porin genes. We also found a gene called VAV2, which caught our eye, because what we wanted to do was really triangulate this with human genetic data to see what things might really modify disease in humans. And so Michael Snyder's group had just recently published a paper where they essentially leveraged iPSC motor neurons and said, motor neurons are likely the causal cell that's affected in ALS. And so what we should do is really only concentrate, or one approach is really to concentrate on the motor neuron genes and the regulatory regions that regulate the motor neuron genes, and sort of eliminate all of the other variants that are just contributing noise or decreasing our power of genetic analysis in this already sort of rare disease. And when they did that, they found genes that were really enriched, either in the gene themselves or in the enhancer region of the motor neuron gene, enriched in genes that were differential, enriched in SNPs that were differential between people that had ALS and controls. And the top gene that they found where there were either lower VAV2 expression or loss of function variants in the gene that was enriched in controls was VAV2. So what that meant was that regulatory regions of VAV2 were enriched in variants that lowered the gene in controls, and in the gene itself, there were loss of function mutations that were more often in controls than ALS patients. On the right is some data that they pulled from this Answer ALS collaboration, where they made 1,000 iPSC lines from patients, ALS patients, and then made motor neurons from them. What was interesting is that in the motor neurons from that study, they found that the motor neurons from patients where they had lower VAV2 expression had a later age of onset, tended to have a later age of onset. So this suggests that whatever is in the genome, if it causes lower VAV2 expression, they might have later age of ALS onset. So less VAV2 is good to protect against ALS, essentially. We also looked in some existing clinical data from Eddie Lee's group, who had essentially taken brains of FTD or FTD-ALS patients at autopsy and sorted out the nuclei from the neurons and then stained them for TDP43. And so they could actually do the RNA-seq on nuclei that from neurons that either had TDP43 or didn't. And what we found was that in neurons that didn't have TDP43, they had much lower VAV2 expression, they tended to, than the TDP43 positive neurons. So this could be because TDP43 regulates VAV2, but in another published data set from Kevin Egan's group, when they knocked down TDP43 in iPSC neurons, they did not see a lowering of VAV2. So it's not regulated by TDP43. And so one interpretation of this is just that the TDP43 negative, loss of function neurons that survived the latest actually had lower VAV2 levels in the brain. So as I mentioned, we're really interested in this ASO approach for many reasons. I think it's a very rapid way to test a therapeutic target in patients. And there are some other benefits, like you might not knock down VAV2 too much in peripheral tissues compared to a small molecule approach. So as a tool and as a test case, we started testing this ASO approach in vitro. So we made ASOs that suppress VAV2 by around 50%, and then we tested them to see if they could rescue the survival of C9 ORF72 ALS motor neurons, about 6 different sporadic ALS lines, and the TDP43 patient line that we started with. And in all cases, treatment with the ASO dramatically improved the survival of these ALS patient neurons across these different forms of ALS. VAV2, we're still trying to find out exactly how this works, but it turns out that VAV2 can activate multiple sort of GTPases downstream of it. Some of the things, interestingly, that regulates is protein clearance, protein acetylation. And so there may be multiple ways in which this single target is activating protective pathways. We do know already from the graph on the right that each of these different downstream targets of VAV2, when they're suppressed, as you would happen when VAV2 is suppressed, they all can contribute to improved neuron survival. So again, it seems like a single target acting through a multi-pronged downstream protective mechanism. What happens when we look at TDP43? I think one of the things we're interested in is seeing if we are upstream of TDP43 pathology and can remedy that, because like there are dozens of genes like STAthmin2 that when TDP43 is lost from the nucleus also are lost as a result. And so what you'd really like to do is restore proper splicing for all of those targets. And so what you can see here in the case of the C9 ORF72 motor neuron, as we saw before, there's a lot of there's a lot of TDP43 out there in the cytoplasm, but treatment with the VAV2 ASO seems to restore that protein to the nucleus, which is really what you want. We're beginning to now look at STAthmin2, for example. As Don introduced in the last talk, there's a cryptic splicing event that occurs in the absence of TDP43 from the nucleus. And with suppression of VAV2, we can suppress that truncated STAthmin2, that cryptic exon event, and begin to restore STAthmin2 levels. And I think we needed some optimization to look further at that. But I think we're seeing trends in the right direction. We've gone on to see how this might work in an in vivo model. We made an ASO for the mouse that suppresses VAV2 by about 50%. And this is a mouse that overexpresses human TDP43, so very rapidly develops the loss of nuclear TDP43 and the cytoplasmic inclusions that you see in patients. They also start having gait impairment very rapidly. So you can see the black line is the increase in gait impairment in these TDP43 mice without the ASO treatment, and in blue is the treatment of the VAV2 ASO. You can see that's quite a bit lower. And the survival time, on average, they improve by about 50% survival, but some of the animals have lived much, much longer. And we're looking into that. We think they have better biodistribution than some of the other animals in the study for the ASO. So and then what we also see is that the number of motor neurons is preserved in the spinal cord when we suppress VAV2 in these mice. We also see that neurofilament from the serum starts reducing even in this early time point of the study. This is, of course, a really important biomarker that looks at axonal degeneration or neurodegeneration that we think will be important clinically. So that's one example of using the iPSCs. And then in just a couple of slides, I wanted to illustrate a very similar but slightly different way in which we're using the iPSC data. So in collaboration with Sai Zhang and John Cooper-Knock, they have been looking at the genetics and these rare variants and how they relate to survival of ALS patients instead of likelihood of getting the disease. What's interesting is they found a variant next to a gene called CCDC146, and they predict from the enhancer map of motor neurons that this regulates CCDC146. But the interesting thing is that patients that have this one variant basically survive like that blue line, whereas the average survival of ALS patients is the red line. So they essentially go downhill extremely quickly. And CCDC146 is another hit that came up in our screen, so we looked at that further. What it turns out is that variant actually seems to regulate CCDC146 expression in motor neurons. And so If you look here, the variant that is in the patients that decline very quickly leads to a slight increase in the expression of CCDC146. It also leads to shorter neurites in the neurons made from those patients. If they correct that variant to the other variant, they get much longer neurites. And also this variant that's in the patients that decline quickly, that leads to higher CCDC146 expression. It leads to higher TDP43 aggregation in these cells. And so that suggests that potentially, increasing a variant that increases CCDC146 levels causes very rapid decline for ALS patients. What happens if you now suppress CCDC146? So we made motor neurons from patients, C9 patients and sporadic patients, and suppressed CCDC146 by 50% with an ASO. And that now rescues the survival of those lines similarly to the controls. And then if we use that same mouse model and suppress CCD146 in those mice, you can see that they live quite a bit longer than the negative control treated mice. So, in conclusion, I think that there is a lot of potential for using, familial and sporadic ALS induced neurons to mimic some of the key hallmarks of ALS. I think it's probably not mimicking what happens with, aging of protein clearance mechanisms in humans, but very good at maybe understanding some of the, root causes of pathology in ALS patients. And using a genome-wide screen and convergence with genetics, we found that these two targets might be highly neuroprotective, at least they are in model systems. And so we're looking forward to seeing if we could advance ASOs against those targets. A lot of this work, most of this work was done by Jasper Rubin Sigler and Sharon Tu in our labs, two great scientists. Some of the legacy people have also contributed to this. And our collaborators, Sai Zhang and John Kupernock, have been working together nicely with us on the CCDC146 story. So thank you very much.
Thank you, Justin. We've been hearing a lot of scientific reports. Now we're gonna switch gear a little bit. Our next speaker, Sherry Straw. She is the president and CEO of the ALS Network, formerly ALS Golden West, one of the largest nonprofits in the nation focused on ALS. With a master's degree in public health and another in business administration, Ms. Straw has excelled as a nonprofit leader, community advocate, and public speaker. Ms. Straw has been on the executive teams of several of the largest nonprofits in California, providing critical strategic visioning and effective leadership. Ms. Straw currently serves on the board of two highly respected nonprofits, ALS United and Child and Teen Counseling. I met Sherry the first time at a reception of the Death Valley Challenge. I was impressed immediately by her distinctive and charismatic leadership. She's great at making people cry and laugh. So let's welcome Sherry.
Thank you so much. It's so great to be here with all of you. Thank you to iXCells for having us. And what a day it has been. I've learned so much from all of you. So thank you for that. It's very generous. And thank you for setting that up. So, okay, I need more hands. So here we are. I'm sharing about the ALS Network today. I'm going to tell you a little bit about who we are, what we do, giving you an idea of the patient advocacy part of the ALS ecosystem. And then I'm also going to tell you a little bit about how we interact with industry and what our research program looks like. And you can read our mission right there for you. I'm not going to read it out loud. You've got that. And I do want to point out that ALS Network, it's a newer name in the ALS nonprofit landscape. And you may, if you've been around ALS for a while, especially in California or Hawaii, you probably know us as ALS Golden West chapter of the ALS Association. We did leave the ALS Association a little bit more than a year ago. And with that, we changed our name to ALS Network. Fret not, we are the same organization that has been proudly serving the ALS community for more than 30 years. We are deeply, deeply mission driven. And we are passionate about the work that we do. You will all relate very much to this. Lives are at stake and people are counting on all of us. So we work with a sense of urgency. We tend to be very driven, very nimble. We're not afraid of change. We're not afraid to do something in a completely different way. We're known for being innovative and also very collaborative. People often share that, you know, it's so easy to work with you and we love working with you and thank you for collaborating with us. And we usually feel the same exact way. And, you know, the ALS community comes first in everything that we do. And that is the perspective from which we frame our decisions. We work through 3 mission pillars. I'm going to tell you a little bit about all of them. One is care, one is advocacy, and one is research. But first, let me tell you a little bit more about our scope and scale, if you will. You heard Nyanwe mention that we are one of the largest ALS nonprofits in the nation. We rank around 4th if you remove the nonprofit hospitals and medical centers. We do directly serve more than 2,000 people with ALS each year. We also serve the entire family ecosystem. So if you take that number of people with ALS that we're honored to work with, we're also working with loved ones, caregivers, family members, and oftentimes friends. There are people who are completely alone with ALS until we meet them. We make sure no one is alone with this disease, and we mean it when we say that. To date, we've raised more than $19 million that's been invested in ALS research. More than 50,000 different advocacy actions have been taken nationwide. And we do have 3 physical locations. 2 are in California, 1 is in Hawaii, but we really work across the region with staff working remotely throughout the state and across Hawaii as well. So you're gathering California and Hawaii. Yes, that is where our in-person boots on the ground services are focused. However, because we serve so many people with ALS, we do have programming that's relevant for others who live in other places. These are things where we have the critical mass and other regions don't. So anybody from anywhere can come and participate in those services. And an example, a couple of quick examples to kind of give you, you know, what does that really mean? We have support groups for people under 50 with ALS, unpartnered people with ALS, LGBTQIA plus people with ALS, Spanish language support groups for people with ALS, and so on and so forth. Anyone from anywhere can come on in and find community with us. And as I mentioned, we're known for collaborating across the ALS ecosystem. And, you know, we are the ALS network, but there are other ultra super duper, I'm creating a new ultra super duper rare diseases that mimic ALS. These are people who often don't find themselves in any of the other organizations. So we do also serve people with diseases that mimic ALS. So moving right along, let's start by talking about our care services and what do I mean by that? What is that all about? And again, tying back to, I think it was Yael's presentation earlier about patient advocacy organizations and the power of what they're up to. And the beauty too of what your patient advocacy organizations are doing is we're helping our medical professionals, our research professionals focus on their roles, and we can step in and bridge those gaps so everyone can keep their eyes on the prize, right? So every person with ALS that we're connected with is provided with a regional care manager. And these are people in their communities who know where's a good place to go where I can come in with a power wheelchair and get a haircut. I mean, really simple things, and also very complex things like navigating ALS, which is different, as you've heard, different for everyone. So these care managers provide concierge one-to-one services for people and families throughout their entire ALS journey. And this includes doing things like safety checks in the home. And those safety checks are important, again, throughout the journey as ALS progresses, providing referrals, providing information, social and emotional support, help navigating benefits. This is a very complex medical system that we live with here. And there's a lot of assistance that's needed for people to really maximize those benefits. We know that the out-of-pocket costs for someone with ALS can go as high as $250,000 a year. So really leaning in and maximizing benefits is critically important from early on in the diagnosis. I mentioned those support groups. We host more than 30 each month. And we also have equipment loan programs, and these are really important. This can be very expensive, durable medical equipment and communication devices, either things that are not covered by insurance at all, which is all too common, or also all too common, lengthy delays once the prescription has been written for the piece of equipment. We help loan equipment so people can start accessing it right away, even if they aren't able to get it right away through their providers. We are also proud to host educational webinars through what we call our Ask Me series. They live in a library online, so people who are newly diagnosed can access a webinar from years back that's relevant to them today. So a lot of educational materials and resources. And if you know someone with ALS, or you find yourself interacting with someone with ALS, feel free to connect them to us at the ALS Network. And if they're not in a place where we provide support, we will happily connect them with their local patient advocacy group too. Okay, so advocacy. Oh, my friends, is there ever a need for advocacy? Today, in fact, today is a day you can do. I'm going to invite you to do an advocacy action today because we need you. So advocacy, we're talking about policy and legislation to benefit the ALS community. We're doing this locally. We're doing it statewide in California and Hawaii, and also across this beautiful nation. We really focus on, you have our focus areas up there. We focus on access issues, things where there are policy problems that are creating barriers to access to key medical, and also this includes equipment, funding for research, and of course, generally raising ALS awareness across our legislative bodies. We know that awareness is often the first step, that familiarity breeds affinity. We want people to know more about ALS so that when we have an ask, they will say yes. Related to that, I'm going to invite all of you to find an example of an ALS network action opportunity. If you find us on social media, ALS Network, or sometimes we're listed as your ALS Network, just last week we put out an action alert that enables anybody to go in, you put in some information, it will automatically identify your representatives, provide a template letter that you can customize, and then you push send and it will kick it off to those representatives. The action alert we sent out last week was specifically about the NIH cuts that are proposed that are absolutely devastating to the research ecosystem in the nation, not only for ALS. So if you want to if you've been feeling like I want to do something, research is so important, how can I help? There's a nice, easy way you can send a letter on that very important issue. So let's talk a little bit about research. We're really, as a patient advocacy organization, as the ALS Network, we are focused on supporting every person with ALS who's in front of us today, making sure they have the very best of everything that's available to them, and also taking action to help prevent ALS tomorrow, right? And this means accelerating the search for effective prevention strategies, treatments, and cures. And you heard from the presenters already today that we're not there yet. We're working on it, we're getting there, but we have a lot more work to be done. So how are we accelerating this search as a patient advocacy organization? Through a few different things. First, thought leadership. We need the best ideas, the best minds coming together. We need Don and Justin sitting next to each other and duking it out and arm wrestling. And Don is known for asking very difficult questions. You have a little bit of yes, you do have a little bit of a reputation for that. It's good. It's pushing the field forward. So every year we host the ALS Network Research Summit, bringing together about 250 researchers. It was very lovely to have iXCells here with us this January. And again, putting out what's happening in the field, what's working, what's not working, how can we learn, how can we keep moving things forward. We do also have, related to thought leadership and really encouraging thought leadership, the Fred Fisher Excellence in Research Award is a new research award that we're launching. We plan to announce the first recipients in next January to really help inspire others to be mavericks in their thinking, to take good risks. And then recruitment. So again, your patient advocacy organizations and the ALS Network, when it comes to ALS, we take research recruitment very, very seriously. We would like our researchers, our biotech companies, our pharma companies really focused on science, and we can be a great asset and are a great asset to helping to populate that research that you're doing and get it fully populated faster so that we can get things going. And we also help populate other research opportunities. It's not only clinical trials. There are a lot of other ways the ALS community is participating in research, including, if you don't already know this, there is a national ALS registry that's hosted by the CDC too. So then funding research, I'm going to talk a little bit more about that momentarily, but another way the ALS Network is contributing and accelerating research is by directly investing in it. And we take this very seriously and are proud to do it. And then collaboration. It's going to take all of us. It's a very complicated disease, and we're happy to be collaborating to help accelerate, act as a catalyst to our friends doing this research. We have a world-class scientific advisory committee. Two of the members might be sitting in front of me, who you heard from earlier. And again, we're happy to collaborate with them and have their input and great thinking informing the research that we're funding. We also co-fund research. We're very open to that and love doing it. We co-fund research with ALS United. We're also proud to be a member of ALS United, and it's exciting to be pooling dollars to propel research. We co-fund research with the ALS Cure Project and also with Target ALS. So again, this example of how patient advocacy organizations and nonprofits can pool their dollars to really push good science forward faster. We're at the table for that and oftentimes leading that. So this ties to partnering with industry, and it's important and we're happy to be doing it. how do we do this? How do we go about this? So the first part I mentioned, directly funding research. So we're looking at high risk, high reward, smallish grants. ALS Network in late April, we're launching our first request for proposals, and we will be funding 7 innovation projects at about $150,000 each. It's open to anybody in the United States. So anyone, anywhere, if you have an innovative ALS idea up your sleeve, please submit an application this spring and put your hat in the ring. The other thing that we're doing, I mean, there's one, I'm going to mention one other thing about our direct funding and that request for proposals opportunity. Something that we're also being really aware of, and again, this goes back to the urgency concept, is okay, we want to directly fund research. It's got to be great. I'm not a scientist, so these guys are going to figure out, you know, and the 22 other people on the scientific advisory committee, they're going to decide which are the best ideas that should be funded. But how do we do that quickly? So we're working within a very nimble approach. It's a brief application followed by the people who are invited, make it through the paper application screening, are invited for a fast pitch session. And we're doing this fast pitch with the reviewers. They're face-to-face. We know scientists oftentimes don't get an opportunity to interact with reviewers at all. They'll be able to interact, Q&A, right then and there. The applicants jump off, and then the scientific advisory committee makes a go, no-go decision pretty quickly. And then we will rapidly move from there into contracting, and then we'll plan to issue the funding in the same year as the application is submitted. So again, how can we be additive in this research space and bridge gaps and do it differently? And I want to also mention that you can be a biotech company. You know, you can be for profit. You can apply for funding with us, even if you're a for-profit entity. Again, we want to fund the best science. And in fact, one of the projects we're funding this year ties to Don Cleveland's presentation, where there may be an alpha-synuclein subtype in ALS. So we're funding an innovative company named Amprion, who's looking into that to confirm if there may be a subtype in sporadic ALS related to alpha-synuclein. So then advocacy, education, and consulting ties to community-centric approaches. We are there. We have a community research committee made up of people from the ALS community, people with ALS, caregivers, direct lived experience. And these are folks who can interact with our research partners, who can come in and make sure, is your trial design community-centric? What does a person with ALS think about what you, researcher, scientist, are planning to do? And are you on the same page? We can bring that perspective into the room, and we're happy to do that. And a lot of the advocacy I mentioned, education as well. More than once, I've come into a biotech company or a pharma company and provided a presentation or been on a panel about what is patient advocacy and why does it matter? And just really making sure to help the scientific community understand that. That's a service that we can provide here at ALS Network. We also have found ourselves, we're pretty good matchmakers. We have connected scientists. We're talking to so many of you, and we'll talk to one over here is doing this thing, and we'll realize you need to talk to this one over here. And so let's put you together, make the magic. Okay. And now it's up to the rest of you. And there have been some really powerful things that have happened because of that. So we have fun doing that, and also help, again, propel innovative ideas by doing that. And then we also partner with industry as a way for industry to give back. and a way for industry to be in the community that they're working so hard for. And one of the things I learned that I was surprised to learn early on is that there were a lot of scientists in labs who never met someone from the community. So then we have an opportunity to make that different. And there are a lot of companies who have the ethos, who want to make that different. And they're the ones who we see at our community events. I'm going to talk a little bit more about that in a moment. So one way everyone here who's from California can make a difference this year, if you want, I know you all want, like, who wants to help directly fund ALS research? I mean, everybody wants to, right? Everybody wants to do that. ALS can affect anyone, so it's on all of us to be a part of the solution, right? Here's how you can do it in a really, really easy way. If you're in California and you are filing your income tax form, on that voluntary contribution part towards the end, don't skip over it. Stop there. And we're the newest on the list, which means we're last. Not my favorite spot to be in. I'm going to be honest. I was hoping it was work the other way. Newest at the top. Scroll all the way down. We're code, it's line, but they use the word code 447. It's the California ALS Research Network is what you're going to hear. California ALS Research Network. 100% of those dollars will be reinvested in ALS research in the state. So I encourage you all to donate any amount, and to encourage your circle to also donate any amount. So one easy way you can make a difference. And then you can also get involved in, Michelle talked a little bit about this morning. I think it was Michelle. Yeah. Talked a little bit about this morning. Join us in California or Hawaii at an event. We have so many different things that we're up to. And there, we have rides and we have walks and golf tournaments and, you can hike and you can do whatever. We have people who paddle across oceans to do this. We have a gala, all sorts of beautiful things. There's something for everyone. The other thing is you can be creative about how you're going to interact with the patient advocacy organizations you're connected with. And I love the example of iXCells, who participated in the Death Valley ride. And that is a way to get involved. We were a part of that as well. Denali has come up a couple of times in presentations. Denali and Cytokinetics, that each year they host an executive splash where they set up dunk tanks and people can pay to throw the thing and dunk the CEO. And that's a fundraiser and a fun way of getting involved. So we'd love to see everyone here involved. If ALS isn't your thing and ALS Network isn't your thing, that's okay. There's a patient advocacy organization connected to what you're up to, and absolutely being involved in supporting their work. They're supporting your work. We're supporting your work. So you can support the work as well, and also be in the community, right? It's the people with ALS who are making so much of the research possible once it gets out of the lab. So there's also that thing, how do we give back to them? And it really is by being involved. It really is, in whatever way that's meaningful for you. Okay, that's everything that I have. And I just want to say thank you for your work. And thank you for this session. And for our ALS researchers here, thank you for choosing ALS. Thank you for choosing rare diseases. It's not easy work, and we need each and every one of you. So thank you so very much.
Yeah, related to Sherry's talk, we also have a small fundraising box here. So snaps. We have we try to get people involved in your internal team, but compared to what you have achieved, this kind of like sounds like a joke, but nothing is too small, right? So we are having our next speaker. This speaker is dying from a different country, China. So this this event is representing a really global thing, right? So in the morning we have people dying from Poland. Now it's the 8:30 something in China. So in the morning, so we have Longchen Li, a physician scientist dedicated to translating basic research discoveries into transformative therapies. With over 20 years of research experiences and 13 years of clinical practice, he co-founded Rectigen Therapeutics in 2016 to advance his pioneering work on RNA-mediated gene activation into clinical trial. He is a former associate professor at UCSF and professor at Peking Union Medical College Hospital. So I visited his company in Suzhou, China, just last summer with Dong. We were very, very impressed by his innovative delivery and RNA targeting platforms. When I first saw his SOD1 mouse survival data, I almost couldn't believe it because it's too good to be true. It's 200% more effective compared to tofersen. So I went there with Dong in the morning. We spent 5 to 6 hours, very intensive discussion, throwing very, very difficult questions. But at the end of the day, I believe I was convinced this is such an innovative delivery system and also the new modalities. So we hear a lot about antisense. You will also hear a little bit antisense, but it's in a different way to treat ALS. So without further ado, I would like introduce Longchen to introduce his new delivery RNA therapeutic delivery platform called SCAD.
Okay, give me one moment. I'm sharing my screen. Can you share my screen? Hello? Can you hear me?
Yes, we can see your screen.
You're
Okay,
ready to go,
great.
Dr. Lee.
Okay. Thank you very much. And then wait for your invitation and for your last introduction. It's my honor to join this event from let's see. It's not advancing from the beautiful city of Suzhou here. Okay, everything good. Rockgen is a clinical stage multi-platform company founded in 2017 on the siRNA modality or technology. A few years ago, I think 4 years ago, we thought we have the technology, we can deliver duplexing to the CNS. Why don't we tackle ALS with our technology? So obviously, the first target we want to work on is sort of one, and very quickly, this program advanced from a concept, from idea to IND enabling, and to patient within just less than 3 years. So of course, this success was built on the foundation of what Don has done in this field, and and also the many other work have done by in the Anderson's oligo field. And to build on the success of the SOD1 program, we quickly launched another program targeting fast ALS, and this program is now also in we have reached the PCC stage, waiting for advancing this program to IND enabling. And of course, the major focus of our work still is on saRNA. So we have a saRNA program now in phase 1 trial, which is an oncology program. We have another saRNA program. We use saRNA to turn up a gene for the treatment of DMD, and this program is now in IND enabling. As you can see, your Rockgene is really focused on a lot of rare disease. So today I'm going to introduce our SCAT delivery platform, which is enabling several CNS programs, and focus more on the ROC17 program for treating ALS associated with SOD1. So oligonucleotides fall into two major categories, single-stranded ASO and dual RNA, including siRNA and saRNA, which is what we are working on as our main technology. The key difference arises from the backbone modification. So ASOs use uniform phosphorothioate backbone or PS modification, and this promotes strong plasma binding, protein binding, leading to broad tissue distribution and in vivo activity without specialized delivery. So in contrast, dual RNA can only have limited PS modification, their function, which restrict the bioavailability. As a result, effective delivery systems are required for therapeutic efficacy, particularly outside the liver. So currently, FDA approved the siRNA drug designed for liver target delivery using platform like LNP and GalNAc. However, delivery dual RNA to extrahepatic tissues, especially the CNS, remain a significant challenge for dual RNA. So this situation underscores the critical need for innovative delivery platform to enable effective CNS delivery of dual RNA therapeutics. So we know that siRNA is generally more potent than gapmer ASO for gene knockdown, as demonstrated in this example. So the two siRNA targeting SOD1 gene were almost 100 times more potent than the gapmer ASO targeting the same gene in culture cells. And this ASO use in this comparison match this sequence in chemical modification or autopsy. So the key challenge now lies in how we could translate this superior in vitro potency of siRNA into effective in vivo activity. To address this issue, at Rockgene, we developed the SCAT, which means Smart Chemistry Aided Delivery, and it's a simple and efficient delivery system. SCAT leverages the self-delivery property of ASOs through a unique design that links a single-stranded oligonucleotide, which we call accessory oligo or ACO, to one of the strands of the dual RNA payload. It's modular architecture allows independent optimization of the payload and the delivery component. When administered locally to confined space like the CNS, non-SCAD achieve broad biodistribution and long-lasting activity. It's compatible with both siRNA and saRNA delivery. So for the CNS, SCAD enable duplex RNA delivery via intrathecal injection with no dosing frequency. Potentially every 3 to 4 months, once every 3 to 4 months. And this technology is undergoing clinical validation and advancing multiple CNS program at Rockgen Therapeutic. So the process of creating a SCAD-enabled drug candidate is very straightforward. You can first opt your duplex sequence in vitro with chemical modification, and then conjugate one of the strand with the ACO. And these graphs show the scarification process and how it retained very well the potency of the 5 siRNAs, highlighting the benefit of modular design. And of course, it's also possible to start with fully chemically modified duplex and the scarified duplex at the design and the screen stage. To evaluate SCAD siRNA in vivo, RNA was administered in mice via ICV injection at 20 nanomole. So after 14 days, it achieved over 80% SOD1 mRNA lockdown across the CNS regions, including the spinal cord, indicating broad diffusion, while peripheral tissue like the liver show minimal activity. So in comparison, the same siRNA without the delivery component exhibited only marginal activity. So we did directly visualize the distribution of size 3 labeled as SCAD siRNA after ICV injection. 3 days after injection, we could see broad and uniform size 3 signals throughout the CNS, including the brain and the spinal cord. So we also evaluated biodistribution of SCAD siRNA in rat by in situ hybridization. 14 days after single intrathecal dose, the Anderson strand of the duplex was detected in different parts of the CNS, including spinal cord, as shown here as an example. So next, we wish to know how long SCAD siRNA tissue exposure and its lockdown activity could last. So mice were sacrificed at the various time point up to 5 months after single ICV dose. In the motor cortex, which is near the injection site, the drug concentration peaked immediately. Here, as shown here. Followed by a slow decline with substantial level remained at 5 months. So here, the red line represent the SCAD conjugated siRNA. The blue line is the siRNA, the same sequence without the delivery component. So you can see the duplex along also showed high and durable drug exposure in the motor cortex. So now let's look at the PD activity. So both the siRNA and the SCAD siRNA peaked at the 2 weeks, but the SCAD siRNA demonstrated significant deeper and more sustained lockdown. So despite the high drug exposure, siRNA lockdown activity rapidly returned to baseline. So this is the site near the injection. Let's look at the deep brain structure. The substantial higher drug concentration was achieved by the SCAD siRNA with peak knockdown activity at 80%, which sustained very well towards the end of the study. At the most distant part of the CNS from the injection site, drug concentration was lower overall than in the brain, but still detectable at the 5 months. So despite the lower drug exposure, peak activity was still reached over 80%, and residual activity lasted till the end of the study. So in the liver, there was an initial high drug exposure, which declined very quickly to undetectable. Similarly, there was a initial moderate PD activity, which returned to baseline in 3 months. Due to slower CSF turnover in large animals, such as monkeys and humans, so the durability observed in mice may translate to even longer lasting effect in monkeys and in patients. So indeed, in the CNS of a crop-eating monkey, after 2 biweekly doses of scattered siRNA, over 80% peak mRNA knockdown was seen in both high and medium dose groups. And this activity was very well maintained over the 70-day observation period. So interestingly, CSF SOD1 protein level continued to decline at the last time point, suggesting a long-lasting PD activity of scattered siRNA. So we anticipate that even longer durability could be seen in patients. So most of the data I present so far have focused on the SOD1 gene. But SCAD technology is broadly applicable to and can target any gene with a duplex sequence. So here, a SCAD siRNA targeting the tau gene was tested in PS19 mice, a model of tau path. 2 SAV dose of 10 nanomole administered on post-latal days 179 and 254 significantly reduced phosphorylated tau protein accumulation in the hippocampus and the cortex to an extent close to similar to the wild type animal. So this demonstrates SCAD ability and the delivery siRNA to deep brain structure effectively, preventing or clearing toxic protein buildup. So next, I would like to introduce our ROC17 program powered by the SCAD platform. So ROC17 is an siRNA targeting the SOD1 gene aiming at treating ALS associated with SOD1 mutation. And this drug is administered to patients via intrathecal injection. And this program has received IND clearance from both FDA and the Chinese counterpart. IIT study has been completed with 6 patients treated, and a formal phase 1 study is currently ongoing. So it's widely accepted and the clinically validated concept that mutations in the SOD1 gene leads to toxic gain of function. So ROC17 works by degrading SOD1 mRNA, reducing the synthesis of both mutated and the wild type SOD1 protein. And this approach helps to decrease the accumulation of toxic aggregates associated with the disease. In several preclinical studies using the SOD1 G93A model, our SCAD siRNA has demonstrated remarkable therapeutic efficacy. For example, in this single dose efficacy study, the model mice were given one single ICV dose at PND 60 compared to the vehicle control. Scattered siRNA significantly sustained body weight and extend animal survival in a very nice dose-dependent manner. So similarly in rat, SOD1 G93 ALS model, scattered siRNA treatment also significantly improved the muscle strength and extended survival. Here we use a control duplex with the same SCAD architecture as a negative control. Here, the blue line, the red line is our ROC17 treated rat, which has similar performance as the white type rat, as indicated by the green line. At the end of the study, the remaining rat simply didn't die, so we determined to terminate the study because we already observed significant benefit of the treatment. As we know that the SOD1 G93 mice is one of the most used model, ALS model for studying therapeutic efficacy, not just for SOD1 ALS, but also for sporadic ALS. And this model typically shows signs of disease onset around the PND 100 days and reaches terminal stage around the PND 160. So in the literature, in order to demonstrate the efficacy, treatment is typically given before disease onset. So to our best knowledge, no one has ever tried to initiate treatment after PND 90. So encouraged by the remarkable treatment efficacy demonstrated in mice and rats study I've just shown, we decided to challenge the status quo and treat the mice way after disease onset, which is more relevant to clinical situation. So we give 2 doses of our drug, PND 126 and 151. And we also include a lot of early treatment group as a positive control. So reproducibly, early treatment demonstrate remarkable benefit in improving muscle function and extending survival, as indicated by the blue lines. As we anticipate, late treatment also shows significant efficacy in both improving muscle function and extending survival, as denoted by the purple lines. So this result gives us a huge confidence that our drug could translate into clinical meaningful benefit in patients who have already developed symptoms. Building on the strong preclinical efficacy data, we investigator-initiated trial, or IIT, in April 2023. And this is a multiple ascending dose study with the primary endpoint being the safety, focus on safety and the ALS function rating score, and the secondary endpoint including CSF SOD1 and plasma NfL concentration. In this study, 6 patients have been treated with the longest treatment duration reaching 40 months. All the participants have completed the stage 1 dose escalation phase, with some now on stage 2 maintenance dosing. So I'm sorry today I can only present the interim result from this study. Because the final study report is planned for the 2025 AAN meeting in April. And these slides provide an overview of the dose regimen for all 6 patients at the end of the study. For the first 3 patients, the starting dose was 60 milligram per dose. So after 3 loading dose, the patient transitioned to bi-monthly maintenance dose. After seeing very favorable safety signal from the first 3 patients, the starting dose for the last 3 patients was increased to 90 milligram per dose. So after 2 loading dose, the patient transitioned to maintenance dose every 2 or 2.5 months. The highest dose reached for the first patient is 180, and for the rest of the 5 patients is 150. The interim safety data indicate that RUX17 is safe and very well tolerated. All the AE reported were mild, including muscle tremor and headache, with no DLT, no SAE observed. Notably, the AEs occurs only after the first few doses. And in later doses, patients report no AE at all. And this is very encouraging that this drug is very safe, even at when it was escalated to a very high dose. So interim biomarker data indicate that the RUX17 treatment significantly lower CSF SOD1 protein level, reaching 50% reduction after 4 doses. The RUX17 treatment also significantly lower plasma NFL levels, reaching over 60% reduction after 4 doses. So very encouragingly, RUX17 treatment appears to have stabilized or even improved ALS functional reading score compared to baseline, suggesting potential clinical benefit. Similarly, RUX17 treatment appears to have a positive impact on patients' lung function, further supporting its potential therapeutic benefits. So building on the strong preclinical and patient data, we are expanding our CNS pipeline using the SCAD platform. So a second ALS program targeting FAST mutation, as I have mentioned at the beginning, is now progressing towards IIT study in China. We also have programs targeting tau for AD and the SARA program for spinal muscular dystrophy. So in summary, the SCAD platform is a very reliable, efficient delivery system for RNA therapeutics targeting the CNS. So RUX17 has shown to be safe and well tolerated at dose up to 180 milligram, with evidence of target engagement and reduced plasma NFL level and stabilized or improved disease progression. So lastly, SCAD enable rapid development of new RNA therapeutic for ALS and other rare CNS disease. Thank you. I'll stop here.
Great. We're gonna wrap up, but before that, we have a Q&A for the last section. So can I ask the last 4 speakers? Also, Dr. Lee, if you can stay online.
Yeah, of course.
Hello. Yeah. I have one quick question for all the speakers, especially my mentor Don. As you already mentioned, that 3 failed clinical trial disease use ASO. One's Taxin 2, another one is the Huntington, a third one is the C9. They are all belongs to polyQ disease. So is that a coincidence that ASO cannot treat the polyQ disease? Or
So I don't think so, but and I mean they're not all polyQ. One, they're all repeat diseases, but the ataxin one, I think was just it's a complete drug failure. The suppression of ataxin was extremely modest, and I would have. So I have no, that was run by ALS Biogen, and I have no part to that. But I looked at the final data; they barely suppressed ataxin, so I think it was just a complete failure. And now with regard to what Dr. Lee just described, it may well be that the drugs chosen, the ASOs chosen, were just simply not effective enough. And this delivery method that you just heard from Ractogen, and I'm going to say it in a very, very direct way, they're gluing an ASO onto an siRNA. And they're now making a quite big drug. But the question, already the ASOs are 15 times the size of a typical drug and heavily negatively charged. But by using the pathway of uptake, the ASO uptake has really made those siRNAs more effective. So I think we should be very open-minded that maybe this is and delivery has always been a key issue. It remains a key issue for neurodegenerative disease. So and improvements should be welcomed. And there I think the failed Huntington trial, the failed C9 ORF72 trial, I think there's the team. The broad team does not agree on the basis for the failures. So, and I'm sure that the audience here is going to have their own views as well about why they failed. Certainly, the drugs were not as active as you would have liked, and there was some evidence that could be interpreted as the drugs had their own toxicities, and so dose dependent. That is indeed what Roche concluded in terms of the redo of the Huntington trial, where they're starting way earlier, patients way earlier in disease, but also with a lower dose of the drug because they were afraid that actually that they were seeing drug-mediated toxicities. And these ASOs, actually, I think for the in Lorem and for the other very rare disease examples, the issues about toxicity of those ASOs, those are very real. The Ionis examples that I'm familiar with, they've made and tested 1,000 different ones every time they went to trial. Their competitors have frequently shown that they do know how to make toxic ASOs. And Ionis was kind of quite pleased that actually, and I should have mentioned in my talk, and then I'm going to shut up. So I meant to mention, especially because representatives of ALS Network were here today, the founding ASO that went into the human nervous system, Ionis was not committed to that project at the time. Their CEO was opposed to working in the nervous system, and the preclinical toxicology of the first drug that went into man was not paid for by Ionis. It was paid for by ALS Association, Golden West ALS, and by MDA. It was Tim Miller and I wrote applications to both of those organizations to provide $1 million of funding, a large amount of funding from each of those organizations in order to enable the first in-man trial. And because that was safe, that then enabled that was the birth of the SMA program. It was the birth of Ionis's focus on neurodegenerative disease, because they really could deliver them broadly and safely. But they work very hard to find ones that are sufficiently safe.
I might just add that in the case of C9, I think that the disease mechanism is more complicated than some people thought originally, and I don't think there was a lot of like listening to the cells and what the cells say about when you treat with a certain approach, which I think is a good lesson for just a lot of the things that we're doing now with these, N of 1 studies or other, ASO studies. And it's really important to test that therapeutic mechanism and make sure that's the right one.
So thanks, everyone. This has been really inspiring to think about. Looking at the heterogeneity and all the preclinical models that you guys have all presented and talked about, there have been a couple paths people have chosen when looking at these. CRISPR-mediated, so you have an isogenic control, versus patient-derived, so you can look across a panel. Kind of tying this to oncology in a multiple hit model. So you need some sort of genetic, then you need 2 or 3 other hits along the path to actually get disease. Do you feel like in these neurodegenerative settings, we need to be dealing with panels of patient-derived as opposed to 1 or 2 well-characterized models where we've CRISPR engineered in a diverse set of mutants?
So certainly many definitely think that, and I have to say I'm not one of the primary advocates that you need 1,000 in order to be able to get an answer. I'm actually worried about a different thing, and I'll let the others talk to that question, but then I add to your question, which is that and I know this is going to be deeply unpopular at iXCells. So the idea we had demonstrated, at least for disease in mice, that SOD1 is a ubiquitously expressed gene, and the cells that drive rapid disease advance are not the motor neurons. They're the microglia and the astrocytes, their synthesis of mutant SOD1. So now if you're taking an isolated motor neuron, is that going to be adequate for, are we reverting to now assuming that the disease is cell autonomous from mutant action within individual cells? To me, that's actually, it seems to me to be unlikely to be true. It didn't say it doesn't play an important role, the synthesis within the motor neurons, but it does kind of, so my question to Justin or others would be, how are we going to, are we going to consider other contributions from those mutants in other cell types other than the primary motor neuron?
Absolutely, we're going to have to.
Yeah, I think that's a huge part of it. It's way more complicated to incorporate those cells in my experience, but it's really important. I think for things like ALS, there's an opportunity where certain patients may harbor genetic lesions and genes that are highly associated with ALS, but maybe not. There's not a strong example of many families, where this mutation is separating with disease, but likely it may play a role in disease. And I think there are many people initiating efforts to make ASOs for those genes, and in hopes that it might help the patients that are just a subset of ALS patients with mutations in certain associated genes. And so there, I think it could be useful to see if indeed, this ASO strategy could work for those patients. This would just be a way of trying to get at the sporadic population in some subsets.
Thanks. I am as someone who 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 is not a neuroscientist, but has been 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 following the 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 literature closely, and and who thinks about this in in terms of in terms of developing developing technology. I I I don't think that we know the answer to the question yet, but but one reason that 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 I find the work that that Justin has done and others like Alyssa Coyne and John Fauci in particular, that that we now have several independent examples of of these of these of these heterogeneous patterns showing up in 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 the in the transcriptome and and I think that a big question that we need to answer is is how much can we learn from that, and that is why with Project Mosaic, at least, we in the early phases we want to know are those patterns reproducible in the same mother neurons, and if so, is that indicative of the underlying biology, and then if it is, we can begin to answer more complex questions around well how much can these models themselves about how drugs might work in different subgroups, and to your point, what can't these models tell us. Sorry, and then that is where we will probably need more sophisticated co-culture and eventually possibly 3D organize, but we have to you have to sort somewhere.
My question is for Justin. So I am very impressed by the results you showed that you did RNA CRISPR-I library screening to identify two target genes, which knocking down both either of them showed a protective effect. So I'm just wondering if you have gone to the other direction, so try to screen like overexpression library to see whether overexpression certain targets also may have a protective effect.
Yeah, thanks for that question. We didn't directly do that, but we do have genes that are sort of negatively enriched when you knock them down. So they sort of kill the cells more when you knock them down, and so those might be candidates, for overexpression. If maybe some of this RNA, small saRNA technology, can work to upregulate genes, I think that would be powerful if that could work. And so I'm kind of curious to see how that develops. Yeah.
Yeah, maybe if I can return to the former question as well. So maybe where a broad, large number of samples, one place there are about 30 diseases for which there's a nucleotide repeat that's causative. Maybe the most famous one is Huntington's disease, but also for C9orf72 in ALS. And there, the different populations, right? The C9orf72 expansions are almost absent from China, but they are the largest cause outside of China. They're the largest known genetic cause worldwide. And there was a long discussion at the nucleotide repeat meeting about the partial penetrance of the C9orf72 mutations, with multiple authors identifying in different populations that it's only about 25% penetrant. So if you inherit the expansion, you have a real chance, but a 25% chance of developing disease and a 75% chance of not developing by age 80 in any ALS-like phenotype. And so so maybe I'll even turn that into a question for Justin or others. The question, if you do look at those, are there sequence variants that can now be protective? And is it in the in vitro making these neurons? Do we see differences in the phenotype in those?
Yeah, that's a great question. I don't think we've done enough lines carefully enough to say if there are differences. I think there's that TMM106B gene that might be a good example of with a certain variant in that gene, you strongly protect against getting FTD with the C9 repeat expansion. Maybe there are things like that for the ALS side. Yeah, I totally agree.
So that's definitely an area where many different cell lines, especially if they were collected like in ways in where they're really comparable, been produced comparably. I would also then I would really strongly advocate that that none of us should be arrogant enough and to think that we know what the right way to do it is, and nor should we think that the perfect should not be the enemy of the good. And so the strategies Justin now has proposed three targets. Those are good targets. I proposed one today as a Stathman, where I think we can do good. It's not by no means perfect. It's not putting TDP43 back, but it's putting back one of the proteins that's required for maintaining innervation. And so this is and there'll be others. And so I would really broadly support the idea that different ideas is the right direction, and don't let Sherry's gonna kind of dislike part of this. the idea that organizations want to band together to identify the best idea that a whole broad spectrum of folks will think is an okay idea, that's probably not the right way to go. You want to see the discoveries, the true discoveries are going to come from unexpected directions, because otherwise they wouldn't be the grand new discoveries. So I would really want to see that a broad spectrum of folks that can defend their strategy. It won't be perfect, but it will be, it's another shot, and we need as many of these shots on goal.
I just want to say that those are the exact ideas we hope will come into apply, and that people like you with running labs who have scientists who may have a real out of the box idea, you know, this RFP gives them a place to get some funding for that. And you're on the committee to help say, hey.
So again, while today's meeting, and I could see it's now just accepted as an obvious fact, the ASOs were initially not at all accepted, including at Ionis. The idea that they could work in the nervous system, that was not believed inside Ionis, and the investment was made by ALSA and then MDA to develop that, and we did not understand that we could deliver these things broadly. We did not think we could. We thought we'd had to use continuous flow. We had no idea we could get 4-month efficacy periods, and as you saw from Ractogen, even better for this fusion of an ASO onto an siRNA. It's just like you have to do it and then critically assess it, of course. But then you applaud success. You're disappointed by failure. But that I think is the real process that has enabled us to get to ASOs are going to be used broadly in the nervous system. We heard it all day, right? I didn't include today. I do frequently, but I didn't today. The original we sent that work for publication, including showing that it was effective in non-human primates. And that was editorially rejected as no contribution by Nature Medicine. It's like then Huntington, we showed we could get partial disease reversal in 3 Huntington model mice. And they said, well, that was nothing either. It's just, and in the end, I got that editor on the phone, and he said, well, he admitted that showing that you could do it in non-human primates, it was remarkable. But then he concluded that it was no intellectual contribution. Because you knew if you turned the gene off, it would be good. And then he said, ASOs will never be used in human therapy.
We're running a little bit late, but I have one last question. Sorry, I took the last one. So for Dr. Lee, are you still there online with us?
Yes.
Okay. So because after hearing your presentation, I am again confirming my belief that the technology can be, because it's targeting, they have very tissue specificity, and also the delivery efficiency is much higher with this, conjugation with ASO. So I think this modality can be very beneficial to some of the ultra-rare neural disorders. So as one of the biggest audience today. So I'm just curious, do you have any strategy how to utilize your technology to help with those ultra-rare patients? I know we have a little bit discussion with Amber-free, SL661, but it seems that we don't have very clear roadmap and path how to move this forward. Partially it's because of the politic barrier, and also part of it because of the financial barrier. So I'd like to hear about your thoughts.
Oh, yes, of course. That's a good question. The last year at the Boston CNS delivery meeting, I talked to some people from the ultra-rare disease organization like Everyone Medicine, and so I propose to them that if they are interested in using siRNA for ultra rare disease, and they can use the scalable nervous system to quickly deploy the therapeutic, because it's just like a plug and play, right? Very quickly. And actually, we are doing this in collaboration with the very famous patient, ALS patient, China in China. We are now designing and the screening allele-specific siRNA for some mutation identified in a certain patient. Yeah, it's quite feasible.
All right, so I guess we are closing. We're gonna have a closing remark by our one of the speakers. So can I ask? Thank you so much for you guys. So I ask you to stay with us a little bit, 5 more minutes. As a tradition of our events, we always think this kind of events and initiatives should be patient focused. So the patient's voice matter a lot. So we always ask one of our patient advocates to close our events. So this year, so it's gonna be Michelle.
Thank you so much. I just want to take a minute to highlight the need right now for cross-disease collaboration, especially right now. It's obviously an extremely uncertain time for the rare disease space, both in terms of federal funding and potential changes at CIRM, as well as ongoing challenges on the industry side. So it's really, and we all know that when funding dries up, rare disease programs and grants are the first thing to get cut. So it's really important for us to all come together and put a spotlight on rare diseases and bring attention to the needs and the opportunities in this space. We need to get the message out in the mainstream that rare is not actually rare, bring attention to these devastating diseases, but also the historic and transformative treatments that are on the horizon. And I'm going to put in a shameless plug for my Zebra Run for Rare, which is not actually planned yet because the zoo is not cooperating very well. But I'm not letting the dream die. As one of the world's leading biotech hubs and the home of the world famous San Diego Zoo with our real life rare disease mascots, I think San Diego is uniquely positioned to host a really large scale, high profile event that can raise funds and awareness for rare diseases. Neonway and I and some other groups were actually in talks with the zoo last year, and we even had the mayor's office involved, but in the end they kind of told us that, well, we can't do any more runs unless it's for organizations with close ties to our board. So if any of you happen to have high-level connections at the zoo, please let me know. Oh, there's also going to be a great event on Rare Disease Day downtown at the Brigantine next week. So it would be great to see many of you there to continue these connections and collaborations. Finally, I just want to say thank you to iXCells for hosting this event. Thank you to everyone and all of you online as well for being here today. Thank you all for caring about rare. I have 100% confidence that life is not always going to be like this for my son Tristan and for our family. And I know that by working together, collaborating to accelerate progress, we can make a difference for hundreds or thousands or millions of other kids and families like mine. So thank you all.